બેન
હેલો
નમસ્કાર બેન તમને બનાવી દઉં
હા મને હોસ્ટ અને વિકાસભાઈને પણ હોસ્ટ co-host
કે મારા કોર્સ નો ઓપ્શન જ નહીં આવ્યું તમે દિશાબેન હોસ્ટ
yeah thank you
હવે હું કઈ નહીં કરી શકું ઓકે
હા
ચલો મળીએ આપણે
હા
યુટ્યુબ લિંક ચેટ બોક્સમાં મૂકી છે અને બાયોટેકનોલોજી ગ્રુપમાં મૂકી છે તમારે શેર કરવું હોય તો
yeah thank you
તમે હોસ્ટ બની જાઓ અને પછી તમને પોસ્ટર બનાવી દેશો.
બીજું છે ને એક ફીડબેક લિંક છે એ મળી છે તમને લેવાની
એક સેકન્ડ ત્યાં ગ્રુપ પર મૂકેલી છે ને
ગ્રુપમાં પહેલા મૂકી હતી પણ ધારો કે ઓલામને પણ કહેજો ઓલા ગ્રુપમાં પણ કે બધા સર્ટિફિકેટ આપવાનું હશે ને એને આ લિંક ઉપર ફીડબેક ફોર્મ છે
ઓકે
બરાબર
હા
આ લિંક ઉપર ફીડબેક ફોર્મ ભરશે એને આપણે સર્ટિફિકેટ આપશું અને ઇવેન્ટમાં એક મેક્સિમમ કેટલા લોકો લાઈવ હતા ને મેક્સિમમ આંકડો જાય ને એ મને ખાલી કહેજો છેલ્લે ચલો બેસ્ટ ઓફ લક
હા થેન્ક યુ સર સાંભળો
Good morning, sir.
Good morning, sir.
ના લ્યા
Uh, Good morning. Good morning, all the participants and the delegates in the virtual room B. I, Dr. Solar Sharma, on the behalf of GBRC, welcome you all in the virtual room B. Now, for the next session, we have a first lecture of Dr. Pratap Mukhopadhyay. Welcome, sir. I welcome you, sir. And
Thank you, ma'am.
yes, sir. Now moving towards the second session of our event at virtual room B, I would like to invite Dr. Pratap Mukhopadhyay for his talk on repurposing the ubiquitous glucometer used by diabetes to adapt to the cost-intensive HIV-1 molecular diagnostic industry. Allow me, sir, to introduce you. Sir is a director of Vobel-based Bioresearch Private Limited. His company offers end-to-end solution for various molecular diagnostics, including nucleic acid extraction, infectious disease detection and quantitation, as well as oncology-based tests, inherited diseases, and genotypic tests. Sir has done his PhD in Science of Molecular Biology from the Department of Microbiology, Bose Institute, Calcutta. And also he is a editorial member of various scientific journals and has published more than 40 research articles in various national and international reputed journals, and also have 2 patents on his name. Sir, can we start?
Yes, so I'll
Yes.
be sharing my screen shortly.
Yes, sir. Yes, sir.
So will you kindly enable the participant screen sharing?
Sir, we have already done it.
Host disabled participant screen sharing, it says. So will you kindly enable?
Yes, sir.
Yeah.
Yes, sir. Visible. Screen is visible.
Right. So moment, I will just, yeah. So a very good morning to everyone out there. It is a pleasure to be a part of this conference. I am Pratap. So the title of my talk is Repurposing the Ubiquitous Glucometer used by diabetics to adapt to this infectious disease industry of HIV. Per se, this particular topic do not fit into the orthodox definition of industrial microbiology. Nevertheless, it does speak about a cross-fertilization of a technology of the lifestyle industry into the infectious industry. And I thought to take the liberty to stretch the definition and talk about this very interesting program. So this program essentially is a walkthrough of a research program that is funded by the Gates Foundation in my laboratory. And this program introduces to my audience a probe labeling technology that brings a high-end molecular diagnostic test of HIV viral load directly into the doorstep of a common man. HIV viral load is a very technology-intensive test, and if this can be transformed into a point-of-care test, it will be a boon to the infectious disease industry per se. So this technology can adapt to resource-limited setting. It can reduce cost enormously. It makes the entire process very simple. And it pulls out the humble glucometer that we know for ages from the lifestyle disease, that is diabetes, to directly into the infectious disease industry. So why HIV? It is just a model that we took to show this technology of transforming a glucometer into a viral copy number machine. HIV, we know, is almost 38 million as on 2019. It has increased now, and the global market for HIV diagnostics is huge. So the most important test once a person is diagnosed with HIV is viral load testing. The reason is very simple. Viral load testing is required to be done very frequently when a patient is diagnosed with HIV and the patient is regularly taking drugs, for the simple reason that even if the compliance to taking of drug is 100%, the patient is on drugs 100%, but still the viral load is increasing. This is a clear sign that the patient is not responding to the drugs. This means that there is a need to change the drug regimen. And this is how the life of a HIV patient is generally extended. So if we miss HIV patient drug resistance profile, the patient loses his life. Hence, viral load testing on a regular basis is important. Now, what is the approach of this technology? So the approach of this technology is we use a oligonucleotide primer that is labeled with an enzyme rather than a dye. Typically, in the days of TACMAN PCR, that is dominating the infectious disease, labeling with a dye is very popular. It's a FAM, VIC, NED, PET, and etc. But here we label the primer with an enzyme. This enzyme is invertase, the one that converts sucrose to glucose. The oligo probe that is designed and labeled detects HIV nucleic acid. Once this particular probe binds to the HIV nucleic acid, this mixture is incubated in a sucrose solution. The invertase linked to the probe converts the sucrose to glucose, and this glucose is detected by the glucometer, which is an indirect way of detecting HIV RNA. So broadly, in a single slide, this is the entire crux of the program. So here is the workflow in a very simplified way. We have HIV RNA that is isolated by a non-electric method, if I may say so. It doesn't use a centrifuge. It is a slide-based rapid extraction process. This RNA is amplified by NASBA. NASBA, we know, is an isothermal way of amplifying RNA, pretty popular these days, particularly in the days of COVID. So once the RNA is isolated, this RNA is incubated with what we say a capture probe. The capture probe is nothing but a small strand of oligo with a biotin on one end. And this particular probe binds to one end of the HIV RNA. Now, once it is bound, this biotin is captured by streptavidin to concentrate the analyte. And then it is incubated with the most crucial sensor probe. This sensor probe binds to the other end of this particular HIV RNA, and this sensor probe is labeled with what I say invertase. So once it is captured, this biotin labeled entire complex is captured to the wall of the tube. The other things are washed out, and this particular captured molecular mix is incubated with sucrose. The invertase that is labeled then converts this sucrose into glucose and makes this entire DNA complex visible to the glucometer. And the glucometer senses the glucose and gives a call. So that is briefly the workflow. So there's a HIV RNA and then it is amplified. So we have a rapid protocol for extracting HIV RNA that uses a small membrane that is put on a glass slide with a bunch of absorbents at the bottom. It is a patent protected technology and does not need a centrifuge. And the RNA that is required for this test is extracted by this process. So once the RNA is there, it is rapidly amplified by NASBA and then followed by detection. So stage one is essentially extraction of RNA, and stage two is its rapid amplification. So the other stages are the stage three, that is binding of the RNA that is amplified from HIV with a capture probe. A capture probe essentially, as you can see just below label 3, a capture probe binds to one end of the viral RNA, and capture probe, incidentally, do have a biotin at one end so that it can be captured to the wall of the tube. So then comes step 4. Step 4 is binding of the critical sensor probe. The sensor probe binds to the other end of the viral RNA, and the sensor probe has the invertase tagged to it. So step 5 is concentrating the entire molecular mix to the wall of the tube using a magnetic, using this magnet-based separation, because we know that streptavidin biotin has a strong binding capability. So once it is concentrated, it is incubated with sucrose 0.5 millimolar, and then the entire solution is dropped on the strip of a glucometer to get the reading. So that is the strategy at a glance. We transform DNA into an quantifiable analyte for glucometer. We essentially went to identify the HIV target to start with. It was the GAG gene, popularly called P24. So that shows the complex HIV map and the region we identified for this particular amplification. It is around, say, around a 1 kb away from the 5' LTR. So that is the region that we have identified to target and identify HIV. So these are the designing kinetics of the capture and the sensor probes. The capture probe essentially binds to the 5' end, and it has got a biotin, a pretty simple structure per se. And the sensor probe, it binds to the other end. It has got a thiol tag, and I will explain why it has got a thiol tag. Thiol, we know, is an SH group, and that is a tag that is there on the 5' end of this primer. Now, this capture and sensor probe, I must say, this particular tag is separated from the actual probe by 12 bases of mononucleotide A, that is 12 stretches of A, the same with the sensor probe. This is required for optimal functioning of these tags. So once this capture and sensor probes were designed, we first developed a bioassay to check whether they are performing as desired or not. So what we did was we took the analyte, that is in this case HIV RNA, we dropped it on a biodyne membrane, nitrocellulose membrane, and we then hybridized the probe with that by the typical classical traditional dot blot technique. And then the biotin was detected by nitroblue tetrazolium and bromochloroindolyl phosphate, NBT-BCIP reaction to generate a non-radio radio signal. It's a colorimetric signal. So we then did a dot blot. You can see there our results that we found. So when the DNA was getting diluted, the fluorescence also was getting diluted. So this was unequivocally showing that the target probe was functioning as desired. This experiment was done on a proviral DNA. So, so then we went on to develop a bioassay for the sensor probes. What we essentially did was we took the probe and started incubating in glucose with different concentration of DNA and measuring the glucometer reading. And we found that around 10 nanomolar concentration of DNA was the point where saturation was reached. So that was the maximum amount of DNA this capture sensor probe combination can detect. The next step was reasonably obvious. We went on to clone the P24 region that we are targeting in a typical PGM-TZ vector. Sequence to confirm the identity, and then we went on to generate what we say in vitro T7 transcription-based generation of RNA. I'll come to the next slide. Now, for a large number of experiments to be done for this project, it was not possible to get so much volume of RNA of HIV from clinical source. So what we did was we took this particular analyte, this particular fragment of HIV, the P24, and then we engineered a T7 promoter on the 5' end of this strand. And the amplicon that was generated was incubated with the T7 promoter and T7 RNA polymerase. And we synthetically generated a large number of viral RNA using the T7 in vitro transcription technology. So this particular process gave us endless supply of the analyte, in this case, the viral RNA, to do all our experiments, the dilutions, the sensitivities, the bioassays, and others, before going for actual clinical evaluation of the system. Now, I may mention that invertase tagged oligonucleotide probes are not available commercially. But what is available is thiol labeled probes, oligonucleotide probes from companies like IDT, are available that are labeled with SH group, and invertase can be linked to an oligomer through the SH group. So we developed a technology or a process in our laboratory to link the invertase to an oligoprobe when it is labeled with thiol. So the thiol is an intermediate motif that is there between the oligomer and the invertase enzyme. And once we have a thiol labeled probe, we are capable of linking that thiol with invertase. So this particular technique is a proprietary, requires a proprietary formulation, and we can label any probe per se with this enzyme. So that typically shows our process of generating synthetic viral RNA from T7 in vitro transcription technology. 16 hours of incubation for this was found to be optimal. The more we incubate, we found that we are losing the transcripts. So the next was measuring the mismatch tolerance capability of the capture and sensor probes. This is important. Why? Because if the probes can tolerate a reasonable amount of mismatch, this indicates that it will be lacking specificity. And therefore, it was imperative for us to record and know what are the mismatch tolerance capability of these probes. So what we did was we took help of a very commonly used technique called the SOEE, or the splice overlap extension technology. A very popular method of introducing site-directed mutagenesis into amplicons by introducing this mutation into the 5' end of each primers, making overlap amplicons, joining them after linearization, after making them single strand. So there is a process that is pretty well laid down in the procedural books. So we followed that and generate a large number of synthetic templates of HIV where there were crucial mutations induced at different points in order to check the mismatch tolerance of the probes. This particular gel image shows how we do it. There's a smaller fragment, there's a larger fragment. Both these fragments are generated by primers that has got mutations introduced into them. And these two amplicons are then denatured, mixed together, and the outer primers used to generate a large amplicon that is on the top. And this large amplicon do have all the mutations that we have introduced into the primers. So this is the challenge. This is the challenge that we took. We took this synthetic mutation-induced templates and used the capture DNA and the invertase probe and kept on taking the data. So that is what we found in case of capture probes. We found that two mutations, a mutation in the C, a mutation in the G, the all different combinations of mutations, none of them were recognized at a significant level by the capture DNA, except for the target that was specific. So this was, I must say, a desired data. And we saw that 100 nanomolar of this probe worked the best. So same experiment was done for the sensor probes. And again, we saw that the sensor probe was equally specific. And this is expected because these probe binding technologies are pretty sensitive, we know, and based on this, a large number of techniques and methods are there based on this probe specificity. So the next, what the project did was to optimize the sensor probe concentration. And we found that 1 millimolar concentration of the sensor probe was optimal. As you can see, there's an S curve that was generated. And at around 1 millimolar, we see a plateau indicating that that is the maximum amount of sensor probe that would be required for the best possible sensitivity under this scenario. A similar experiment for the capture probe. Capture probe required was around 5 micromolar. We see a plateau reaching at 5 micromolar around. And so this was the concentration that was used in all further experiments. This is an image of how we are capturing the biotin labeled complex. Remember what I said, the biotin capture probe has captured one end of the HIV RNA, while the other end has the invertase probe. The entire thing is pulled at the wall of the tube using a magnet. And this particular solution or this mixture of biotin streptavidin and then used for detection. So this is an image that one of my students was particular of putting it there. Incubation time is important. So once we have the complex with us, with the HIV analyte, with one end the capture probe, the other end the sensor probe, how long should we incubate it in sucrose to get the maximum amount of glucose that can be visible by a glucometer? Well, we find it between 4 to 7 minutes. Different concentrations of this target DNA, the analyte, was used for the process, and we found that around 4 to 7 minutes is perhaps the incubation time that is required to generate the maximum amount of glucose that is possible. So the experiment that followed and is currently under process is calibrating the glucometer. So this is the logic is pretty simple. We take a known number of HIV copy numbers as is identified by a gold standard. In this case, we are using an ARTIS kit from QIAGEN. So those are the known copy numbers, and those are the glucose readings, 266 for around 10,000 copy numbers. So that is calibrating the glucometer reading with the HIV copy number. So we see there is a linear range of around 5,000 to 10,000 glucometer reading. The negative control is there on the right. We see that when there is no isothermal amplification, the amount is very less, indicating that the NASBA process is pretty important in this method. So that is how we calibrated. One of the downfall of this experiment, of this entire project, was that the linear range we found was pretty less, indicating that if the viral load was more than 10 to 15,000, the 600 reading of a glucometer was breached. We are working to find out a solution for that. But right now, it appears that if 6,000 is crossed, we can at best say that the viral load is more than 10 to 15,000. But we cannot say exactly how much it is until we do a dilution experiment. So if I may say so, that is one of the things that that has to be overcome for the glucometer to truly take up the complex and sophisticated real-time PCR. So coming to an end of this narrative at a glance, HIV viral load is a very crucial test for the infectious biology, particularly in the domain of HIV. There are many limitations that prevent the scope for frequent testing. Frequent testing is required because a patient must know when exactly the viral load is increasing despite taking the drugs properly. It is that point when the drugs are failing. And if the drugs regimen are changed at that point, the life of an HIV patient can stretch to as long as 20 years. So it is crucial how frequently we test. If the testing is, say, every 3 months, well, then if the viral load has crossed at a particular time between the 3 months, then the patient is losing crucial life. So here, using an existing technology of glucometer that is widely popular in the lifestyle disease domain, diabetes, and a new chemistry where invertase is tagged to probes, there is a potential to help and evolve a home testing technology for HIV viral load in a similar way that we do blood glucose testing. So that is where we are right now. I hope my audience, learned audience, enjoyed the talk. And thank you for your attention. This project is funded by BIRAC, the Gates Foundation and Wellcome Trust. And right now, when I'm speaking with you, we are busy increasing the linear range of detection of this test on glucometer. I am Pratap from Wobble Base, and I welcome your questions. Thank you.
Thank you sir. The lecture is very informative, and I hope many of us get the insight that how the old views can be converted to be make more advanced and more easily approachable. Thank you, sir. And
Thank you, ma'am.
now, yes, sir. Now the next 5 minutes are for questions. So if any participant have any question, you can write in the chat box, or you can raise your hands. So any questions from participants? If not, we can move for the next lecture.
Thank you.
Yes, sir. Yes, sir.
Thank you, ma'am.
Okay. Thank you, sir. Once again. Now for the second lecture, I am welcoming Dr. Snehal Bacteria, and I would like him for his talk on, welcome, sir. And I would invite him for his talk on state government initiatives in antimicrobial resistance, superbugs, and One Health. Sir, please allow me to introduce you. Sir has done in all the education is from the Saurashtra University up to PhD, and presently sir is working as a joint director at Gujarat State Biotechnology Mission at the Department of Science and Technology with the government of Gujarat. Welcome you, sir, and you can share your screen and can start the talk. Please, sir.
I hope the full screen is visible.
Yes, sir, visible.
Okay, yeah. So today we will discuss in brief the title of the talk, as it is given, is State Government Initiatives in AMR, Superbugs and One Health. We'll talk in brief about biotechnology scenario, followed by activities of the organization which I am representing right now, Biotechnology Mission, and what initiatives we have taken to address the challenges related to AMR, Superbugs, and One Health. So if you look at globally, biotech industry is 752 billion and it's growing at the rate of 15%. In India, biotechnology business is 70 billion and it is growing at the rate of 16.4%, and by 2025 it will be 150 billion. In India, biotechnology is majorly represented by biopharma, followed by bioservices, bioagri, and bioindustrial. Notable things is, as we discussed earlier also, we are amongst top 12 biotech destinations in the world, ranks third in Asia Pacific. We supply more than 60% of the vaccines. We are having highest number of US FDA plants, and we are hub of biosimilars with near about 100 approvals in the country. If we look at the state scenario, Gujarat is a home to 232 companies. Majority of the companies are in agri-biotechnology and pharma-biotechnology. We have 83 companies in agri-biotechnology, 75 companies in pharma-biotechnology, followed by bioservices. Our revenue comes, state revenue comes in biotechnology majorly from pharma-biotechnology, representing 46%, followed by agri-biotechnology, representing 20%, and bioservices 13%. If we look at the sub-segments of biotechnology in the state, biotherapeutics is a major area in pharma biotechnology, followed by nutraceuticals, vaccines, and diagnostics. Agri-biotechnology, the three areas are tissue culture, biofertilizer, and bioinsecticides. To develop biotechnology innovation ecosystem in the state government has developed 4 key organizations, namely Gujarat State Biotechnology Mission, Gujarat Biotechnology Research Center, Savli Technology and Business Incubator, and Gujarat Biotechnology University. For developing biotechnology innovation ecosystem in the state, Gujarat State Biotechnology Mission is focusing on 3 key activities, namely biotechnology business development, biotechnology human resource development, and biotechnology research and development. If we look at business development, state has a biotechnology policy where support is provided for developing, various segments of biotechnology. These segments include development of biotechnology park, where support is up to 25 crores against capital investment. 25% capital subsidy is provided. For establishing the new biotech unit, support is provided up to 50 crores in form of capital subsidy to the tune of 10%, and interest subsidy is up to 10 crores per annum. Capital subsidy for setting up incubator is up to 50%, and total support provided is 1 crore. For software procurement, support is 50 lakhs. And for establishing pilot production facilities for startup is 10 lakh support is provided. For developing finishing school, capital subsidy is provided up to 1 crore. For establishing shared industrial testing facility, support is provided up to 50 lakhs. And for establishing the contract research in industry, 25 lakhs is provided. IPR support is also provided. Domestic patent support is up to 2 lakhs, and for international patent support is up to 5 lakhs. And there are, good outcomes of this policy support. For example, Concord Biotech has established one of the world's largest fermentation facilities. Zytex Biotech is Asia Pacific's bacillus producing company in Gujarat. Sandiota is one of the world's, one of the largest probiotic company in India. Lester Biosciences, we all know, Asia Pacific's largest company. There has been good, outcome of Vibrant Gujarat with near about 3,000 crore of investment has been realized. Venture support, we have seen a good success where, key projects are supported. New biotech policy will be in line with our Honorable Prime Minister's vision of Atmanirbharta, where dedicated support will be provided for frontier technologies and import substitution, developing, the key technologies in the country and in the state, leading us to Atmanirbharta. And there will be special support for technology creation. Another area where Gujarat State Biotechnology Mission is working is human resource development. Notable programs are PG Diploma in Biotechnology, Law and Public Policy, Industrial Biotechnology Training Program for the students, and conference, seminar, and symposia support. Research support is one of the key areas where we support research across the academia and institutions. Till date, 187 projects are supported with a total support of 68 crores in the state, which provided support to 200 faculties, 200 research fellowships have been provided, and more than 240 publications have been the outcome. The notable programs are Network Program on Cancer Research, Environmental Biotechnology, Animal Genomics, and COVID-19 research. The new initiative, which is a topic for today's discussion, is a network program on antimicrobial resistance, superbugs, and One Health, on which I'll discuss in detail in the present presentation. So if we look at antimicrobial resistance, as we all know, AMR occurs when bacteria, virus, fungi, parasites, they change over time and then they no longer respond to the existing medicines, making it harder to treat infections, increasing the risk of disease spread, severe illness, and death. If we look at global burden of antimicrobial resistance, it is predicted by WHO that there will be approximately 10 million deaths by 2050, costing total $100 trillion in terms of economic output. If we look at Asia Pacific scenario, Asia Pacific will be highest affected amongst all the regions with 4.7 million deaths by 2050. If we look at AMR as compared to other diseases, AMR will be the leading cause of death, followed by cancer, diabetes, diarrheal diseases, and road accidents. So if I further emphasize it, if we look at the COVID-19 deaths at present, total COVID-19 deaths in last 2 years, approximately 2.5 years, is 5.6 million. It is predicted that if we do not address the issue of AMR, there will be approximately 10 million deaths per year because of AMR. Hence, AMR is predicted as a major concern and a developmental threat, and it is also included as one of the important parameters in sustainable developmental goals, by UN. If you look at AMR is a one health issue. AMR cannot be addressed independently. AMR means addressing the issue of AMR in humans, addressing the AMR in non-human hosts like wildlife, animal, etc., AMR in environment. So it is an interdisciplinary, multi-sectoral initiative where all the organizations working in human health, working in livestock diseases, working in fisheries, working in wildlife, working in environment, they have to come together to address the issue of AMR. We all know what are the reasons of AMR. And AMR, is an issue because of overuse of the antibiotics, inappropriate prescriptions, self-medication, increasing number of elderly and immunocompromised patients. Another area is antibiotic use in food producing animals and agriculture. We are using a lot of growth promoters in poultry value chain, in animal husbandry, and because of which AMR is increasing. This is being released in the environment. There are a lot of fertilizers and pesticides we use, because of which it is also entering into our crop systems and plants. We have overuse of antimicrobials in hospitals for treatment, like widespread and prolonged use of antimicrobials for therapy and prophylaxis. Invasive surgical procedures and invasive medical devices. These treatments also use the antimicrobials, and because of which AMR is becoming a global issue. If we look at WHO data, WHO has a global database which is known as Global Antimicrobial Resistance and Use Surveillance System. It is broadly known as GLASS database. On GLASS database in 2021, 3 million laboratories confirmed antimicrobial resistance. High number of bloodstream infections caused by E. coli and Staphylococcus aureus, MRSA, which were resistant to third generation cephalosporins. The resistance was observed in low and middle income countries than that of richer countries. There has been resistance to last resort antibiotics such as carbapenems in bloodstream infections caused by even common hospital infections such as Acinetobacter and the members of Enterobacteriaceae family. Considering such a high level of AMR globally, WHO in its meetings, policy meetings in 2021 and in 2020 decided to move from sentinel survey to population-based survey. Another approach which WHO has suggested is to move from microbial surveys to genome sequencing-based survey. These were the key decisions taken by WHO, and world over several countries have adopted. If you look at the numbers, 88 signatory countries have adopted to move from sentinel survey to population-based survey of AMR, and committed for appropriate resource allocation. And also countries are moving for genome sequencing-based surveillance. If we look at Indian scenario, WHO and DBT, Department of Biotechnology, has classified the organisms in 3 categories. Group 1 is critical priority pathogens, which is to be addressed immediately. Group 2 is high priority pathogens, and Group 3 is medium priority pathogens. If you look at several members of Enterobacteriaceae like Klebsiella pneumoniae, Escherichia coli, Acinetobacter, Pseudomonas, they are resistant to carbapenem, cholesterol, and various other organisms. If you look at high priority pathogens, MRSA is key contributor. MRSA is also identified, by under the Sustainable Developmental Goals to being the key organisms to be addressed. Now, Enterococcus is another group of species which is to be addressed, and Salmonella is also to be considered. If you look at the Indian scenario, each signatory country has resolved that they'll have a national action plan, and accordingly, Government of India has identified a national action plan to address the AMR, and ICMR and National Center for Disease Control is focusing on implementation of National Action Plan. And as per National Action Plan guideline, each state also has to develop the state action plan. And there are 5 components identified by WHO for developing a National Action Plan. One is generating awareness and understanding on AMR, developing a knowledge and evidence by establishing laboratories, establishing surveillance mechanism for human, animal, and environment by each partner countries, by taking appropriate measures for infection prevention and control, by developing a policy mechanisms for optimizing the use of antimicrobial resistance, by making appropriate provisions for resource allocation, for innovations and R&D, for developing the new medicines, for developing the newer diagnostic tools, and supporting through research support. This has been the key area. In our action plan, Government of India, Honorable Prime Minister has suggested that we should slowly develop the scientific leadership in the country, so that the sixth parameter is added by Government of India is to develop the scientific leadership of the country and the state, so that we can have we can be at par with global initiative. This has been the 6th pillar added in our national action plan of the country. Considering the National Action Plan and global initiative, state has also decided to develop a network program on AMR superbugs and One Health in Gujarat. And as visualized in the country also, where at a national level, ICMR, ICAR, fisheries, Department of Biotechnology, Ministry of Forest and Environment, everybody is coming together. In state also, we have visualized that we'll have key organizations working in medical sector, working in veterinary, working in fisheries, research institution and university should come together to address the issue of AMR. Further, state government under Department of Science and Technology has established GBRC for biobanking, and GBRC facilities for the AMR purpose will be used, and key activities of the GBRC as a hub will be biobanking, sequencing, and bioinformatics. We'll have close coordination with line departments, health department, animal husbandry department, forest and environment, municipal corporations, and GPCB for addressing the issue of AMR. And accordingly, we floated a program with following objectives. The key objectives for the national, for the network program on AMR is to establish the surveillance lab for AMR in humans, animal, food, environment, in academia and research institutions, to identify priority pathogens and to initiate the biobanking activities of reference strain and novel strain, to study the transmission dynamics. This has been very important. If you look at COVID scenario, transmission dynamics, how the virus is going across the value chain, is very important, and hence, to study the transmission dynamics in medical setups in environments will be very important using the One Health approach to initiate the genome sequencing, to initiate the molecular studies and characterizing the drug-resistant pathogens, to develop the novel panels of the genes responsible for AMR, to develop low-cost diagnostics for kits for leading to rapid detection, to develop technologies for infection prevention and removal of antimicrobials will be important area for us. One of the important area that we learn from the COVID research is, and if we know that drug repurposing is a very important area. As we all know, drug discovery is a very long-term goal, and short-term goal could be repurposing of the existing approved drugs. Hence, using the bioinformatics tool, drug repurposing has been added as important objective in the state supported program. Another area, similar on the lines of GLASS, WHO database, state will have its own database for AMR in Gujarat, and of course supporting various awareness activities in Gujarat to partner in national programs and to develop the state action plan, which is a mandatory requirement under the national action plan. So under this program, GSBTM invited the proposals and has supported several nodes. If you look at key nodes who will be initiating the activities in human healthcare segment, key medical colleges are involved. For working on AMR is BJ Medical College, Government Medical College, Vadodara, Surat Medical College, Bhavnagar Medical College, JUMS Gandhinagar, GMERS Gandhinagar, NHL Medical College, Ahmedabad. Key institutions involved in health segment is Gujarat University, Ahmedabad, and MS University, Baroda. If you look at the institutions supported in veterinary and fisheries sector, key institutions supported is the Department of Animal Husbandry, Animal Testing Laboratory, Ahmedabad, Kamden University, Sardar Krishinagar campus, Anand campus, Gandhinagar campus, Fisheries campus at Himmatnagar, Gujarat Vidyapith Sadra will be working on anaerobes. Dairy College will be working on dairy related AMR issue in Amreli. And then Veraval and Navsari and Ukai campus will be working on the fisheries sector, focusing on inland and marine fisheries and AMR related issues into fisheries. If you look at the environment segment, key universities will be working on environment and AMR will be Veer Narmad South Gujarat University, Gujarat Biotechnology University, Gandhinagar, Charuset Changa, Gujarat Technological University, NIOH, Ahmedabad. These are the key organizations working. If I summarize in brief the entire network program, in entire network program, there will be 6 medical colleges, 1 national institute, 7 university departments, 18 veterinary state labs, 50 investigators will be working total, 8 veterinary and fisheries campus, and 19 AMR nodes will be establishing, 26 institutions will be participating into it. This altogether makes it one of the largest AMR and Superbugs program in the country, covering the 3 key important area as suggested by WHO. A is population-based survey, B is genome sequencing-based survey, and 3 is One Health approach, which is applied across the sector, that is human health, environment, and veterinary and allied area. Total support as decided by state government under Department of Science and Technology will be provided to these organizations will be 18 crores in next 3 years. Year 1 support will be 6.5 crores, year 2 support will be 5.5 crores, and year 3 support will be 6 crores. What these organizations will do, the human health organizations will be working on bacterial isolates, fecal samples, UTI samples, blood samples, nasal swab, upper respiratory tract samples, environmental organizations will be working on bacterial samples, sewage treatment and sewage sludge samples, river sediments, river water, municipal wastewater, fish gut samples, nasal swabs, blood, personal dust, human gut flora, marine water, solid waste and wastewater from industrial area. NIOH will be focusing on medical waste also. If we look at veterinary bacterial isolates, milk samples for mastitis, poultry swabs will be taken, animal fecal samples will be taken, raw meat and processed meat and milk samples will be taken, probiotic and fortified milk products will be considered into this segment. Fisheries also similarly, range of samples will be taken. So there will be a diverse sampling and One Health approach. GBRC will be focusing on various types of sequencing samples, including WGS, amplicon sequencing, AMR panels, metagenome and shotgun sequencing, gut microbiomes. They will be also focusing on biobanking. Total 10,000 samples will be processed in next 3 years. So it will generate a large amount of data, which will give us important insights in the state. So readily available amplicon panels will be used, which will be focusing on 28 different classes of antibiotics. Total 815 amplicons will be targeted, focusing on near about 480 genes contributing to AMR. So what insights it will provide? It will put us on a platform which is in line with parameters set by global organizations for international surveillance. It will make us make us at par with national surveillance modalities and mechanisms. We'll have a robust network of local surveillance. We'll be able to identify source of infection and outbreak. We'll be able to trace the transmission chains and network. We'll be able to identify high risk clones, high risk groups, and we'll be able to correlate virulence factors to patient outcomes. We'll be able to identify AMR pathways for and how the AMR is evolving, how it is contributing across the human value chain. We'll be able to determine the molecular mechanisms for AMR, and we'll be able to develop the point of care tests in times to come. With this, state is gearing up for addressing the challenge of AMR, and I hope that in times to come, many more researchers under this program will participate. So I'll end the talk with a famous quote of APJ Abdul Kalam. Look at the sky, we are not alone. The whole universe is friendly to us and conspires only to give the best to those who dreams and work. Thank you. Thank you.
Thank you, sir, for sharing such facts and figure about biotechnology, for reflecting the growth of biotechnology sectors and the very appreciative contribution of GSBTM as well as the state government. Yes, sir, it is definitely right that biotechnology related technologies has very much potential in providing solutions of various problems, like approaching to One Health approach for fighting with superbugs and AMR. Thank you, sir. Thank you, sir, for a very enriching lecture. Now it's time for questioning. If any participant has a question, can write in the chat box or can raise his or her hand. Any participant have any question? If not, thank you, sir.
Thank you.
Thank you, sir. Now, before proceeding for the next talk, we have a 5-minute break here. Please stay connected and return back in 5 minutes. Thank you, all the participants. Please come again in 5 minutes.
Yeah, hello. Yeah, is anybody there?
Yes, sir. We'll start in a few minutes.
Okay, fine. Sure, please.
Uh, shall we start for the next session? So for the third lecture, I would like to invite Dr. Manish Raj for his talk on scope, challenges, and opportunity in biopharmaceutical sector amidst COVID-19. I welcome you, sir, on the behalf of all the participants and all the coordinators. Now, allow me, sir, to introduce you. Yes, sir.
Thank
Sir
you.
has Yes, sir. Sir has completed his Master of Pharmacy, Pharmacology and PhD from the LMCP College of Pharmacy, Ahmedabad. Currently, sir is working as a director at the Acupric Research Lab Private Limited. Sir has more than 12 years of academic experience in various pharmaceutical institutions. He also served as an associate professor at Gujarat Technological University GTU, and sir has a member of many councils and advisory committees such as state level advisory committees, IPFC, Government of Gujarat, Research Council, GTU, Gujarat, and many more. And as well as sir is also has experience in patenting. He is a registered patent agent and IP facilitator. Sir, I welcome you again, sir. Now, sir, please start your screen and lecture.
Okay, yeah, thank you.
Yes, sir.
Yeah, can you able to see my presentation?
Yes, sir.
Okay, fine, perfect. Yeah, very good morning to one and all. And I think as a part of this, our 75th Azadi ka Amrit Mahotsav, the Vigyan Guruzari, which is a part of, you can say, Vigyan Bharat is organizing this important event. And so I must congratulate all the, you can say, the stakeholders and members at the national level as well as the state level for promoting, you can say, science and research among the academia as well as in the industry. So at the outset, I thank GBRC for inviting me to share some of the views. And I just selected a topic which is, I think, I feel it is relevant for this audience as well as the current scenario also, because the situation since last 2 years are something which everybody is very well aware. But of course, any, you can say, the situation or challenges always bring a lot of opportunity. So I'll just try to give some glimpses because the time is very limited. So I'll just try to give you, you can say, some kind of, you can say, scope, challenges, and opportunity, particularly in the field of biopharmaceutical, because I presume majority of the participants belongs to biotechnology side. So which are the opportunity for the biopharmaceuticals amid this COVID-19? So let's start. So I think before December 2019, nobody is aware about this COVID-19. But I think nowadays, even in like school children, they are aware about COVID-19. But let's understand more about the science behind this COVID-19, and then followed by the, you can say, the biopharmaceutical related with the COVID-19. So of course, this gentleman is also having the same question: what is COVID-19 science behind it? So this, I think, very well everybody is aware. So it is a kind of, you can say, respiratory illness or a syndrome which caused by the coronavirus, or say SARS-CoV-2, in particular the virus which is most prevailing. And nowadays various, you can say, starting from different forms or variants. Now we have Delta and now we have Omicron also. So this is just to start, you can say, preamble of my presentation. But now, as I said, about we have to focus more on a science part, because you may be working in academia or maybe in a different part of a research in the same segment. So this is the, you can say, the core structure of this particular virus and how it is encapsulated and having a spike protein, which is a very particular type, and lot of, you can say, research already has been done and still ongoing. A lot of, you can say, new research is required to be done. And that's why I have selected this topic, because still COVID is not over and fight against the COVID is yet not over of our mankind. So these are the various, you can say, that starting from spike protein, that is S, then membrane protein, that is M, then spawn alveolar protein, that is E, nucleocapsid protein, that is N, which is inside the nucleus of this mRNA virus, and of course the genomic as well as the ssRNA. As everybody might be aware, okay, this is the RNA-based virus. So in the core capsule of this virus is, we can found the ssRNA. So these are, in short, you can say, the broad targets if we want to hit this COVID-19 virus. Of course, we must have to understand, suppose we have to hit anything. So for example, when we were a small kid, we used to use that, you can say, the gun to hit the, you can say, balloons. And of course, nowadays also that trend is available. So if the balloon size is, say for example, 5 centimeters, of course, the bullet size should be smaller. Same fundamental, same principle is applicable for this virus also. So that's why it is important to understand what is the size of this virus, because if we want to hit it, we have to use, you can say, that either chemicals or biopharmaceuticals which should be below the size of this virus. Then and then only it will produce its impact. So this is just, you can say, the comparison slides wherein you can get an idea. The virus in general are in a nano size, 20 nanometer to 400 nanometer, and our this COVID-19 virus is also having a less than 100 nanometer size. So you have to now understand it's a very tiny molecule, but creating a lot of, you can say, turbulence across the globe. Of course, bacteria size is larger, almost, you can say, up to the 2 micron, that is a 2000 nanometer. There are smaller airborne particles which are available in surrounding our atmosphere, in my office area as well as wherever you are sitting. This particular size is commonly found. Then larger airborne particles are always bigger than that. There's a 10 micron, and pollen grains, which are kind of, you can say, allergens, that are of size of 15 to 200 microns. And just to understand more in a detail, again, in terms of the size wise, if you want to compare this our COVID-19 virus, so as I said, it is 100 nanometers, that is a 0.1 micron. Nowadays, people are using masks. Government is also enforcing a lot of, you can say, stress to wear the mask, and particularly N95 mask, which is having a more capacity for the filtration. This N95 mask has a capacity to, you can say, the prevent any kind of particulate matter, having a, you can say, the size of 0.007 micron. So that means almost up to the 7 nanometer. So 1/10 approximately about the COVID virus can be filtered out if we are using the N95 or higher than that mask. Bacillus bacteria, of course, as I said, is a 500 nanometers or 0.5 micron. Then airborne particles, there is 2.5 micron. RBC of our red blood cells, almost you can see the size, 7 micron, almost 70 times bigger than our coronavirus. So coronavirus, that's why can able to penetrate our body cells, including the RBCs, very fastly, very rapidly. And this is the particle size of 10 micron. So you can understand and imagine our target is very tiny in a nano size. So we must have to think some kind of, you can say, the treatment approach, which should be tiny in nature or which should be nano in nature. And that comes, you can say, the field of biopharmaceutical, because this particular virus is biological origin. And anything, if we want to hit with the biological origin, it is always better if we can target some biological weapons against it. So biological weapons here, it is nothing but for the treatment part. So that's why it comes as a biopharmaceutical. Some may say this COVID virus itself is a bioweapon. Yes, there are a lot of, you can say, issues going on across the globe and a lot of discussions going on whether it's a biological weapon by some countries who have started and released from their lab. We don't want to discuss that part over here, but we want to focus ourselves on a, you can say, a hitting of this bioweapon with the help of biopharmaceutical. So what exactly biopharmaceuticals are? So it is nothing but you can say the biological derived medicinal product or a biologicals having some kind of, we can say, pharmaceutical importance, manufactured as well as either from extracted from, or it may be semi-synthesized also from the biological sources. So in short, anything chemicals which are available from the natural sources, it may be plant, it may be animal, it may be humans, it may be microbes also, that can be termed as a biopharmaceuticals. Just for your information, the first biopharmaceutical product developed was insulin. And again, it was a chance discovery by the two researchers. We'll come to discuss a little bit about it in my last part of my presentation. But if we want to classically categorize the biopharmaceutical, it can be categorized in 7 broad terms, starting from hemopartic growth factors, then monoclonal antibodies, then vaccines, thrombolytic agent, interferon, hormones, as well as the blood factor. Let's see a little bit about each type of, you can say, biopharmaceutical. So first is a hemopartic growth factors. These are the nothing but a group of proteins that cause blood cells to grow and mature. For example, like erythropoietin, which is a glycoprotein hormone that control the erythropoiesis. That means the generation of RBC inside our human body. So it is basically produced by the endothelial cells of the kidney. And if it is not present inside the body, or if the level is not sufficient, the patient, the person become anemic. That is, person is going to suffer from the condition called anemia. And nowadays, you can say in the market, the erythropoietin alpha injections are available. So in case of severe anemia, patients are need to take this, you can say, erythropoietin alpha injection prescribed by the, of course, the doctors, then only. The next is the monoclonal antibodies. The name itself suggests they are the antibodies and going to act on a single target. So they are customizedly developed inside the laboratory and then they're manufactured in the biopharmaceutical companies. So it is antibody produced by the single clone of a cells and produced by using hybridoma technology. And of course, they are popularly known as a magic bullet, because the type of, you can say, the target is there accordingly, it has been customized. So just like lock and key mechanism, if we know what is the structure of lock and it, then we can able to prepare a particular key for that lock. So just like that way, these monoclonal antibodies are working, and a lot of monoclonal antibodies are nowadays available in the market for the treatment of rheumatoid arthritis, cancers, and like so on. And now in COVID also, we'll also discuss a little bit about the kind of, you can say, monoclonal antibodies whose use has been started enormously. The next group of biopharmaceuticals are the vaccines. Nowadays, everybody is aware about the vaccines due to this COVID, but before that also a lot of vaccines have been developed, starting from, you can say, the polio vaccines, the tuberculosis vaccines, and like so on. So biological preparations that improves the immunity against a particular disease, that are known as a vaccine. Vaccines, again, it may be active or a passive kind of, you can say, the vaccines. So it is nothing but a subunit of vaccine composed of only surface protein produced in a yeast. So you can say, using a recombinant DNA technology, majority of nowadays vaccines are getting manufactured. And even about these, you can say, the COVID vaccines, again, it required some host cells. So normal, in majority of pharma companies nowadays, they're using E. coli as a host cell, because it is very easy to mimic the E. coli, you can say, bacteria, and that's why they act as a, you can say, the host cell for the development of vaccine. The next group is the thrombolytic agents. There is a protein involved in the breakdown of the blood clots. Again, very powerful kind of, you can say, the biopharmaceutical product in a when any patient is getting, you can say, severe stroke, severe heart attack, At that point of time, no medications can able to protect. No medications, that means the pharmaceuticals cannot able to, you can say, protect the patient. So at that point of time, the biopharmaceuticals come in the pictures. That is like tissue plasminogen activators, like serine protease. They act with the serine protease enzyme, and by that way they just cleave the coagulated you can say parts inside the blood vessels, particularly coronary blood vessels, and by that way it can act. Next group of biopharmaceuticals are interferons. So interferons are nothing but a cell signaling proteins produced by the cells of the immune system in response to the challenges such as viruses, cancer cells. So basically we have T cells, B cells in our as a part of our immune system. So when any like foreign bacteria or viruses or any other organisms get inside the, you can say, the body, then T cells and B cells starts releasing certain kind of, we can say, cell signaling you can say molecules, or say kind of, you can say, particular antibodies against that particular bacteria or virus. For example, this interferon alpha, beta, gamma, etc. Lot of, you can say, that's kind of, as a natural body defense system, body start creating that kind of army against that particular invading organism. So nowadays interferons are also in a you can say big research due to this COVID. That also we'll see. The next biopharmaceutical category is a blood. That is a blood factors. That are the components of the blood, like factor 8, factor 10. So when anybody is suffering from the, you can say, the blood coagulation disease, so at that point of time, particular type of blood factors need to be transferred in such patients. So it is again known as anti-hemophilic factors, and if this kind of blood factors are not produced by the body, the conditions called hemophilia, means just by simple, you can say, cut, blood continuously flow out of the body, and patient may get collapse also. So at that point of time, again, normal pharmaceuticals is not helpful. Again, we have to use a biological weapon or the biopharmaceuticals that are the, as I said, various factors are available. Depending upon the requirement, doctor prescribe the various factors to such patient suffering from hemophilia. The next group of pharmaceuticals are hormones. As I said, it is a chemical substance that control and regulate the particular activity inside our certain cell or a tissue or organ. Very common is, as I said, the first biopharmaceutical that is developed was insulin, and nowadays a lot of, you can say, the hormonal products are available, like HCG, then testosterone, and a lot of, you can say, the biologically derived hormones are available in the markets. So for instance, in case of insulin, if body cannot able to produce, the condition of the disease that everybody, I think, aware, it is a diabetes mellitus. And that's why external insulin may be required, in particularly if the patient is suffering from type 1 diabetes. So now this all so far we have seen about what is COVID-19, the structure, or say, you can say, targets of the COVID-19 virus. Then we have seen what are the biopharmaceuticals in general. Now let's see the scope of biopharmaceuticals in this COVID-19. So of course, as now you are aware about the structure. So first of all, can we have some drugs or biopharmaceutical which can able to prevent the entry of virus itself inside our body's cells, that is a host cell, what we can say. So thus, first target would be the inhibitors, which prevent the entry inside, you can say, the host cell. If already some, you can say, virus get entry inside the host cell, then we need some, you can say, inhibitors which prevent this proliferations, and that's why it's prevent the further progression of the disease. And this is just to give you some broad idea about the various targets so far identified by the various researchers across the globe. Various drugs already have been prescribed or recommended by the WHO as well as ICMR at the various phases of time, like starting when in April 2020, when the first, you can say, the guideline against the COVID-19 has been published. Even WHO and ICMR has recommended to start uses of chloroquine, which was very old drug for the malaria, treatment of malaria. But this chloroquine, or say semi-synthetic, this hydroxychloroquine, was found something, you can say, important in this case also, because you can see over here, it inhibits the virus entries inside the host cell. And that's why initially it was prescribed and suggested and recommended by the regulatory, but later on we came to know that it is not so much effective. So COVID has, you can say, starting from, you can say, if you see the various kind of, you can say, series nowadays on Netflix or like that, we started with, you can say, series one, or say the first part, and then various part have been came, and each part is increasing our, you can say, the thirst to know what, or say eager to know what will happen in the next episode. Similarly, starting from, you can say, December 19, or as I said, from March or April 2020, when our Indian regulatory become more serious about this COVID-19, lot of, you can say, the new episodes came, lot of new guidelines came sequentially, apart from, say, chloroquine, hydroxychloroquine, lot of other drugs have been suggested. Likewise this. So these all, I think you are aware, but just to understand, make you clear, okay, these are the various targets against which the treatments are nowadays available. Regulators are suggesting, depending upon the, you can say, severity of the disease, which drug to be used. Like very common during our second phase last year, everybody is aware about this Favipiravir, and then if the case is moderate, then remdesivir injections. And if the still the severe case happen, then you can say tocilizumab, like a MAP series of drugs are prescribed by the doctors and recommended by the regulatories like ICMR and WHO. So let's focus on monoclonal antibodies, which is more relevant for this talk. So of course, if simply say, what are the antibodies? So of course, without antibodies, virus can get entry inside the host cell, and then depending upon our immune response, antibodies get developed. But if monoclonal antibodies have been given, so this monoclonal antibody, as I said, it is customizedly developed against the particular, say, in case of COVID, it is against a certain spike protein. It will just adhere to the spike protein of the COVID-19, and that's why it prevent further, you can say, invasion of the virus inside the host cell. So whatever the disease progression, that can be immediately gets halted, because virus cannot be able to invade the new body cells. And that's what you can say use in, again, in cases of severe case only, we have to use this biopharmaceutical, because as I said, they are the tiny biomolecules. So though they have a good effect also, and of course it is another side of a coin, a side effect or adverse drug reactions also. So only WHO and ICMR recommend only in case of severe COVID, we have to go for the MAPS group of drugs. And MAPS are again 100 plus nowadays. It is not only 2 or 3. 100 plus are already approved by the US FDA, and many of them are available in India also. So a lot of MAPS have been so far developed. These are the some of the, you can say, MAP which are commonly used in the Western world in case of mild to moderate, like depending upon the, you can say, age and body weight, different kind of, you can say, the MAPs have been prescribed. Even like post-exposure prophylaxis also, as well as pre-exposure prophylaxis also. Some of the, you can say, MAPs have been available. Apart from MAP, a lot of, you can say, research have been targeted for the novel therapeutic approaches, starting, as you know, about something about the serum therapy. Again, in the last year, after like chloroquine and other drugs have not been successful enough, or maybe in a like moderate to severe cases, then again, WHO has recommended the serum therapy. And again, a lot of, you can say, clinical trial happen in a lot many government hospitals across the country for the serum therapy. Then MAP therapy, then adoptive immunotherapy, mesenchymal stromal cells, then nanomedicines. As I said, that is a, you can say, new avenues wherein a lot of research has been happening, because our target is nano. So if we can hit with the nano, of course, it will produce much more impact than decoy biomolecules. And of course, there are some classical antiviral drugs like Favipiravir. Nowadays, recently you heard about the Molnupiravir, and apart from that, also like a lot other vir series like Lopinavir, Ribavirin, etc. are already tried in a different part of the world, and some of them are also recommended by the WHO as well as ICMR also. But I think recently, still Molnupiravir is approved by the DCGI of India, There is a Drug Control General of India office, that is CDSU, but still ICMR has reluctant to approve the molnupiravir, which is a more stronger molecule than the favipiravir, which we have used in the last year because of of course, the side effects. Yeah. So after this, monoclonal antibodies, the next big group of biopharmaceutical, which is commonly used across the world for COVID, is vaccines. So nowadays, I think everybody is aware about the various kind of, you can say, vaccine platforms which have been developed by many companies across the world. More than, you can say, 500 different vaccines are developed by the different companies across the world. Many of them are already approved and available in the market. But please understand, availables are maybe 40 or 50 across the world, but still you can say a lot of research, 400 plus vaccines are under development under the various stages of preclinical toxicology as well as the clinical phases of the trial. And each vaccine have their own different targets. It may be the protein subunits or virus-like particles or DNA, RNA based, like so on. And again, it may be inactivated or maybe the live attenuated type of vaccines. And this is just for your understanding, like these are the commonly worldwide used vaccines. There is from Pfizer, there is RNA-based, Moderna, again RNA-based, because virus core is RNA. So that's why they have targeted on RNA and then developed this particular vaccine. Of course, this third one is Oxford AstraZeneca vaccine, which is again a viral vector-based vaccine. And in India, this Covishield from our Pune-based Serum Institute, they got the license to manufacture the same vaccine over here. And of course, the Sputnik is from Russia. Again, it is again the virus vector. And these are the, you can say, the Indian-based manufacturers who are leading in this COVID vaccine. So as I said, Covishield, again, they have collaborators from AstraZeneca. Covaxin, of course, it is 100%, you can say, Indianized the vaccine developed by the Bharat Biotech Hyderabad in the collaboration with the ICMR. Third is a Zycovd, that is a DNA-based vaccine developed by our this Ahmedabad-based Kedila Healthcare only, and again in collaboration with the Department of Biotechnology. So again, we have to take a proud care. A lot of big countries could not able to succeed to launch the vaccines in the market, but in a very shorter period of time, our companies like Bharat Biotech and next is this Kedila Healthcare can able to make availability of the vaccine in the market. Of course, Kedila is in a final stage to make it available, but Bharat Biotech, the Covaxin is available across the country. Now, let's see about we have so far seen the, you can say, the various scopes, how this biopharmaceutical is very much important to combat against the COVID. Let's see the challenges. Biopharmaceutical like MEB have a severe, you can say, adverse drug reaction, like already the patient is severely ill, and in that patients, if we are using such kind of biopharmaceutical, it is, as I said, double-edged sword kind of things, because it is nothing but a biological weapon only. Weapon may hit to its target, and if it's not able to hit its target, it hit something else. And hit something as this, it means it hit a lot of other body systems which is available inside our body, and commonly is the cardiovascular system, and a lot of, you can say, cases happen, but not too much talk has been happened on this, but a lot of, you can say, patient even died after having a, you can say, injection of this tocilizumab and other kind of MAP series, because it produced severe, you can say, cardiovascular collapse condition and lead to that. So there is a big challenge. Of course, in a particular time, in a particular dose, if it is given, it can save the life. But if, as I said, something goes missed and target get missed, but MAP will not miss, it will definitely hit something. And if it hits some other body system, this is the chance. And of course, the COVID-19 vaccine, it is also not free of side effects. A lot of, you can say, the problems already, many of might have already taken the vaccine. So you know about these symptoms of the COVID-19 vaccines problem. And the major drawback of the COVID-19 vaccines, after every 6 months, we have to re-immunize ourselves, like after first dose, second dose, and now we have to take the third dose also, or the booster dose also. So situation will continue. after every 6 months. And not only in India, we have tried, our government has tried a lot of efforts to do the vaccination across the country of the senior citizens and moderate age, and now children from 12 to 18 groups also, and simultaneously started the third dose. But you know, the COVID-19 vaccines have a lot of, you can say, the turbulence across the world. And this is the latest turbulence happen in Canada. And it is very, you can say, the pathetic situation. Like Prime Minister of Canada has to fly away somewhere down the line because his entire capital is locked down by all the protesters from across the country. Why? Because PM of the Canada has announced compulsory vaccination to all the truck drivers who are crossing the Canada border and entering to the US. Of course, we consider it is a good step, but in Canada, the situation is something different. Not in Canada, many like European countries and US also, many people are reluctant to take the vaccine. So these are, you can say, the challenges. And of course, as I said, from challenges only, we have to find out the opportunity. So which are the opportunity? You can see the worldwide, you can say, the biopharmaceutical or biomedicine market by 2025, it is going to become this much US billion dollar, $800 billion. So big market, even bigger. It is going to be bigger than the pharmaceutical market, which is now dominating. But now this biopharmaceutical market, due to this COVID-19 only, it's going to make very big jump, and it is going to cross over the pharmaceutical market of the world. So please understand, biopharma is the next future of the next decade. But of course, the development of safer biopharmaceutical, that is a need of an era. Can we think about the plant-based biopharmaceutical? You may surprise, a lot of research is happening across the world in developed countries to derive certain biopharmaceutical from plant-based, and which can able to target, you can say, COVID-19. And I'm also happy that in our India also a lot of research have been started. But we being a CRO, become a part of one of the, you can say, the studies from our sponsor who have developed a plant-based biopharmaceutical, and we have conducted, you can say, various studies as well as the clinical trial in the COVID also. And 2 clinical trials have been conducted and results were fantastic. So in short, a lot of opportunities are there for the plant-based biopharmaceutical. So being academic researchers, I would suggest you can also give your focus on this also, because a lot of, you can say, biomolecules can be derived from plants. And last but not the least is the better vaccine, which can be active against the multiple targets, so that we don't have to take newer vaccine dose each time when the variants get changed. So these are the, you can say, the opportunity. And next question comes, how to identify the thirst area for the research. Being academic researchers, what we have to do? That, of course, after understanding this scope, challenges, and opportunity come in our mind. So this is, you can say, my some of the summary slides are going to come. So this is the one of the summary slides, wherein being an academic researcher, we must have to understand the PMT. What is this PMT? To identify, you can say, the thirst area or a research area, if we want to think, and now both me or myself or my team would like to start against certain, you can say, treatment approaches for the COVID-19. So these are, you can say, the some steps which you have to follow. First, you must have to do the patent landscaping. That will give you an idea which are the various technology, you can say, patents available in India as well as worldwide against the target area in which you want to work. So for example, as I given you example of plant-based biopharmaceutical. So if you do some kind of, you can say, patent landscaping, you will get an idea which are the Indian companies or Indian research groups or maybe the worldwide research group who are working on the plant-based, you can say, biopharmaceutical against the COVID-19. Of course, the next is the market landscaping. What are the markets? So now you know about the COVID-19, it is not going to be over, at least for the next couple of years. So market is very huge, very potential. You can see the enormous amount of, you can say, revenues this big pharma and biopharmaceutical companies are generating. So if you can able to develop something, of course, the market is going to be very huge. Technology landscaping, where the technology is changing. So as I said, we have tried many drugs in a initial phase, like March, April 2020. Then after every 3 to 4 months, you have seen the change in the, you can say, the government guidelines and ICMR guideline to use a new drug or a change in the, you can say, the treatment regime. So a lot of, you can say, the technology platforms have been created, developed, and still modified. So by doing a technology landscape, you can come to know maybe in the future what kind of things can be useful. And in biopharmaceutical, as the core of the virus is RNA, nowadays a lot of research has been happening on a like mRNA technology, wherein if we can develop some kind of, we can say, RNA-based medicines, it is going to play a very crucial role in next couple of years against this COVID-19. But of course, you have to do thorough research for this PMT, what I have mentioned over here. So which innovative pedagogy as an academic researcher I have to follow? So after doing a PMT, I must have to follow this MIT. That means a mentorship. Like if I am like a academic researcher or a professor, I must have to act as a mentor for my research team so that they get complete orientation about the, you can say, the research objective, hypothesis, and the targets. Then, of course, we have to use ICT, that means information and communication technology platforms. So nowadays, a lot of, you can say, things we can do using this word like docking study. So of course, we have to use it. And of course, we have to act as a, being an academic professor or researcher, as a torch bearer. So we have to, you can say, support our research team or research students to and motivate them to go further. So in the last part of my presentation, just a couple of examples. Is there any like academic researchers who have developed something and which become commercialized and having a lot of, you can say, the valuation? Of course, there are a lot many, but I just try to summarize few of them. The first and foremost, as I said, in biopharmaceutical, the first, you can say, the development was happening about the product was insulin. So these two are the gentlemen. One is a student who is on the left side of my side. It is on the right side of a screen. And right side is a professor of the university. So this, the team of professor and his student, the name many of you might be knowing, who have developed or discovered the insulin, Benting and Best. Yes. So this Benting and Best story, in 1921 in Toronto, in Canada, they were doing this research, and accidentally they found the extract from the pancreas of the dog. They found some kind of, you can say, the blood glucose lowering effect. So by that, you can say, the by chance discovery, then followed by a lot of research they have done. So this was, you can say, the first development of biopharmaceutical in 1921. Now come to the middle part. That means in the last century, that is in, you can say, in between year 2000 to 2010. This is again, you can say, the team of left side, you can see the professor, Dr. Alex Ulrich, and his student, Dr. Birgit Müller. They worked on a particular, you can say, biopharmaceutical product, which later on purchased, their technology get purchased by the world's biggest pharmaceutical company, that is Pfizer. And this particular molecule is available in the market as a called Sutent. So again, it is nothing but one kind of, we can say, the monoclonal antibodies against the particular cancer virus, against the cancer. So this Sutent, which this team has developed, you can see their annual sale, almost 1.5 billion US dollar. And this again projection of 2018, and now in 2021 too, it may cross maybe 1.75 billion US dollar. So this is the value of one particular product, which was initially developed by this academic researcher. Of course, they may not get 100% $1.75 billion, but at least if you can imagine, if they get 3% royalty, so almost you can say how much revenue this both gentlemen is availing as of now, due to the development of this biopharmaceutical platform. And of course, in this current situation, that is in 2020, everybody is knowing about from Oxford University, initially there was a development of, you can say, the major part of the initial work was of the COVID vaccine, which is nowadays in India developed by this Serum Institute. There's a Covishield was done by the Oxford University, which later on get collaboration from the AstraZeneca to make it on a larger scale. And of course, doing a lot of clinical trial by the AstraZeneca and make it regulatory approval across the many countries of the world, including India, with help of Serum Institute. So you can see again, the value of, you can say, the business of these vaccines is almost, you can say, more than 50 billion US dollar by this group of AstraZeneca and University of Oxford. So out of this 50 billion US dollar, let's assume maybe 10% is going to be get by the Oxford University. So that means almost you can say 5 billion US dollar university is getting. So in short, my point is, being in academia, lot of avenues are there, lot of opportunities are there, which get created due to COVID-19. It is up to you and your team to identify the right targets. And as I said, we can do the development up to certain level. And that's what these universities in abroad are doing. They do the initial work and then they try to collaborate. And nowadays, I think in India also, a lot of due to this, you can say startup policy. Once you have developed something at your initial stage, lot of now platforms are available like GBRC or GSBTM or DBT. They are providing, you can say, the platforms. Once you have developed certain idea up to the, you can say at the initial R&D scale, then pilot scale, and lot of, you can say revenue also you will get to do the R&D. So you must have to first of all identify the right target, get some funding from the various funding agencies. Nowadays, funding is not an issue for academic research. A lot of funding options are available. But the important is you must have to identify a very valid target. So target identification and validation is very crucial. And that's what you have to do. And that's what you can say you require a lot of due diligence. And once you do it, that means once you serve the Mother Saraswati very effectively, and of course, as I said, a lot of, you can say, patent search and other thing you have to do, and you try to make your this new concept, idea, and target and protect it by means of patent. That means you use a tool of IPR, and then this IPR can act as a catalyst for you. And of course, you will also get a lot of, you can say, the blessings from Mother Lakshmi, which universities, professors in abroad have already started getting. So I hope and I wish from you and your team also should prosper by Mother Lakshmi also. So thank you very much to all of you.
Thank you, sir, for enriching us for in the challenges and opportunity in development of biopharmaceutical. So it is very necessary to develop new drugs, especially in fighting with COVID. Now the session is for open for the question. If participant have questions, please can write in chat box or raise the hand. Only 1 or 2 questions are allowed. Okay, I think so. No questions are there. So thank you, sir, once again for your talk. Thank you very much, sir.
Okay. Thank you. Thank you. Thank you, David.
Now, for the 4th lecture, I would like to invite Dr. Manish Kumar for his talk on contaminant phyto or bioremediation and sustainability. Welcome, sir. Sir, allow me to introduce you. Sir has completed his PhD in environmental engineering from the University of Tokyo, Japan, and currently engaged as a professor at UPS, Dehradun, India. He has published more than 80 international 80 international papers in various journals and have 17 years of research as well as teaching experience. Sir is the recipient of DST Young Scientist Grant as well as GSPS Research COE Young Researcher Fund and Linus Palme Stipend from the Sweden and other various fellowship. Sir is also the member of various advisory committee for COVID-19 as well in the DBT and other national and international institute. Sir, you can proceed for your lecture and can share the screen. Please, sir.
Thank you. I don't know that whether I should call you Vikas because that cannot be your name. But yeah, this was something. Thank you very much for introducing me. I hope my slides are visible. If not, then you can tell me. Yes, so without doing further ado, because we are already overshooting the expected time, I would like to just say thank you very much for the platform. I accepted, of course, because of course the Gujarat being a kind of my third home, because of course And this Gujarat Vigyan Sammelan is something which is, and the overall umbrella is such a nice thing. So as the inaugural session, several speaker and during the panel discussion, also it came out that India should get back to their traditional ecological knowledge. And in that context, that whether how we should value our traditional ecological knowledge, how can we, I think India has been a great recycling country. We were having had a lot of recycling inculcated in our daily life, but we are losing. So that's why I would, my talk would be, some data will be technical, but still I would focus on the overall takeout. So I will keep my talk very common to people. You don't need to be a specialist in here. But I will have the data to show for those who are really working on this field. Again, I didn't forget, but it is not the any part of the country has these natural challenges, artificial challenges, and social challenges. And I would not talk about because we are much aware. But the current scenario is not safe and not sustainable. And what do we mean by sustainable is that we should not make or compromise the ability of future generation to meet their need. So no problem if we cultivate, but that land should remain fertile for next generation and generation after generation. That is sustainability. So how do we go about it? And so that is will be my focus about this work. And but the point is that we have to deal with the pollution. And for pollution, contaminant, anything which is more than a permissible limit, it becomes contaminant. Many times I ask my student, what is pollutant? And they say that, of course, there are many definitions, but the correct thing is that anything more than needed is a pollutant. Whether it is fluoride is needed. You say that iodine is needed. And many times, iron is needed, but once it crosses a limit, it becomes toxicant or contaminant. So here we know as a chemistry or chemical engineer or many things that there are several methods. I would not go on that, but the point is that here I bring the challenge that how to apply it in the field. So there are several thousand or several 10,000 of the studies published in a scholarly world in every year, but which is working on precipitation membrane and ion exchange, extraction, adsorption. But the implication lab to land, and that is also been a USP of this government, who are trying to say that, okay, well, it works in the lab condition, how to implement in the field. So there is a little bit that aspect I would like to touch. The motivation when I started in this field, phytobioremediation, I realized, or maybe with GBRC, we applied Madhvi as a PI, and Akash. So there were, we wrote about using Ganga project, and where we wanted to remove geogenic contaminants using the indigenous microbes. Of course, always, why not to use the natural microorganisms? Because if it is equilibrium condition, everybody is working fine, then there is no need of, there will be no pollution, in fact, because there are microbes to break down. During my childhood, I was thinking that bread never being wasted, because whenever you throw the bread, either dog will eat or microbes or the fungus or something, at the end of the day, it will be decayed. Later on, I realized that this edible thing is should not be thrown. But the point is here is that how to use plants and less chemical, because in chemical engineering, if I am treating 10 kg of the waste, just for example, and adding 5 kg of the treatment chemical, so then it becomes 15 kg overall. So mass balance says that if we will add chemical into the thing, then probably it is going to increase overall sludge. And this can be cost effective. This can be the major option. So this can be, which should be environmental friendly, and so on and so forth. Of course, there were a cycle of the crop cycle we know in India, Indian traditional knowledge. Every 3 crops, there is a, like we used to put mustard or leguminaceae family just to fix the nitrogen. That time we probably was not needing a lot of chemical fertilizer. We used to use the of course, cow dung and several other things, and we never needed any additional thing. Of course, those things is there 5 minutes down the lane. So we started working on that concept, that how to we will deal with something which is already a problematic, but we will make it resource. It means waste to resource. So first thing that we published under these, all papers are under my leadership. In my lab, so in Ipomia plant, we prepared a Ipomia is bee weed, which is so frequently available in all over the India and all over the, even on the global scale. And we try to remove the cadmium. And we thought that cadmium is the most toxic element. So let's go with first cadmium. Then of course, we vermicomposting. Vermicomposting is one of the thing that has been there in India, but it is a very slow process. We in parallel, we try to do vermicomposting test as well. This is the second thing, because at that time I was working in the northeastern part of the country, and so there we found that Burmese grape leaf, there are tall trees which is unnecessary and like that. So we try to extract the leaf extract and then try to remove cadmium again. So at that time my focus was most toxic chemical, and it was that is cadmium, and it was published in Ecotoxicology and Environmental Safety. Then there is a then we thought that no, we can go a little bit more, so then we extracted the manganese dioxide from the spent battery, a discharge battery. We extracted manganese dioxide from that. We extracted the silica. We extracted calcium from the human urine, and we try to remove parakeresol. Parakeresol is very stinky chemical, which is present in the urine of the many animals. So it was a poultry farms and those animal husbandry. This is a big problem, so we try to remove that one. Tylosin is a macrolide antibiotic, and we try to also remove using this activated carbon and allogenate and MnO2. So this is again hazardous material. But in parallel, and always you can find a local collaborator as well, because my another colleague was working on vermiremediation, and I told why don't we try to see that whether vermiremediation would work on sludge treatment or not. And we went ahead, and all earthworm get killed. They could not grow in the severe sludge. Then we start adding cow dung, and at that time we published this ecological engineering. It was probably the first kind of proof that vermiremediation, even earthworm can be used to fix the metal content in the severe sludge. Severe sludge are very good in nitrate and phosphate, but it has high metal content, which makes it toxic. I would not go on each paper that what I have done, but there were many vermicomposting related work we keep on doing, and which is highly cited. And also by that time, I was a bit of, because my basic the research I was doing on the groundwater quality. So arsenic and fluoride has become my the problem, which was limiting the potable water availability in the villages, in the to the common people, and people has no choice. And you know this arsenic problem. And the first time ever, I established the fact that arsenic and fluoride can coexist. Earlier, it was like fluoride is a problem of semi-arid, arsenic is a problem of alluvial area. But it was just because something was more, we didn't pay attention to the less. But when I wrote the work, that this is not the case, it can exist together as well. So of course, in the why I say partnering sustainability, because we always focus on what is our waste. And so there was one waste come across is corn cob. Of course, in the winter, this is our, if you go to mountain, this the corn cob is very much. And so we extracted silica out of it. We already extracted the calcine from the precipitate from the urine. And we used them for several removal aspects and like that. But here that I want to bring to you is about what we did with the arsenic and removal. After having this kind of experiment, there were a Wind Foundation. They came up and they funded this project when I was with IIT Gandhinagar. And so when they funded me, even if I had proved that arsenic fluoride can exist, I wrote this review paper. You can see I was the first author. And there we proved that arsenic and fluoride can exist in the same area. And it is something which if we do not, because how many villages have the piped water supply? So most of us are dependent on, if we, the groundwater heavily in our country. And if groundwater got the problem, we really need to address it. So we proved that even in China or even in India, there are the fluoride and arsenic problems are coexisting. Of course, I don't want to say tell you that arsenic causes carcinogen cancer and fluoride causes you even crippling fluorosis. You cannot even stand. But I would also not tell that I was the first person to think about removing arsenic or removing fluoride. But I was the first who was thinking to remove both at one time. So in that case, to reduce the arsenic concentration, we had this kind of method. There were several the persons who were doing the publishing and mechanism. Again, I referred you in the first slide, and then again, I am referring it here. Then this is just something that everywhere we have found, that arsenic and fluoride. But one interesting thing has happened is that when I was, I had a sabbatical leave, and at that time I happened to study a little more. And then I realized that uranium uranium oxides and arsenic oxides are likely to behave same. These are a kind of, we had 5 samples collected. That time I was in the US, and I called my student and I said that the 5 sediment where we found high arsenic, can you get it analyzed in the EDX SEM and check whether there is a uranium or not? You may not be, or it sounds like a very exciting thing, but all 5 samples had uranium itself. So then we wrote arsenic, fluoride, and uranium coexistence in the thing. That was the first work of mine which came in the newspaper. But of course, that is the thing. But I wanted to have something else. I wanted to have something which can work for arsenic as well as fluoride, and they both are very different as elemental wise. So you can see that. So then at that time, I came across with this Janus particle, or the god where there is a 2 face. So then idea popped up is that can we have the adsorbent which have the 2 different surface? One will remove arsenic and one will remove fluoride. Well, at the conceptual level, it seems great. But when we start doing it, there were a lot of problems. So you can know that about that. I don't know. So there were, so we had this experimental plan that we will remove arsenic fluoride. But we never forgot that we have to, challenge is not only to remove arsenic fluoride, but challenge is to struggle with other anions or cations in the family, present in the water. In a groundwater, there will be chloride, there will be sulfate, there will be nitrate. They are major anions in water. How our adsorbent is going to remain competitive or selective for arsenic and fluoride? So that was the bigger challenge. So we said, okay, even if it works or not works, but let's try it. And so invention comes with the trial and error method only. You have to try after again and again. And that's why you know that the famous bulb experiment and those things. So we have done, and then we thought that okay, we planned some of the experiment that we will put arsenic chloride selectively or step by step, and we wanted it to be cheap, and it has to be regenerated. It means once the sites are exhausted, we should be able to regenerate it. So what to do it? Should we work in composite or several material? So there were steps that we followed, but the most important step was to do literature review. And when we did the literature review, we found that we wanted quick remediation. So we found this Liu et al. 2012 paper, which where equilibrium time was 40 minutes. It was working at 7 pH, and it was absorption of that. And we wanted that there should be a pH, like many adsorbent works at very acidic pH. Then it doesn't work. Then it is not a good adsorbent. Then we saw that equilibrium time, okay, and then DMA is a good functional group that can help. So that like that we have picked up, and here you can see that iron, the nanoparticle can help. So then, of course, there was a talk on the nanotoxicity, but of course, if it is covered by the several things, so we have done cost-benefit analysis also, that at the end of the day, everything is going to be determined by the cost. And we realized that if we take the clay, red mud, these kind of substance, maybe it will work more. And ferric oxide is also very cheap. So it means something which is above than this, the ferric oxide and above. That is what we wanted. Genus particle, of course, I can tell you when the time will be there. But of course, there is a method to follow, and you can make double phase adsorbent. And then we wanted to install this in each hand pump, and so that in the hand pump itself start giving the clean water. So objective were that let's develop the genus particle which can work both and which can help to removing geogenic contaminants. We cannot say that don't remove nitrate, because nitrate is also minus charge, fluoride is also minus charge, and those things. But of course, if absorption sorption process and those things, and it has to be recyclable, it has to be it should help. So these were the an innovation point was that we want to challenge coexisting ions, that, well, even in your presence, we will be able to remove arsenic and fluoride. And if not in one go, maybe in several steps, or cascadal, if the way that, okay, stage-wise, that if 10 ppb becomes 5, then 5 becomes 2.5, it's done. So even if, so that way, we wanted to have this thing plan. We did several tests. And after several tests, we could achieve, we made one compound that we named as win-win composite. Win because the win was the funder, and win, yeah, of course, because it is win-win situation for us. So we have seen that how it is removing with the fluoride, how it is removing with the arsenic. And here you see the competition. Fluoride, okay, 68%. If chloride is present, 63% removal. If nitrate is present, 57%. Only arsenic, okay, the 57.61. And if water has fluoride and arsenic both, it is 55% of removal. So it somehow brings down half the concentration. So I can pass 2 times or 3 times through that material, and maybe I can get the desirable amount. But I didn't want to remove it zero. First step is that let's bring down it to the less than permissible limit. So we made this substance, and it will look like this. And there were several experiments. If you will, whenever you will ask me, I will do. I will let you inform. But this was our composite work, which we had 4 criteria. It should be non-toxic, because nanotoxicity and less contact time should be required, and comparable adsorption has to be there between among the other adsorbent that is present already, and there has to be less cost. And in all this, our this composite stands good, And then we thought that, okay, let's go ahead. And we compared it with only allogeneic or which were very promising adsorbent. And we saw that our substance is much better. Then we have filed this patent, which is, of course, under consideration. And we, after filing the patent, we published it in just arsenic part in Journal of Hazardous Material. And of course, those things were there. Now, I am left with 4 minutes. And in 4 minutes, I will give you a little bit that, but we went one step ahead. We went one step ahead is that we thought how to manage sewage sludge and how to extract something meaningful with the sewage sludge. Idea behind was that in India, we don't have too much lined drains. There is an unlined drain. So a lot of soil component was there in the sewage sludge. So this was a PhD of my student at IIT Gandhinagar, where we just focused his PhD that let's do it, how to remove it, the arsenic, the sewage sludge, and how to get meaningful substance out of it. I would not say that there is no time to tell you that how sewage sludge is bad, but sewage sludge, even if being rich in the nutrient, it has high toxic elements, and that can affect the point. And lot of waste and sewage sludge is produced all over the world. Sewage sludge management is one of the very important aspects. We published work with where we converted sewage sludge into brick. We converted sewage sludge into the construction material. We gave them provide some. We did some geotechnical modifications so that sewage sludge can be used in the road construction. So those things we published. But here, what I want to tell you is that we wanted to use sewage sludge for contaminant removal. This is also, there are several steps that in the nowadays we want to manage everything. Sustainability has never been so much focus. You know, see, I am heading at sustainability cluster, which is the two department, the civil engineering, health, safety, and environment at UPS, and that we call together as a sustainability cluster. So nowadays, so we want to get rid of the waste at the same time produce something which is useful. So our the shift that has happened in the last decade or two is the green engineering principle. So so we thought that geopolymer, can we extract geopolymer from the sewage sludge? And because geopolymer is just a aluminosilicate structure, and if it is there, then it can be a good adsorbent. And I told you that, so in that case, we did the extract geopolymer from 10 gram of the sewage sludge. We could produce 40% of the geopolymer, but of course it can vary from the sewage sludge to sewage sludge. So we took sewage sludge from one of the wastewater treatment plant in Gandhinagar, and then we mixed it with the geopolymer, then mixed it with the nanoparticle and our win-win substance, and then we went ahead with the removal of this. You can see that there are a lot of removal happen, and this is the functional group. That when sewage sludge has no functional group, when we heated it and make a calcined sewage sludge, all organic matter we oxidized, then it was some more functional group. But when geopolymer had more than that functional group than these two stages, but when we added with the nanoparticle and those things, the functional group and their capacity has got a boost. And then we have done the removal, and we removed arsenic using that, and we used sewage sludge to remove the arsenic. We have seen here recyclability. Recyclability means until 5 cycle, the first time if you will use that substance, it will remove 30%, but in the 5 cycle, it will be until 15% or 20% of the things. So still it is fine. So this is available. You can read these both work in Journal of Cleaner Production and all. What next? Before concluding that, I have 1.5 minutes left. That we want to do, we were not interested in building another wastewater treatment plant, but we want to have every hand pump be capable to remove arsenic and fluoride. I had a video which I can you can look at, but that one, because we thought I thought that this is the process that the revolution will occur the same like mobile. Mobile is a decentralized information system. This telephone, which was kept at one place in the home, was a centralized system for a house. And see the revolution that mobile has bring in information in India or anywhere. So we thought that whatever the adsorbent that we made, we will stitch them with the filter. That filter that is there in the hand pump. And so we can put the hand pump in there, and we can put across, and contaminated water will come and clean it. We had several ideas that whether we can put it in the down, and then it can come, because we do not have luxury to take the hand pump out of the system every time and change it or regenerate it or something. So we have been thinking of the several plans that there can be a this kind of the structure, that a open well, and then it will go into there, and then we can pump it out. This is so this has to be tested in the pilot scale, but this is what we have been thinking. In order to put it in a like you can say that every hand pump, I don't know that the need utilization can have the different peak in the morning or the afternoon or in the summer or winter. So then we came up with this idea that permeable reactive barrier, that we know that groundwater always follow the slope. Groundwater, there is a direction in the groundwater. Can we put this as a wall of the permeable reactive barrier? It is a fact in the US, in the around the several contaminated places. And so maybe permeable reactive barriers. So whenever this is, it is at the last, if water is not good enough, we can change the material between them. So permeable reactive barrier can be also the part of that. The conclusion slide, of course, I am now so waste-based remediation resource are way to go about the sustainability, and traditional ecological knowledge is a savior. And then also is that the lab around the world, like GBRC or the other people, should come together to make a consortium to advance the field of remediation and sustainability. Thank you so much.
Thank you, sir. Thank you very much, sir, for enlightening us about the environmental issue like contamination in water. I think the sustainable technologies through bioremediation is the future of to combat the water pollution. So the session is open for the question. If any participant has question, please write in chat box. Any participant? So no, I think no questions are there.
Yeah.
Thank you, sir. Thank you, sir. Thank you very much, sir,
Thank you
for your time and being here. Thank you, sir. Now, for the next session, for the next lecture, I would like to invite Dr. Datta Madhavar sir for his talk on Phycobili proteins from cyanobacteria, a therapeutic perspective. Welcome, sir. Please allow me to introduce you. Sir did his PhD from Birla Institute of Technology and Science, Pilani. Currently, sir is working as an advisor as a PD Patel Institute of Applied Science, Charotar University of Science and Technology. And he is a recipient of various awards, including Lifetime Achievement Awards of Biotech Research Society of India for the year 2019, Lifetime Devotion Award as Microbiologist Society India, Fellow of International Bioprocessing Association. Sir has, during his tenure, sir has published more than 500 papers in various international and national journals. I welcome you again, sir. You can start and can please share your screen.
Thank you very much, madam. Thank you very much.
Thank you, sir.
I'm just trying to share the screen.
Yes, sir. No problem.
Yeah. Just a minute. Stuti. शेयर कहाँ से करूँ? शेयर किया है, ये यहाँ को लगाओ।
हाँ सर, वो सिलेक्ट करो ये वाला।
ये वाला कहाँ से?
ये यहाँ पर।
ऐसे करो, यहीं से
क्लिक
करो।
करो, हाँ। अभी शेयर।
ओके।
अभी स्क्रीन आपका शेयर हो रहा है, अभी पीपीटी खोलिए।
पीपीटी खुली है।
तो
Are you able to see my screen?
Yes, sir. Yeah.
Hello.
Yes,
Are you
sir.
able
Yes,
to
sir.
you are able to hear me?
Yes, sir. Yes, sir. Yeah.
Thank you. Thank you.
Okay, sir.
Okay. Yeah. First of all, let me thanks Dr. Chaitanya Joshi, Dr. Madhuri, and also all organizing committee for inviting me to share my information with this August gathering. Thank you very much. I thought of talking something about the characterization and therapeutic application of Phycobili protein from cyanobacteria. As we all know that the cyanobacteria are considered to be the oldest branching group of prokaryotics. They have evolved almost about 2.5 billion years before. As they are widely, they also possess what is called oxygenic photosynthesis, and it is believed that today's oxygenic atmosphere is mainly due to the contribution of the cyanobacteria. They are widely distributed. They are found in the aquatic ocean as well as terrestrial. Not only that, they are also found under the extreme situation, such as hot spring, polar regions, or saline deserts. I mean, and since they came into the existence so early, they have undergone into the various abiotic stresses. And in order to cope up with these abiotic stresses, they have developed, they have got, you can call it that positively pressurized to develop certain molecules, to produce certain molecules to, you can say that they can themselves grow even under the abiotics. In the process, they have also produced various types of the pigments. In our lab, actually, we were maintaining almost more than 150 cyanobacteria, and we were exploiting these cyanobacteria for two purposes. We were trying to purify the Phycobili proteins from this cyanobacteria. We were going for the structural characterization and therapeutic application. At the same time, we were trying to isolate the mycosporin-like amino acid. And these mycosporin amino acids, once they are purified, they can be used in the cosmetic, especially as a sunscreen lotion. Cyanobacteria basically possess a photosynthetic mechanism as that of the higher plant. However, they differ in their light harvesting apparatus and functioning. If you look at the plants, plants have ability to absorb light radiation starting from 650 to 720 nanometer, and that is mainly because of the chlorophyll content. However, cyanobacteria have ability to have a wider wavelength range. They can capture the light energy starting from 450 almost to the 720, and that is mainly because that they contain some protein known as Phycobili proteins in association with the chlorophyll. And therefore, we can say that their pore window is enhanced or increased significantly. These Phycobili proteins are considered to be a family of brilliantly colored pigment proteins. They are highly fluorescent. They are basically made up of two dissimilar peptides, and we call them alpha and beta subunits. Each subunit may be about 20 kilodalton, containing about 160 to 180 amino acids, and they have a helical globule-like structures. Though they are dissimilar in terms of amino acid sequence, in spite of they are dissimilar, they have a similar folding patterns. So they form a chain, and they are covalently attached through their cysteine acid, a chromophore group, which is a porphyrin. Based on the chromophore group, Phycobili proteins are divided into three groups. Basically, there are four actually, but mainly we talk about three major things. One is called phycoerythrin, then phycocyanin, allophycocyanin. And their spectral properties are something like that. Phycoerythrin, as indicated, it is pink in color. Phycocyanin is cobalt blue, and allophycocyanin aquafin. Like any other molecules, the protein part is synthesized. Starting from you have a corresponding genes through the transcription and followed by the translations. These alpha beta subunits are synthesized. We call them apoproteins. And similarly, the chromophoric group is synthesized from a precursor, what is not HEMB, and different types of the chromophore groups are synthesized. With the help of enzyme lyase, they get attached through the cystine inlet and forms a functional, hollow Phycobili protein. These Phycobili proteins, they convert into the very giant structure. We call it photosynthetic unit. We call them as phycobilisomes. Now, in the phycobilisome, when you talk about, it is available on the thylakoid membrane of the cyanobacteria. Alpha beta subunits, which are already appended with the chromophore, they combine together, forms a heterodimers. Three such heterodimers further get joined together, forms a trimer. In case of allophycocyanin, these trimers are stacked one above other. With the result, they form what is known as core cylinder, which is at the bottom. Whereas, in case of phycoerythrin and phycocyanin, these trimers further again get joined together, forms a hexamer. And these hexamers are again stacked in the formation of the rod. So what you find that at the bottom, you have a allophycocyanin, and from this center, radially in perpendicular, this so-called phycocyanin and allophycocyanins are getting arranged and forms a very giant structure. In addition to these, they also possess non-colored linker proteins, and they form a. This is how they form. Now, these Phycobili proteins have multiple applications. They are used in the food industries as a colorant. They make a fluorescent color, so you can use in the various form in the food industries, like whether it's a milk product, whether it's a dessert, whatever it may be. They have also application in the pharmaceuticals as an anti-inflammatory, neuroprotective, hepatoprotective, and those things. Because of the very strong fluorescent colors, they also have an application in the various cosmetic industries. We have also noticed that they have a role to play in anti-aging, anti-Alzheimer's. Maybe by further talk, you will have the more details about this. At the same time, they are strong antioxidant, and since they are antioxidant, they can be useful in the nutraceuticals. And they can also be like green fluorescence protein. This is also a sort of a fluorescent probe, can be used for as a tag for variety of purposes. What basically we are trying to do that, we go for the purification of this phycobiliary protein using the different types of the cyanobacteria which we have in our lab. We develop the cost-effective process. Then we try to get the genes corresponding to this protein. We also go for the mass spectroscopy for the protein so that we have a little more detailed information. And if possible, we go for the highest level of purification, which is followed by the crystallization, get the X-ray diffraction data, and based on that, then we are able to get the three-dimensional structure. We also perform the docking studies, whether how they can be used as a drug. And then we have a plan to use a single polypeptide, which can be applicable for variety of purposes. And we use C. elegans as a model. We have also done the experiment with the Drosophila and eukaryotic cell lines. We have developed fairly good number of protocols for the purification. These are all the conventional basis where we go for the precipitation, followed by the dialysis, then gel chromatography, and so on, so on. But what we have done small modification and here and there. For example, one of the very well cited paper where we have used the Triton X100, and that has helped us for the resolution of all the three proteins so well. Similarly, when we went for the purification of the allophycocyanin, we have used ethidine as one of the components in the during the process of purification that has helped. And then obviously others like ion exchange, hydrophobic interactions, various protocols, and we have come out with several protocols, and they could get published in fairly good journals. Very interesting that we were in fact the first persons, rather it is the first report from India also, where from one of the organism Formidium, we extracted a phycoerythrin, and that phycoerythrin happens to be a single polypeptide. It was a single peptide molecule. It's not poly, it is single peptide molecules, and that we could purify and went for the crystallization. And we got the X-ray. This work was carried out in association with the Professor T.P. Singh at the All India Institute of Medical Science, New Delhi. And we could resolve the structures to the level of 2.6 nano by X-ray. And what we noticed that this particular polypeptide is divided of 31 amino acid at the end terminally. And we initially thought that since it is a truncated molecule, it may not undergo into the polymerization, it may not be able to form the, but we found that this is still functional. And what is happening in the case of cyanobacteria, and when the cyanobacteria is grown for a longer period of time due to the nitrogen deficiency, they use some of these amino acid as a nitrogen source for them. Another protein which is called phycocyanin, which was isolated from the leptolingia from this molecule was also purified. We have done the X-ray diffraction study of its crystals, and then we did the docking studies. As we all know that one of the enzymes which is involved in the plaque formation during the Alzheimer, that is the beta secretase. So we try to find the efficient or the what you call that binding efficiency of beta secretase with our protein, and we noticed that we tried with the different types of that. We noticed that phycocyanin has a higher ability to bind with the cell, as indicated by the negative value, much higher negative red color one, that indicates that they have it. So it seems that it is a better molecule than already existing molecule, which shows the higher, so that beta secretase, if it is not available, there will not be plaque formation. So that's what one of the things. Like that, we also got the purifications of the various proteins from the another organism which is a marine origin called the Lingbia. And from this organism, we isolated 3 different proteins: phycocyanin, phycoerythrin, as well as allophycocyanin. And we did the protein purification to the highest level, did the crystallization, and got the X-ray. A part of our proteins, of course, were sent to the Grenoble to get the X-ray diffraction data. And later on, we were using the BRC facility they have in the Indore, and we started getting the data from them. And then we could get the 3D structures, and we could, what do you call that? We submitted this data, we provided, and these data have been published in the variety of good journals. So this is about the phycoerythrin, where it has got the 2 chromophore in the alpha regions, which is attached to the 82 cystenyl and 139 cystenyl. Beta has a 3 chromophore group, of which one is attached to the at 2 locations, 48 and 59. Another is at 680 and 165. And we noticed that in this particular, when it is compared with the already known phycoerythrin, we found the insertion of 9 amino acid residues. Like that, we have done for the phycocyanin also. We did it for the allophycocyanin from the same cultures, and we could edit. Now we wanted to understand about the application of this phycocyanin. As we all know that under the stress condition, whenever there is a stress, the RNSs are generated. And as we all know that these reactive oxygen species which are generated, to certain extent, our cellular enzymes are able to take care about. There are certain scavengers already available in the cells, which somehow absorbs, and these ROAs are not allowed to carry out its role. But if they are producing much more than what our defense mechanism has, then obviously we all know that they will create variety of other consequences. Whether you talk of protein oxidation, DNA damage, Fenton reaction, transport to the cytosol, and so on, lipid peroxidation. So various things do take place, and obviously it can lead into the various complications. We are trying to correlate reactive oxygen species with the aging. We believe that whenever there is an oxidative stress, the normal proteins would get unfolded. And depending upon the level of the unfolding which has taken place, maybe this protein undergo into the natural protein degradation by proteasomal degradation process. Some of these unfolded protein may undergo into the aggregations, and that can lead into the aging. Or if they are partially unfolded, maybe repairable, they may come back to the original form also. What is our theory is that we say that whenever there is a stress, the ROS are generated and these ROS has a role in the aging. So oxidative stress is responsible for the aging. What you require? You require to take care about that. You require antioxidant. So that antioxidant will work as an anti-aging. We notice that Phycobili proteins work as an antioxidant. Therefore, Phycobili can be used as an anti-aging agent. The conventional methods of in vitro antioxidant activity, whether it's a DPH, ABS, PAB, or whatever, these assays can be carried out and they can be compared with the control molecules, and thereby you can understand that these molecules have a very high antioxidant activity. We wanted to show its effect in vivo. So what we did, we used C. elegans as a model. C. elegans is a very comparatively simpler worm, you can say. It's a nematode, and it can be grown in the laboratory very easily. You can have variety of C. elegans, wild type, mutant, transgenic, and so on. From it, there is a center, which is at the Minneapolis, that is the University of Minnesota. And they have a Cerebraptids genetic center, and one can procure these C. elegans, and they can be grown in the laboratory. What you require a simple BUD incubator where the temperature is maintained at 20 degrees centigrade, and their food is, there of course you have a nematode growth medium, but their major food is that E. coli. And you have a specific E. coli strain, 05P0. That particular strain can be given as a feed to the C. elegans, and you don't have to go through the various animal ethical committee and so on, and you can perform the experiment. In general, wild type C. elegans have a lifespan somewhere around 15 to 20 days. What we notice that when the same C. elegans are fed with the phycoerythrin, of course we have done with the dose dependent studies and so on, and we notice that they show the extension in their lifespan, at least by 20, 30%. And then there are certain surrogate markers which we can check, for example, pharyngeal pumping. And this pharyngeal pumping can be counted by seeing the rhythmic contraction in the pharynx portion. And with the stopwatch, you can measure the number of contraction is being taking place in this. If you have a normal C. elegans in a first day, you will find whether they are fed with the phycoerythrin or they are not fed with it. It's more or less same. But as the time passes, you can notice that on the fifth day, the contraction frequency, or what you call that rhythmical contraction of pharyngeal activity, is reduced. But in presence of phycoerythrin, the activity is still higher. Same is the case when you go for the next 10 days. So like that, we've noticed that phycoerythrin helps the C. elegans to survive for a longer time. It is found to be highly active. Not only that, you can also check the locomotion assay. The body turns which takes place. In a minute, how many body turns it takes place, which can be measured. And with this also, we notice that it has much better ability. We also gave the external stress, such as heat stress. Instead of growing at 20 degrees, if you grow them at a higher temperature of 35 degrees centigrade, obviously, they are unable to cope up and they die soon. But if you feed the C. elegans with the phycoerythrin, their heat tolerance ability is increased. Not only heat tolerance, if you give the oxidative stress with the help of the hydrogen peroxide, if they are exposed with the hydrogen peroxide, depending upon the concentration, you will find that in presence of phycoerythrin, or you can say that C. elegans, when they are fed with the phycoerythrin, they have a better ability to sustain that environment in presence of oxidative stress. Of course, there are several theories which are associated with the aging. I'm just trying to capture a couple of them only. And one of the say that it is insulin or insulin-like growth factor IS pathway. In this IS pathway, there is one particular transcriptional factor, what is known as DAP16. That's DAP16. Actually, whenever there is a stress, it enters from cytoplasm into the nucleus and able to activate all those genes which are responsible to combat are to take care about the oxidative stress, are to take care about the any sort of a abiotic stress. But DAP16 in turn is controlled by the age. They will carry out the phosphorylations. Age is when it's controlled by the DAP2. So what we notice that obviously, so what we have done is that we carried out small experiment. For example, we have used the one transgenic C. elegans which has a DAP16, and that DAP16 gene is attached with the GFP protein. And we notice that when there is a, depending upon situations, if we have a C. elegans which is in a normal conditions, it will find that DAP16 is going from cytoplasm into the nucleus, which you can measure by the fluorescent. Whenever there is a, you can see the fluorescence by the green fluorescence, which we can notice. And similarly, when you are fed with the P, it also again shows the fluorescence. That means that this having the PER no protein, that entry of DAP16 from the cytoplasm to the nucleus is. Then we use a DAP mutant, which is the DAP16 is not working. And when DAP16 is not working, obviously when C. elegans is given and grown, it has a certain pattern of the growth. But in presence of PE, it shows the enhanced lifespan. So they have a better survival condition. Similarly, we have the mutants of the DAP2 as well as age1. And similar thing, it indicates that DAP16 was independent of the P. Or P do not have any direct relation. Presence of P certainly helps the C. elegans to enhance its life. So there could be another one. So we thought of heat shock factors, which exert its effect. So what is happening at the heat shock? So here in this case, what we did, again we took the C. elegans, and there in a normal course, what factor if you can see it here, yeah, you can have a normal, but you have a control, which is the C. elegans. In a normal condition, it shows the pattern of its survival like this. When this control is supported, with the phycoerythrin, that means when it is fed with the phycoerythrin, we found that there is an enhanced growth. This is what normal. So what we did, we thought of silencing this. To do the silencing of this, we use an E. coli cell, and that E. coli cell has ability to produce double-stranded RNAi. And that E. coli were fed to the C. elegans. In the process, the C. elegans now able to have, or you can able to silence the heat shock factor gene. And once it is silenced, it is not able to. So under the silence condition, for example, if you have this is a silence condition when RN, it also shows the typical pattern of the survival. But under this condition, and if it is fed with the P, it has not improved any survival. It means that P is associated with the heat shock factor. It has a flow to play with this. Not only that, we showed that it has the ability to show. For example, when the C. elegans are exposed to the oxidative stress, and one of the oxidative stress which is given by the paraquat, it's a herbicide. When you have a normal C. elegans and they are exposed to the normal condition, in a normal, you do find, and then this fluorescence, the amount of the ROS which are available in the cell can be detected by this state. Higher the ROS, you will have a higher intensity of fluorescence. So in a control conditions, you would have some level of ROS, but when the same C. elegans are fed with the PE, it shows absolutely no fluorescence. Indicate that whatever the space has been there, whatever the ROS, they have been removed by this or they are scavenged by this. When the same C. elegans are given a treatment with the paraquat, obviously the ROSs are higher generated and you can see the fluorescence level with this very high. But when these treated one are further fitted with the PE molecules are treated when given the paraquat, it has a beta, so you don't find any fluorescent. Indicate that. Not only that, we have seen what is happening in this. With the fluorescent microscope, we can measure the intensity of these relative. So intensity of these and these is very comparatively high, but when they are with the PE fitted, they show the low intensity. It means that ROS generation is comparatively low or they are removed from that. It is not only done with the C. elegans, we have been able to do it with the mouse fibroblast. There also we can see that it is in control condition. When they are given oxidative stress, you can see that fluorescence which is available, which is seen, that is ROS generation, which is stained by this. And when they have feeding, the obviously ROS level is low as indicated by the laser, and which can also be measured in terms of the intensity. This is about the human skin microburst. So at cell line level also, we are in a position to show the similar results. We also measure that, for example, Drosophila melanogaster. In the case of Drosophila, we check the locomotory evidence in a control conditions. When it is a paraquat is not exposed, you have in presence of P fitted, you will find that higher locomotor, and in case of the paraquat exposure, you will find that in the control one it has very less, and here it has a higher locomotor, which can be measured. We can measure the Drosophila movement by how the climbing facility. And we also measure the intracellular one. Whenever there is an ROS generation or whenever there is a stress, the ROS are generated, and that ROS will lead into the production of more SOD, superoxide metals, or catalase. These enzymes are produced. But in presence of PE, their production goes down. That means that PE is able to take care about it. Very interesting phenomena which we have noticed. This is in case of the Alzheimer model. We have used a C. elegans, which is a transgenic C. elegans, and this has an Alzheimer model where what is done that the gene what is known as A beta 142, which is made for the Alzheimer, that gene is placed into the muscle cells of the C. elegans, and in that gene is placed in such a way that it is placed in front of the temperature sensitive promoter, so that when the temperature is shifted from 20 degree to 22 or 25 degrees centigrade, that gene will be expressed and it causes paralysis in the C. elegans. What we notice that paralysis effect is reduced significantly when the same C. elegans are fitted with the PE, and that's how with. So it is a very interesting thing which we have done. We are still trying to understand the whole mechanism behind it. So in conclusion, what we say that phycoerythrin or Phycobili protein is responsible to extend the lifespan and health span of the C. elegans, which has an anti-aging property. It has ability to improve the tolerance against the thermo or oxidative stress. It works independent from that of IS signaling pathway, but it requires what is called heat shock factor. And it reduces the intracellular ROS level in C. elegans, and it has variety of applications as we can see from our publications. And this is the work which is done not by me, but all my students and postdoc who have been associated with me, and it is the present work which has been done is mainly done by the Ravi and the Stuti who has helped in this, and all others have been associated with me and they have been doing. It is a cumulative and we have been able to do. So all credit goes to these students who have been doing, and my job is to keep them live. I require to provide them fellowship, so I require some funding, I require to have a facility. So that is all been done by the various funding agencies. Thank you all. Thank you very much. Thank you. Hello. Hello.
Hello, thank you, sir.
Thank you.
Yes, sir. Thank you, sir, for your enlightening talk, and it is very interesting to know the uses of therapeutic implication of Phycobili proteins. Now the session is open for the questioning. If any participants have any type of question, you can write in chat box. Any questions? Okay, sir. I think no questions are there. So we are going to proceed for further. Thank you, sir. Thank you very much for sir for your time. Thank you, sir.
Good afternoon, Dave sir. Good afternoon. You are on mute.
Yes, sir. Shall we proceed for the next session?
Yeah, thank you. Yeah.
Yes, sir. For the next session, I would like to invite Dr. Shailesh Dave sir for his talk on phytohydrotechnology for non-ferrous metal recovery from the solid waste for environmental protection. I welcome you, sir. Now, please allow me to introduce you, sir. Sir has completed his PhD from Mysore University and presently working as the Managing Director, Loyola Center for R&D Xavier Research Foundation. Sir was associated with Department of Microbiology and Biotechnology in Gujarat University in 1982 for around 35 years and has immense research experience in various fields like industrial microbiology, microbial genetic, bioprocess technology, biogeomicrobiology, environmental microbiology, extremophiles, microbial diversity, and has more than 140 publications to his credit. I welcome you again, sir. Sir, now you can start with your lecture and can share your screen.
Just 1 minute.
Yes,
I'm sharing my screen.
yes, sir, visible.
Good afternoon to all of you.
Good afternoon, sir.
I'm audible?
Yes, sir. Yes, sir.
Okay. So I would like to thank the organizers of the Vigyan Gurjari as well as the Gujarat Vigyan Sammelan, particularly Dr. Joshi, Dr. Madhvi, and those who are looking after these two associations. Today, I'm going to talk about the biohydrotechnology of non-ferrous metals, recovery from a solid waste, and environmental protections. Now, my work is a little bit easy because the previous speaker, Dr. Manish Kumar, now he has worked and he has explained how this bioabsorption of the metal can be taken place. So I am not considering the removal of the metals from the liquid waste. I will mainly focus on absorption, leaching and absorptions of metals from the solid waste. The major source of the waste, what we know is the agriculture waste, domestic waste, electronic waste, industrial waste, medical waste, mining waste, and waste which is generated without our knowledge from the research and academic institutes. All this waste, they are responsible for contaminating the environment in one or the other way. I am mainly focusing on the contaminations or the pollution of the environment with reference to metal ions. And if we consider why the environment is become unsustainable, what are the main causes of the environmental pollutions and unsustainability of the environment, we know that the day by day, as our requirement is increasing, we are going for the industrializations, urbanizations, increase in the populations, excessive use of the resources, excessive use of the hazardous chemical. If we take the simple examples, then farmers are using excessive amount of the chemical fertilizers, pesticides, insecticides, all these things. No, they will add chemical, one or the other chemical in the environment. Then, as far as the treatment part is concerned, at many places, the improper treatment of the waste may be also responsible for the instability of the environment and increasing demand of the sophistication and comfort. So for this reason, we require more and more materials to be consumed, and for that the resources are depleted. If we come to the specific metallic pollutions, and what I am concentrating more in my today's talk, that is, if you look to the present generations, then present generation is changing their mobile phone at the end of every 2 years or maximum in 3 years. And if we look to the data of the generation of a mobile phone waste or the electronic waste, which is tremendous. Sometimes the unavailability of the required treatment may be also responsible. As for example, no, there are certain industries, as for H industry, H acid producing industry, no, there is no proper technique today available for the treating of that waste. And hesitation in accepting the microbial technology. The strategy, what may be responsible for the increase in the pollutions. Before 50 years, no, it was a normal slogan that disposal is the best solution of pollution. And today also, even due to the various restrictions also, people are disposing that pollutants or the waste material without treatments. Second thing, which the people were believing, dilution is the best solution of pollutions, which is responsible for the contaminations of a water body, starting from the lake, river, and even the ocean. Then whenever the pollutants are there, and if it is possible to incineration of that pollutants, we think that we got rid of the pollutants, but the pollutants are converting into a volatile compounds, and many noxious gases are end up into atmosphere. Then the thinking about the land filling, particularly when the things are non-biodegradable, and if it is used as a land filling, that leads to slowly and slowly leaching of that toxic material which penetrate into the surrounding water body and pollute them. Many times, partial treatment, due to the economic point of view, we are not treating the material up to the end. And whatever the intermediate produce, that intermediate may be toxic or sometimes more toxic. The best example is if you are dealing with the azodyes, and if you are treating this azodyes, and if you are going only for the anaerobic process, which is the normal first stop, or the microaerophilic process, then the amines are produced, which is carcinogenic and more toxic as compared to the even the dye itself. Implementation of a cheaper process for the economic point of view, that also leads to the increase in the more toxicity. And our unawareness. Many times, the larger populations are not aware of the toxicity of the pollutants. They are not aware of the what harms that the pollutant will take, leads to the human being that also responsible for the pollutions. Now I'm coming with a general introductions, how the metallic pollutants, what are the sources of metallic pollutants, and I'm concentrating on the biological methods, the major sources of a metallic pollutions, which is divided into mining waste, such as tailing, low-grade ores, mine overburdens. No, but many times this particular waste are overlooked because almost all the mines, they are in a rural area, much away from the urban area. And if the contaminations or the pollution took place due to the mining activity, it is not noticed or it is overlooked. The another source of a metallic pollutions, where throughout the world people are more worried about in the 21st century, is the electronic waste. That what we are going to discuss, how you can remove the metals, how you can make the sustainable development eco-friendly process. Then municipal solid waste. If you go to any urban area and the municipal waste, as Manish Kumar has also discussed in that, the municipal solid waste is consisting of various types of the metal, which is immobilized in the form of metal sulfides. Metal containing used catalyst for any chemical process, catalysts are used, and many of them, they are either having the high concentration of copper or nickel or cobalt. And once this catalysts are used, they would be thrown out, which is having much, much higher concentration of the metal and if it is not taken care of properly, it leads to the addition of this metallic pollutants. Metal containing wastewater. Now, this picture will show you, as I told you, in the mining waste, which is the main source of metallic pollutions, and you can find the small mountain like of the material. This is the example of one of the sulfidic mines present in India. This is one of the lignite mine. On both the mine, now if you look to on the periphery of the mine, both are the open cast mine, and you will find the unused material which is not having the enough metal ion concentration present in this waste. Now they would be dumped on the periphery of the mines, and that dumping will be as high as like small mountains. And the area which is covered, that is in terms of a few kilometer area, now they are there. And this particular dumping of a overburden of the minerals, which any physicochemical method or pyrohydrometallurgical methods cannot extract the metal ion, they would be left over. After dumping, the post-soon method, due to the rainwater which will penetrate through this, and you will find the acid mine drainage, which is consisting of dissolved metal ions. And the volume of this acid mine drainage, it is not in terms of a few tens or few hundreds or few thousands gallon of the water, but it would be in terms of a million gallons of water after every monsoon. And the pH of this water would be below 2.5. Many times it reaches to below 2 also, and which is having copper, zinc, lead, nickel, and some of the other metal ion dissolved in this beyond the permissible limit. This is what we have to worry about the present day is the electronic waste. And this is one of the picture, but if you go to any country and any state or any city of the any country, you will find this type of the electronic waste are dumped. Amount of the electronic waste which is produced on a planet Earth in various countries, it will be in 2020, it was more than 57 million tons. It is not in terms of a kilograms or a few tons, but 57 million tons of electronic waste is produced. And all this electronic waste are having various metals present in them, and particularly all metallic pollutants. Till today, we don't have any method to degrade the metal. Either you have to make it less toxic or you have to remove it. So what procedure what we are using for the treatment of any metal containing waste, either it is electronic waste, it is municipal sludge, it is the used catalyst, it may be the tailing or lean grade ore, whatever it may be. Then you have to think about how carefully you can recover these metals. In case of the minerals and ore, depending upon either it is a copper minerals, copper ore, zinc ore, or it may be a multi-metal ore, you will find various base metal present in them. You also find the arsenopyrite containing ore, which is the main source of gold. So there you will find, along with the other metal, the gold present in them. And amount of the metals which is present in this ore are many times less than the amount of many of this metal which is present in the e-waste. In case of ore or concentrate, where you are having 1 or 2 metals or at the most 3 metals, while in case of the electronic waste, now list is very high. You will be having many metals and metalloids which are used for the manufacture of that particular electronic or electric materials, which are also a very good source of a precious metal, just as gold, galenium, germanium, silver, palladium, platinums, and more importantly, the rare earth elements, which most of the country, no, they are not having the enough source of the rare earth elements. And when we are going for the electrical and electronic equipments, many of these rare earth elements are used in the manufacture of these materials. Now, if we look to the sources and reserve of the rare earth element, China is blessed with the God, and which is having the highest reserve of the rare earth element on this earth. The rare earth elements reserve in China is nearly 44 million metric ton. And in 2020, they have processed 140,000 metric tons. India is coming third or fourth in terms of a rare earth metal reserves, but concentrations of the reserves is very less as compared to China. It is just 6.9 million metric ton of the rare earth metal is there. And as we are not having the well-developed technology to process, to leach out or to recover the rare earth elements, we are able to treat only 3,000 metric ton of the rare earth minerals. And this is the data of 2020. We, India, we are blessed with the longest beach and seashores, which is nearly occupying nearly 35% of the total seashore present, and which is also having the good source of rare earth metals. Now, I'm not going, as I have to complete this talk in 20 to 25 minutes, I will be not going into the various process. Directly, I will come to the biohydrometallurgy, which is a process dealing with the study and application of the economic or environmental potential of interactions between the microorganisms and metal containing waste or minerals, where microbial activity, either direct activity or the production of the metabolites, they will solubilize the metals in the form of sulfate, or they will solubilize the metals in the form of carbonates, and that can be extracted from these materials. If we come to discuss this main mechanism of metal solubilizations, in this field, initially the mechanism were classified as the direct and indirect mechanisms, but then after various researchers, they found that that particular terminology is not able to explain the mechanism of metal solubilizations from minerals and electronic waste. So then they said that instead of direct and indirect method, it is a contact and non-contact mechanisms. That also classifications require the modifications. And now we are having the understanding of a solubilization mechanisms, which is divided in three major groups: acidolysis, that is the proton mediated dissolutions, where microorganisms, they are growing, and due to the growth of the microorganisms, they are consuming the substrate present in that minerals or the substrate which is added from the outsides, and they produce acids, H-ions, and this production of the acids, most of the organisms, where we are talking about the sulfide minerals and when we are talking about the electronic waste. So the sulfur oxidizing organisms, acidithiobacillus thioxidans, and other group of the sulfur and the reduced sulfur oxidizing organism, they will oxidize sulfur to H2SO4. And that H2SO4 will work as the lixiviant for the extraction of many metals. The second mechanism is the redoxolysis, where microorganisms electron transfer take place. Either it is the transfer from microorganisms to minerals or by oxidizing ferrous to ferric. Mainly the ferrous to ferric oxidations is playing the major role, and where acidic thiobacillus ferrooxidans, leptospirillum ferrooxidans, sulfolopus, and many other microorganisms, they are playing important role for the oxidation of ferrous to ferric, and that ferric generated ferric, which is used to extract metal ions from the insoluble material or the solid waste. And this particular mechanisms is mainly used for the extraction of metal ions from the sulfidic minerals as well as from the electronic waste. Another mechanisms which is also playing a significant role, where the complexolysis will take place, where chelation of a microbial metabolite, chelating compound which is produced by the microorganisms, which react with the metal ions and result into the stable metal chelating agent. Some of the organisms what we have worked with the complexolysis are Pseudomonas butida, Pseudomonas aeruginosa, and these organisms are producing HCN or extracellular cyanide compound, and this HCN will react with the gold as well as silver and form the silver cyanide complex, and this silver cyanide complex will be extracted. The production of HCN would be also useful to extract out rare earth metals. I have given only the 3 names, but it will work for many rare earth metals, and that rare earth metal can be separated by the production of a complexolysis. Now, to give you some idea regarding the various types of organisms, of course, I have tried to make it the summary of the organisms which are used for the electric and electronic waste. Acidic thiobacillus acidophilium, acidic thiobacillus ferrooxidans, lactospirillum ferrooxidans, then some of the heterotrophic organisms which are used for the extraction of some of the base metals, that is Aspergillus niger, Penicillium simplicium, and Aspergillus nigers, then Chromobacterium violens, which is used for the extraction of nickel and soil by the complexolysis process. And here our laboratory, as I am working in this field, I have started working on the extraction of metals in the 1977 onward. First of all, I used at that time the acidic thiobacillus ferrooxidans was known as a thiobacillus ferrooxidans. So I have used thiobacillus ferrooxidans and thiobacillus thioxidans for the extraction of zinc from a sphalerite concentrate, as well as extraction of the copper from chalcopyrite ore and chalcopyrite concentrate. Thereafter, we have developed the various combinations of this microorganisms, because when you have to work with a metal ions and the ore and minerals, you cannot work with a single organisms. So we work with a consortia, and in Gujarat University, I have developed the consortia for the extraction of polymetallic concentrate, where main constituents of the metal ion is copper, zinc, and nickel. And we have worked successfully for the extraction of metal ions. Then we have started working on the other multi-metal ore, because in Gujarat, we are having the Amba Mata mine, which is very near to the Gujarat border. And this Amba Mata mine, which is having the polymetallic concentrates and polymetallic minerals, and nobody was using this because the reserves are very small, so they cannot think about starting the pyro or hydrometallurgical process, and they have to depend upon only the biohydrometallurgical process. We have developed the process up to the pilot level productions. Meanwhile, that mining is sold from the Gujarat Mineral Development Corporations to other company, and the process is not further developed. In case of the last 7 to 10 years, now we have concentrated working on the electronic waste, where you will find either you can take radio, you can take tube light, TV, you can take the switchboard, or you can take the mobile. A different this get where they are having the various types of the PCB, and that PCB is the main source for working of your electronic waste. But these PCBs, now they are having the copper or other metal ions for the supply of electricity and currents. Here I have shown you the typical types of the PCB what we have collected directly from the factory. This is untreated PCB. Now they will be colored with an epoxy layer to protect them from this short circuiting. And this PCB, similarly, you will find the small PCB. Now here you will find very small PCB. They are from the mobile phone. So to give you the overall idea, this is the way how the PCB are available. And when we have treated this PCB, you can find the same PCB. After the pre-treatment, the epoxy layer and the color layer would be removed, and all the copper, you can see the copper which has been exposed. This is untreated PCB. After the pre-treatment, because directly, initially we were grinding the PCB, and after grinding, we are using directly for the extraction of metal ions. But when you grind the PCB, it's required the grinding cost. At the same time, when you are grinding, some of the small particles, they will be mixed up in the air. That is known as the dust particle, and that dust particle also have the many metal ions. And when you are grinding, it will increase in the volume, and the treatment become more and more process. So I think in 2010, we thought that instead of grinding the PCB, is it possible to treat the PCB without any grinding? And we got success. So this is untreated PCB. When we have given it the pre-treatment with the alkali solutions, so it removes the coloring part, and the copper linings are exposed. And when this copper lining are exposed, we treat with the biologically generated ferric iron, or with the help of the acidic peroxidants, leptospirillum peroxidants. And after the treatment, you will see that all the copper which will leach out, and the PCB. Now these are just marking without any metal ions. So, but it is completely the metal ion would be removed from this. As I told you before, working with a ungrinded PCB, we used to work with a grind, because most of the people in the world, they are grinding and using. So we have used with the various types of the PCB obtained from the mobile phone or from the electronic waste, and we ground it, and we have developed this type of the reactor where at a time you can treat in terms of a few kg of the ground particles. Here, what development we took, that when initially this organism were used, because all these metal ion, they are highly toxic compounds, and this organism cannot survive. So we have adapted these organisms. We have selected best functioning organisms, and we have prepared the consortia from this. And these methods, we also developed a membrane bound material. So grounded PCB powder were taken in the various types of the membrane what indigenously we have prepared, and they were filled with the metal ion in such a way, so when you pass air in the reactor, they will float. They will not just settle on the bottom of the reactors. They float, and that is very easy to remove after the treatment, the unreacted particles, and you will get this leaching of the various metal ions. This development, what we have patented, and this a graphical extraction of a metal recovery from PCB. So without regrinding or without grinding, if we want to use, then the original PCB will be treated with the NaOH, then you will get the removal of this. Then this is our reactors, where our developed consortia, which is mainly dominated by leptospirillum peroxidants and acidic peroxidants, which generate the ferric iron, where this PCB are dipped, and you will get that PCB, and the metal ion which is extracted copper here, because most of the PC they have 20 to 25% of the copper, and all other metal will be less than 10%. So where the PCB was removed and recovery of the copper can be done from this by various methods we have used, and then you will get more than 96.5% pure copper powder, metallic copper, and that PCB can be used for some other purpose. Now this will give you the idea that what initially I told you, that any sulfidic mine where you will get the productions of the acid mine drainage here. Now this is a Khetri mine, which was considered to be the first copper mine in Asia, and that people have extracted copper from this mine for 56 years. But in 2018, they found that a river, now this is a river like upstream is formed, which is 3 kilometers long and nearly 1 kilometer wide, and which is having full of water, nearly 15 meter depth, and which is having a concentration of copper, iron, sulfur, aluminium, silica, calcium, magnesium, and all these metal ions are very harmful and that can be removed. Now, during this journey, how we have developed the organisms, initially, when we have started without a primarily unadapted organisms, the process used to take 30 days for the extraction of metal ions. And that too, we were not able to work more than 2% of the pulp density. Slowly and slowly, the better consortium were developed, and after adaptations, the process time was decreased from 30 days to 6 days and 12 days for the copper and zinc. Then further development will took place, and we have gone for the development of the laboratory store tank reactors. Their time was reduced from 30 to 6 days and 8 days. And during this process, organisms were so developed and so treated that at present we can able to work with a 20 to 25% of pulp density. And we have developed up to the pilot scale productions leaching process, where also in less than 6 and 8 days, the extraction was possible. The physicochemical property of a polymetallic leachate, what we get. Now, if you look to this, now you will get surprised that how the organisms are surviving. And for that, you have to develop because as we want to use this in a field level, we cannot directly go for the gene manipulations, but we are slowly and slowly adopting the organism. It took nearly more than 20 years to adopt the organisms, and now the culture what consortia we are having, it can leach out metals which is resistant to nearly 14 to 15 metals which are present, not in terms of a ppb, ppm level, but in terms of a grams per liter level. They also work at a viscosity of the 11 cp and ionic strength of 7.45, and at present what we are having, it is 9.23 ionic strength. Now this process are also commercialized in many countries, and amount of the copper produced per day, it is not in terms of a kg, but it is in terms of a few thousand tons. But unfortunately, we are not able to adopt this process in our country. This is now previous was the dump leaching, where millions and million tons of a low grade ores and tailings, now the heap are prepared, and from the heap, now every year you will get the production of the coppers, zinc and lead. For the concentrates, and particularly for the gold concentrate, which is having the arsenopyrites, where due to the presence of arsenopyrite, the cyanide method will not be useful directly, because most of the cyanide is consumed by the iron as well as the copper and zinc. So for that, first of all, you have to go for the biohydrometallurgical leaching. So the arsenopyrites from this, it will be oxidized and gold will be separated, and that gold would be leached out. And this type of the reactor leaching also able to produce the tons per meter cube per day amount of the gold productions. If we compare the amount of copper and gold productions, nearly 27% of the world copper productions by these methods, and nearly 22 to 23% of the gold productions is carried out by these biohydrometallurgical methods. Now, the biology process of this particular field, where in India we are working with now Department of Metallurgy, Bangalore, then CSIR, Bhubaneswar, School of Mining, Dhanbad, NML, Jamshedpur, Agarkar Research Institutes, KREC at present, IISc Bangalore, NML Jamshedpur, and Department of Microbiology, Gujarat University, where we have developed the process for the lab scale, pilot scale, bioreactors, column study, shake flask, for polymetallic ore and concentrates, copper and copper ore, and zinc copper concentrate tailing, refractory gold ore, as well as the electronic waste and the catalyst. I would like to thank the students, those who have worked, because I worked initially for my PhD for 3 years. After 1980, my PhD student, they have worked, and 15 students, they got their PhD on extraction of metal ions, either from the polymetallic concentrate or the polymetallic ore, and extraction of metal ions from the various types of the electronic waste. Thank you very much for giving me this opportunity. And at present, we are having this culture consortia developed with us, which can work with any of this electronic waste, and extraction of metal would be more than 94 to 98%, depending upon the waste. Thank you very much.
Thank you, sir. Thank you, sir, for sharing your knowledge and give us the insight about the solid waste management through biohydrometallurgy. Now the session is open for questioning. If please mention in chat box if any question is there.
Chat box mention करना Sir, how you have adapted this microorganism from the toxicity of these metals?
Yes. So, see, initially we have started with, as I told you, the 1% of the pulp density ore. Then it worked for a few months. Then instead of 1%, we have started with 2%, then 3%, 4%. It took nearly more than 10 or 15 years. Now I can go up to 20, 25% of the pulp as well as the concentrate. Initially we started with the ore, then we have gone with a concentrate.
Okay.
Any other question? Oh, okay, sir. Thank you, sir. I think no more questions are there. Thank you, sir, for your time. So we shall now conclude the session in the virtual room B. I am thankful to all the eminent speakers for their inspirational and informative talks, as well as their time for being here, and all the participants for their listening for patiently. Now there's a small lunch break, and we will meet after 15 minutes at 2:20. Before that, I would like to announce that there are two who are participating in the oral presentation, who are interested in human and animal healthcare presentations and bio ideathon. Please be present in the virtual room A. And while those who are interested in industrial, environmental, and plant biotechnology, please join the virtual room B at 2:20. So please return back on time at 2:20. Thank you. Thank you very much. Thank you all.
A very good afternoon to all dignitaries, guests, and delegates. I, Payal Modi, on behalf of Gujarat Vigyan Sammelan, welcome you all in the third session of today's event of oral presentation in the area of industrial biotechnology, plant biotechnology, and environmental biotechnology. For this session, we have 20 participants for the oral presentation. And in the panel, we have with us Dr. Sonal Sharma, Scientist B, GBRC, and she did her PhD from Nirma University and has published 20 papers in international journals and filed 2 patents. She's having around 5 years of research experience post-PhD. Our second panelist is Dr. Satyamitra Sheikh. She is also Scientist P from GBRC. She did her PhD from Saurashtra University and has published 5 papers in international journal and having around 4 years of teaching and research experience. So for this session, the participants will get 6 minutes for oral presentation and 2 minutes for question and answers. On completion of 5th minute, there will be a warning bell. And after 6 minutes, you'll hear a completion bell. Participants are requested to complete the presentation within stipulated time. So starting from the first theme of the session, that is industrial biotechnology, our first participant is Ms. Armi Chaudhary from Gujarat Biotechnology Research Center. And the title of our presentation is CRISPR-Cas9 mediated knockout of SEG-D gene for overexpression of streptokinase in Streptococcus equimilis. I request Armi to share her presentation and unmute herself.
So, can you see full screen? Hello?
Yes, yes. We can see you and you are audible. You can go ahead.
Okay. So good afternoon, everyone. I am Armi Chaudhary and I am presenting you my research work at GBRC, which is CRISPR-Cas9 assisted knockout of SEG-D gene, which is inhibitor of streptokinase. So overall hypothesis of this project is we have our desired protein streptokinase, which is having a very high therapeutic value. And when we have tried to express this streptokinase in E. coli, we are facing some problem because of its toxicity, because it is itself a kinase. So to overcome this, we have designed one CRISPR-Cas based technology that we found SEG-D gene, this whole cassette, it is a negative regulator of streptokinase. So what is happening over here is when there is a production of streptolysin, it is degrading this mRNA transcript of this streptokinase. So what we have designed is we have just knocked out one of the cassette from this SEG genes, and then we found enhanced expression of streptokinase. To achieve this all experimental work, we have used P-crispomyces2 plasmid system, which is single plasmid system, which is having a Cas9 enzyme and guide RNA both inside it. So this is a graphical presentation where you can see in this figure F, where a 21 nucleotide base pair guide RNA, which is complementary with the SEG-G cassette, and this is how it is binding, and Cas9 is doing endonuclease cut over here. So first we have designed specific guide RNA transcript in this TAFR, where we have submitted a Streptococcus genome, and with the minimum off-target effect, we have screened out some guide RNA, and then we also used the editing template. So what is use of editing template is we have designed a 4 pair of primer, where you can see here SEG-C and SEG-E has a complementary region, which is shown in this pink color. So when we have amplified this SEG-C and SEG-C, and then we have performed overlap extension PCR, where we have only SEG-C and E, and then there will be no SEG-D, which was also confirmed using the sequencing. So we have used Golden Gate assembly and Gibson assembly both. And in Golden Gate, we have used single guide RNA, and for Gibson, we have used this editing template to just what happened in cell when there is an endonuclease cut, cell will start a double-stranded repair. So in that case, it will use something editing template as a repair template to just do a homology directed repair. So in that case, it might use this SEC-E as template, and we might got a complete knockout of SEC-D gene. So this is overall cassette preparation using Golden Gate and Gibson assembly. And this is our screening strategy when we got a clone of Streptococcus. So it was a very rigorous screening because for this P. crispomyces 2 template, we have to let it express for 2 to 3 days. Now in a solution, in a broth, there will be a edited one, red color, and non-edited one, blue color clones. So in the suspension of that, we have to identify whether identify or isolate a particular clone. So it is a very rigorous method to do that. So how we have achieved is, we have first let it express for the high for a longer period of time with a higher concentration of selection antibiotic. And here is a more closer picture of that. Like this is the red color arrow in figure F. It is the desired guide RNA sequence which we want to modify. But what we are getting is mixed peak while we are doing sequencing, because that partial expression of P. crispomyces 2 plasmid, and we are getting a mixed culture of edited and unedited one. Because if you see this green arrow, this sequence is completely matching to the sequence of purple arrow. So this led us to conclude that there is a mixed population. So to overcome this, we have done a prolonged expression of P. crispomyces 2 plasmid for that, and then we have performed the screening of edited clones. So after that addition, we have also performed the computational work where we have found that this, where we have successfully knocked out this. As you can see, there is no this red color region over here in figure A, and we have successfully knocked out the E. coli Streptococcus strain, and then we have modified the protein, this SAC-D complex with 7 amino acid, and there is a huge change between wild type and mutant, which was determined computationally, and we have also determined it using an enzyme assay. As you can see, wild type is having a very high hemolytic activity, while mutant is having a very less hemolytic activity. And for the fibrin plate, which is a fibrin clot, which are present in human when they are suffering from myocardial infarction. So in wild type, our strain was doing this much like 2 mm zone for fibrin degradation, but for mutant, it is having a very large zone of fibrin degradation. Same enzyme activity was also enhanced, which was further determined using real-time PCR. And one problem with this is we didn't have any tag to purify this protein. Like we don't have any histidine tag. So for this, we have just excised out this expressed band from this figure B, and we have performed MALDI for the same, and we confirm it that there is a 100% match with the streptokinase protein. So this also confirmed that there is an enhanced expression of streptokinase protein.
Army, 6 minutes are completed. I request you to complete your presentation in 30 seconds.
Okay, and this is the last where our limitation of this study is that there is no his tag. So we need to develop a strategy to purify this protein. And another future aspect is people are facing hypersensitivity for the same. So to overcome this, we have designed that we can terminate this C-terminal part, which is reacting with the complementary system. So this a truncated streptokinase can be a more therapeutic value protein. So thank you.
Thank you so much, Army, for such a nice presentation. Now we'll have questions from our panelists.
Okay.
The question? I want to know that how your enzyme is superior than the commercially available enzymes.
Yes. So enzyme is same, like the commercially available enzyme are also streptokinase, and this is also streptokinase. So enzyme is same, but it is superior in case of the strategy where it is devoid of streptolysine.
And have you checked any safety effects of these enzymes on plate assay or any other study?
No, we have we have done till this much, but we are planning to do some further study in future.
And what is the yield of the enzyme? Have you quantified?
That I also shown in presentation that this is a crude supernatant based work. To do this same in purified work, we have to develop a purification strategy because this is in a native strain. So we have developed an ammonium sulfate based precipitation method with a fraction purification.
And what is the yield? Can you give details regarding that?
Yeah, it is like 2 mg per 10 ml of the supernatant.
2 mg. Okay. Thank you, Armi.
Yeah.
So our next presenter is Babita Chaudhary. Babita, are you there? Hello. Okay. So moving to the next participant, Keval Chopra.
Yes, I am here.
Okay. So our next participant is Keval Chopra. He is from Department of Dairy Microbiology, Kamdenu University. And his title of presentation is Significance of Yeast Lactic Whey Protein Fermented on ACE Inhibitory, Antioxidative and Anti-Inflammatory Activities and Release of ACE Inhibitory and Antioxidative Peptides. So over to you, Keval.
The screen is full now.
Yes, your screen is visible and you are audible. You can go ahead.
Yeah, thank you, madam. Thank you all. I am presenting my talk on significance of yeast lactic whey protein fermented on ACE inhibitory, antioxidative and anti-inflammatory activities and release of ACE inhibitory and antioxidative peptides. So the peptides are being produced from the whey proteins. You know that whey protein is a very significant proteins in the milk. Milk has two kinds of protein, whey protein and caseins, and 20% of the total protein is the whey proteins present in the milk. So we have taken target to ferment these whey proteins with the help of lactic acid bacteria as well as the yeast. And after fermentation, we want to have the peptide production, and those peptides are having some biofunctional properties like antioxidative and antihypertensives. So for this study, there are three objectives we have formulated. First is to evaluate the ACE inhibitory, anti-inflammatory and antioxidative activities using these two cultures, Lactobacillus paracasi and Saccharomyces cerevisiae. And then the second objective is to optimize the growth condition for the maximum production of the peptides from this whey protein fermented. And third one is to purify and characterize these peptides, these is bioactive peptides, which having these kind of properties, antihypertensive as well as the antioxidative. So these are the methodology we have used for antihypertensive and antioxidative activities. And these are the methodology for SDS page, and then we did the 2D page, then we also did the FTR analysis, and also we have checked the peptide production through HPLC. And also we did work on the cell lines on anti-inflammatory activities. So now I'm coming to the results which we got it from the study. So first is to ACE inhibitory and antioxidative activity, both are being presented here. And we have seen that as the temperature, as the duration, not temperature, duration is increasing, the activity is increasing for all the four activities for this Lactobacillus paracasi. Similarly, for the Saccharomyces cerevisiae, as the temperature time is increasing, means the duration of, or you can say the incubation period is increasing, then the four activities are also increasing. Then we have also optimized the growth condition, rate of inoculum and incubation period for both the cultures, means Lactobacillus as well as the yeast. And we have found that 48 hours and 2.5% rate of inoculum has been given the maximum peptide production, and for Lactobacillus as well as for the yeast cultures. Then we have also fragmented the peptides by using 3 kDa and 10 kDa ultrafiltration membrane, and we have got very good results in the retented 3 kDa retented as well as the 10 kDa retented for these four different activities, antihypertensive as well as the antioxidative activities. And then we have done the SDS space to see the hydrolysis pattern, and we found that this is the normal whey proteins, and this is the fermented whey protein, and then we pass through the different ultrafiltration membrane, 3 kDa retentate, this is 10 kDa retentate, and these are the 3 kDa permeate, and this is 10 kDa permeate. So in this permeate portion, you can see there is no bands, means the complete hydrolysis has been taken place in the permeate portion, but retentate, the hydrolysis are there, we're getting the bands compared to the control. And after seeing the protein profile through SDS page, we did the 2D, and after doing 2D, we got different spots, and these spots were trypsin digested, and then we analyzed the peptide sequences through the LC-MSMS analysis. And we have also checked the peptide fragmentation in the RP-HPLC analysis. So compared to the control, this is the control unfermented one, there is a very smaller fragment of peptides are produced, but it is a fermented one where you can see that a lot of fragments are produced. This is 3 kDa retentate and 10 kDa retentate. The peptides are producing in a higher quantity, one with a various fragmentation conditions. So peptides patterns are being observed in these different HPLC chromatograms. We have also analyzed in the FTIR to see the means fermented and unfermented one. So this blue line, this is the unfermented, and this is the fermented, the yellow line. And you can see the patterns where you can say that the peptides are produced compared to the very, means the bands are quite different from the control. And we have also checked the molecular docking of these peptides. And we only 2 peptides, this AFLD SRTR and ILGA FIQ IITFR, both the peptides are being used to see that how these peptides are being liganded with the protein molecule, mainly the human myeloperoxidase enzymes. Then we have checked the anti-inflammatory activities on the raw macrophages cell line, 264.7. First, we have seen the cell viability as the concentration of the whey protein fermented increased, but there is no changes in the viability. Even nitric oxide production compared to the LPS, it is less. For TNF alpha production, again, you can see compared to the LPS, the fermented plus the LPS, it is also reduced. Even in IL-6 and IL-1 beta, both the cases, the whey fragmented or whey protein fermented product is giving better result than the LPS alone in the raw macrophages cell lines. So we have got these 2 peptides, FLD SRTR and ILGA FIQ IITFR. Both the peptides are being obtained from the LCMS from the 2D pages. We have shown you that we got some spots in the 2D pages after doing the trypsin digestions. We have analyzed on the LCMS, and these 2 peptides are being obtained, and they are non-toxic because we have using some softwares where they showed the toxicity and also give the peptide anchor score, and they showed that source is lactoperoxidase, and their molecular weight around 963.43 and 139.7. Both the peptides gives the
Keval, your time is up. Can you please conclude your session
in
30 seconds?
Just I'm concluding, madam. It is just last slide. So in the conclusion, these 2 cultures, Lactobacillus paracasi as well as the Saccharomyces cerevisiae, both are very potent culture, and they have given the ACE inhibitory activity, antioxidative activities. And these 2 cultures can be used for the production of the peptides from the whey protein or whey protein fermented. And now the key message is these 2 cultures in a combination mode can be a good applicant for consistent value addition for whey protein fermented based products, which having these ACE inhibitory and antioxidative properties. Thank you all for listening me.
Thank you so much, Keval, for your presentation. Now I request our panelists to ask the questions.
I have a basic question, like what method you used to identify ACE in it? You have doing some in vitro assays or cellular assays?
No, madam. No, madam. We use the very well established method, in vitro method.
Yes,
for ACE inhibitor, I am telling you, we have taken the substrate. This is called HHL, histidine hypuryl lysines. And we have taken the ACE enzyme from the rabbit lungs, and we did the reactions. And from this substrate and enzyme reactions, hippuric acid is generated. Okay, but our target is to reduce the production of the hippuric acid. The peptides, they have the ability to restrict or prevent these reactions, because this ACE enzyme angiotensin converting enzymes, they are basically convert AC1 to S2, and then when S2 is produced, then your hypertension will increase. So this in vitro method we have used to check this AC inhibitory activity.
Okay, thank you.
Thank you.
Hello.
Hello.
I just want to know that you have studied crude proteins for your activities, right?
For which kind of activity, madam, you are telling?
For both the activities, antioxidative.
Crude protein means we have separated through the ultrafiltration membrane in different cutoff, 3 kDa and 10 kDa. Then we have also analyzed these all the activities.
But it's
and whey protein itself, it's madam, we have taken the we have taken the 80% whey protein concentrate. Then we made a 5% solutions. Then we have added this bacteria for fermentation. So it's a pure protein, you can say. Only 20% is other materials.
And apart from this, there should be some safety related issues for this proteins, right? Because the application will be in vivo for antioxidative as well as this ACE inhibitory purpose. So have you done any this kind of tests?
No, madam, this no, safety means you see that we have used the whey proteins. Okay, whey proteins are basically food protein. It is milk protein, right? But you can say that how much you'll add in the food and what would be the dose for having this activity, that we did not check. But whey protein itself, people use it who is doing exercise, they take the whey powder. Even those who are, I can say, the pregnant mothers, they used to prescribe to have a whey powder because this is very good proteins, and this protein has a lot of immunomodulatory proteins. Okay, so this protein doesn't have any kind of side effect. But if you say that your protein or your peptides is having this kind of activities and how much is required to have that, we did not check in the animal model. Yeah.
But you have to do the safety assessment for the culture as well.
Yes, yes. Culture has, madam, culture we have already done, madam, for lactobacillus and yeast. It is our own culture.
I know they are having the grass status, and apart
no,
from
no,
this,
we have checked, madam, in vitro test, just like I am telling you. This is the beta hemolysis, antibiotic resistance pattern, then some of the genes which are having a virulency, that is being checked, the DNA assay,
I
urea.
just want to know the third thing, that is the proteins if you consume that will be digested in the gastric pH, right, which is from ranging from 2 to 4. So the digested protein will be having the activity. That is the main issue, because after the digestion, protein loses the activity. So that will be of more concern, because when you consume anything, that will not reach to the intestine in its normal format, right? It will get digested. So after digestion of the protein, the activity will be retained. Or
Yes. Madam, actually, you are asking about the in vitro digestion or in vivo digestion. I'm just yes, in vivo digestion. We did not do the in vivo digestions. Pardon?
After oral consumption, the proteins will be get digested, and for the
yes, but
oxidative as well as ACE inhibitory effect, one needs to consume this peptides, right?
Yeah.
So, so digestion, digested proteins.
Digestion we have not done, madam. We have not done digestion. So what you are, but the thing is, some of the peptides, they are highly resistant to the proteolytic enzymes like pepsin, trypsin, which are present in the stomach as well as in the intestine. Okay, so that because some of the because we are already working in peptides in different food products. Last 10 years we are working on peptides, and still we are working. So in vitro digestion we are doing, but we don't have the facility to go for in vivo, which you are asking. So in vivo we did not do, but we are seeing, we are checking the resistant pattern of different proteolytic enzymes against these peptides. Some of the peptides which are very smaller in size, they are resisting because of these some amino acids.
Thank you so much, Mr. Keval.
Thank you.
Moving to our next presenter, we have Tahreem Fatima. She is from Department of Biotechnology, Institute of Engineering and Technology, Lucknow. And her title of presentation is Plant-Based Bioreactor Design, a simple and efficient model for secondary metabolite production. Fatima, can you please share your screen?
Yes, ma'am. Ma'am, is it visible?
Yes, it is visible.
Okay. Ma'am, can I get 2 minutes if there's something wrong?
Okay. What is the problem, Fatima?
Ma'am, I was unable to. Ma'am, it's done. Good afternoon, everyone. Ma'am, is it visible?
Yes, it is visible and you're audible. Go ahead.
Okay. Good afternoon, everyone. The topic for my today's presentation is plant-based bioreactor design, a simple and efficient model for secondary metabolite production. As we all know, the bioreactors are the vessel that helps to convert the substrate into a product through a series of various biochemical reactions. There are various type of bioreactors like stir tank, membrane, fluidized bed, etc. Here in the figure one, you can see type of bioreactors are that it requires skilled people to operate such type of bioreactors, and there we see difficulty in cleaning, and they are heavy and bulky too, and they do require a large spacing area. So the objective for my work was to construct a plant-based bioreactor using aloe vera leaf, and to study the effect of this aloe vera plant-based bioreactor as batch state fermentation process. As here in figure 2, you can see the bioreactor that I have constructed for the batch state fermentation process. Moving on to the methodology, hollow aloe vera leaf was used to construct the plant-based packed bed bioreactor. And in this, the amylase producing bacteria were immobilized using sodium alginate. Here in the figure, you can see the materials that have been used. The figure 3, it represents the MSM, the starch nutrient broth, and aloe vera gel. Whereas figure 4 represents the sodium alginate, which helps to immobilize the amylase producing microbe. The aloe vera gel was used as a nutrient media for fermentation, and no part of aloe vera plant was wasted during the process. This type of bioreactor serves as cost effective and biodegradable. It gives easy handling and will be beneficial for the production of various secondary metabolite. Here I've constructed two, I have made a comparative study of MSM. In one bioreactor, I have used MSM, that is a minimum salt media, as a nutrient source, whereas in second bioreactor, I have used aloe vera gel. Here figure 5 represents the steps that are involved for the construction of the aloe vera gel bioreactor, aloe vera leaf bioreactor. The first, as you can see in the figure, we have excised a leaf, aloe vera leaf from a healthy plant, and we have hollowed it and saved the aloe vera gel for later use. In second step, what we did was we sealed the base of the aloe vera using agar gel. Next, we in our bioreactor, we have filled it with the sodium alginate beads and MSM and nutrient broth. Figure 4 here, as you can see, it is a plant-based packed bed bioreactor in which the top and bottom are sealed. And figure 5 represent the picture of the bioreactor after 4 days. So moving on to the results, the bacteria was isolated from the soil. So to make a confirmatory test whether it is amylase producing bacteria or not, we did iodine test. So here, as you can see in the figure, we obtained a clear zone. which indicates that the starch has been hydrolyzed. Next is the immobilization of amylase producing bacteria in sodium alginate beads. So here in the figure 7, we can see the beads were prepared with the help of the syringe, 10 ml syringe, and approximately 9 beads were produced by every 1 ml of the solution. Next, we did the qualitative analysis of starch hydrolysis. In this, what we did was the bioreactor that we constructed, the one in which we had MSM as a nutrient source, and in the second bioreactor, we have aloe vera gel as a nutrient source. So here in the tube A, we can see it's the complete iodine solution. And the tube B, we have the product of the leaf bioreactor in which MSM was used as a nutrient source. And here we can see the color is slightly darker than tube C. We did iodine test. So here what happens is iodine, it make a dark blue complex with starch. So darker the color, more is the starch. So here we can see in the tube C, there is a very light or pale yellow color, which indicates that the starch has been hydrolyzed to a greater extent as compared to the one in which MSM was used, that is the tube B. Next, we did the qualitative analysis of the starch hydrolysis by UV visual spectrophotometer. In this, we checked the absorbance at to 620 nanometers. So here we take a took a control tube in which the absorbance was 0.109, and for the bioreactor in which aloe vera gel was used as a nutrient source, the absorbance was 0.184, whereas in MSM it was 0.57. So here what happened is the absorbance, it is inversely proportional to the concentration of the glucose. So the lower the absorbance, more is the glucose. So when starch is hydrolyzed, it is converted into, it break down into glucose. So we see here in case of aloe vera gel, the starch hydrolysis is greater as compared to MSM. So the conclusion for my whole presentation is this, is that the aloe vera based bioreactor has served as an efficient bioreactor for amylase production as well as starch hydrolysis. Hence, it can replace the heavy bioreactors, and it will give a simplified handling. And we have converted a non-vital plant part, that is the outer layer covering of the aloe vera, into a useful reaction vessel. And this is can serve as eco-friendly bioreactor, which can be easily biodegradable. And it is obviously very cost effective, and it is present in abundance in our surrounding everywhere. So when we made the comparative study of MSM and aloe vera gel as a nutrient source, we got the better result in case of aloe vera gel. So we can say that we can replace MSM by aloe vera gel. So we are using the least chemicals and getting good results. Thank you. This was my presentation.
Thank you for Fatima for this presentation. Now we'll have questions from our panelists.
Yes, ma'am.
Commercialized. Hello, can you hear me?
Hello. Yes, ma'am. Ma'am, please pardon.
Yes. Actually, I want to ask about the commercial aspect of your plant-based bioreactor. So you have mentioned that your bioreactor or your idea of a plant-based bioreactor is very efficient and cost-effective. So I want to just ask, can you justify this statement through some case study or any example of any commercial available plant-based bioreactor? Is there?
Ma'am, up till now, there is no plant-based bioreactor, and the idea behind construction of such type of bioreactor was that on a small scale, where we are not confirmed whether our experiment are going to give results or not, we can perform it. We can perform it without using any bioreactors. Means that if someone is not having any sort of bioreactors available, so we can perform it into a plant leaf. So this is quite cost effective.
Okay, but what is the production
We
efficiency?
don't need to. Ma'am, production efficiency, ma'am, I've not done that quantitative analysis up till now.
Okay.
So, yes. Hello.
I want to know that what other
Ma'am, your voice is breaking.
for this kind of plant-based bioreactor, because all cultures will not be able to grow in this type of setup, right?
Ma'am, your voice is breaking.
Hello.
Hello.
Are you getting my point?
Hello. Yes, ma'am. Ma'am, please pardon.
I want to know that the organisms. There are many organisms like aerobic, microaerophilic, anaerobic. So this will be applicable to particularly on these a type of culture. So can you enlighten regarding this?
Ma'am, in this bioreactor, we can perform aerobic as well as anaerobic fermentations. Uh, we have used agar gel to seal the top and bottom. So if we are using lesser concentration of agar at the top to seal the bioreactor, we will get enough of oxygen supply. So this will support both aerobic as well as anaerobic. As per our need, we can control the concentration of the gel that we are using for the sealing of the bioreactor.
But what will be there for the aeration as well as other setups which are available in the conventional bioreactors? Because here in this case, there will be no aeration, no agitation. So
yes,
how
ma'am.
can you maintain those conditions?
Ma'am, this is simply the packed bed bioreactor that I've constructed. And for the stirred thing that and agitation, all that, ma'am, I have to look that. I'll continue it to do in future.
Okay.
Yes, ma'am.
Thank you so much, Fatima.
Thank you, ma'am.
Moving to our next presenter, Amita Mishra. Amita, are you there? I think Amita has not joined yet. So we are moving with the next participant, Darshita K. Pandya. Darshita, are you there? Darshita? Hello, Darshita? Can you please reply in chat, or you can unmute yourself and give response? Okay, moving to an Darshita, you are not audible. You can share your screen. So our next presenter is Darshita K. Pandya. She is from Applied Phycology and Biotechnology Division, CSIR, CSM, CRI. And the title of her presentation is In Vitro Callus Culture and Tissue Regeneration of Kappa Ficus alvazrezi, a red algae. Darshita, you're not audible. There is a problem with your microphone. So you can solve this problem while we can move to next participant, and we'll get back to you again. Okay. So our next participant is Savan Donga. Savan, are you there? Hello, Savan. Okay. Nirmala K. Gupta. Nirmala, are you there?
Yes, ma'am, I'm there. I'm here.
Okay. So moving to our next theme, that is plant biotechnology. Our first participant is Nirmala K. Gupta. She is from CSM CRI Bhavnagar, and the title of her presentation is Cloning and Characterization of Pathogenesis Related Gene from Groundnut. And I request other participants to please remain present and check with your microphone whether they are audible or not. Okay. Thank you. Over to you, Nirmala.
Ma'am, am I audible?
Yes, you are audible and your screen is also visible.
Okay, ma'am. So a very good afternoon to one and all present here. Today's my presentation topic is cloning and characterization of pathogenesis related gene from groundnut. So why we choose groundnut? Actually, groundnut is one of the major oilseed leguminous crop, and it is rich source of oil, carbohydrates, and its oil is rich source of vitamins, minerals, antioxidants, and all the macronutrients like calcium, magnesium, potassium, as well as all the micronutrients like zinc, iron, phosphorus. So all of these values make groundnut as a commercial cash crop. So efforts are being made to increase its production and area under cultivation. So what are the problem we face when we cultivate groundnut and when we harvest this our crop plant groundnut? So according to ICMR, 21% of groundnut in India is unfit for human consumption due to aflatoxin contamination. And another study conducted by AgriSet reveals that the level of aflatoxin in Indian groundnut is 40 times more than its permissible limit. Some of the fungus like Aspergillus flavus and Aspergillus parasiticus produce a carcinogenic mycotoxin that is called aflatoxin, and it is considered to be a threat to human health, global food safety, and this fungal infection negatively affects survival of plant biomass production and crop yield. So these PR proteins gene contribute to plant defense and innate immune system, and these PR proteins are widely used as molecular markers of defense signaling pathway. So this is the crosstalk between our crop plant Arachis hypoxia and this fungus Aspergillus flavus. So when this fungus enters our crop plant, so some of the PAMs, RLKs, and transporters provide and facilitate this entry in our crop plant. And after when pathogen enters in our crop plant, some of the oxidative reactive oxygen species, lipid peroxidation occur, and a defense pathways move on. And in this defense pathway, some of the transcription factors and these PR proteins activate, and these are ultimately helps in our plant defense system. So what I did, I isolate PR10 gene and design the primer according to at NCBI, and then I cloned this PR10, and then I cloned this PR10 in PGMT, then I cloned it in PRT101, and then ultimately I transformed it in Picambia 1301, and then transformed it in agrobacterium. So this first GI image is PCR confirmation of PR10 gene, then cloned 18 PGMT vector, and this is the restriction digestion confirmation of PGMT with EcoR1 and ISMA1, then cloned 18 PRT101, and this gel image is restriction digestion confirmation of this gene PR10. Then I transformed it in Picambia, and these two gel images, restriction digestion confirmation of our PR10 gene, and finally transformed it in agrobacterium. So what is the conclusion? After making gene construction with 35S promoter and agrobacterium transformation, a detailed study is going under process on how overexpression of PR10 will resist this fungal infection. Thank you.
Thank you so much, Nirmala, for your presentation. We'll have questions from our panelists.
Yes, ma'am.
Presentation, Nirmala. Just I want to ask, you have mentioned you have isolate gene. How did you do that? PR10. How did you isolate the gene PR10?
Ma'am,
What method?
First I make cDNA, and I designed the primer, and on the basis of that I PCR and I did PCR, and I got the band at 453 base pair.
Okay, so do you confirm the PCR bands?
Yes, ma'am.
By sequencing and other thing?
Yes, ma'am.
Sanger sequencing. You just
Yes, ma'am. I did sequencing also. Yes, ma'am. Sequencing is done.
Okay, to confirm the whether the isolation is right or not.
What, ma'am?
You have amplify your you have convert your RNA into cDNA, right?
Yes, ma'am.
Convert the with the help of primers. You PCR based cloning. You
With the
do
help of gene specific primer, ma'am.
Okay, you have designed gene specific primer.
Yes,
and
ma'am.
then the particular gene, and then it clone in the particular
PZMT. Yes, ma'am. PZMT and then PRT, and then ultimately transformed it in Picambia.
So yes. So just I want to ask you, have you confirmed the PCR amplification whether the isolated is your gene or not?
Yes, ma'am. It is confirmed by PCR.
Not PCR with Sanger sequencing.
Yes, ma'am. It is confirmed by sequencing also.
Okay. Okay. Thank you.
What is the source of this PR10 gene and what is the function of that?
Ma'am, its source is our cropland groundnut.
But what it codes for? What is the protein and what its activity?
Actually, ma'am, PR10 protein is very helpful in defense pathway, and it is confirmed by some of the research paper by microarray analysis.
But the gene has been originated from where? PR10 gene. From
Groundnut, ma'am. Ma'am, it is from originated from groundnut, and I'm over expressing is also in our cropland groundnut.
Okay, and what is the mechanism of this gene function? How does it control against fungal infection?
Ma'am, actually, it has a conserved size and secondary structure organization with a fold consisting of 3 alpha helices and 7 anti-parallel beta strand, which makes a large hydrophobic cavity that plays important function in stress tolerance.
Okay.
Thank you so much, Nirmala.
Thank you, ma'am.
Darshita, are you ready?
Ma'am, am I audible?
Yes, you are audible. So our next presenter is Darshita K. Pandya, and she is presenting under the theme of Industrial Biotechnology. She is from Applied Phycology and Biotechnology Division, CSIR, CSM CRI. And the title of her presentation is In Vitro Callus Culture and Tissue Regeneration of Kappa Ficus alvarezi, a red algae. Over to you, Darshita.
Okay, ma'am. Thank you so much for the introduction, and I'm really sorry for the inconvenience caused. So I'll be presenting on in vitro callus culture and tissue regeneration of Kappa Ficus alvarezi. Now, Kappa ficus is a red algae, and it is a very important source of carrageenan. It is used for the commercial production of carrageenan. But why are we even taking up the topic? It comes under the blue revolution, which aims to promote the aquaculture So these algae are also a part of aquaculture, and that is why we have taken up the study. Now, why are we actually going for tissue culture? Tissue culture helps us to study basic mechanisms of the algae. Not only that, it also helps us to bring genetic improvement in our explants. And after bringing genetic improvements, it can help us to propagate those improved varieties in mass. And thereafter, we can take them in the field and achieve more production. So for the tissue culture, the three major steps which were used are surface sterilization, tissue culture initiation, and last was subculturing. First of all, the samples were collected and they were kept for lab acclimatizations. Once the sample were completely acclimatized with the lab environment, the explants were incubated in sterile seawater for 2 days to bring out any other contamination that are already present in the explant. Further, after the incubation period, they were sterilized with liquid detergent, continuously rinsed with seawater to remove the detergent. Thereafter, with povidon's iodine, and lastly, again, it was rinsed with seawater. Further, the last step of sterilization involved the incubation of explants in antibiotic solution for 3 days. After the incubation of those explants, before inoculating them on the medium, they were again rinsed with seawater, and the inoculation was on pest media, that is Provesolis enriched seawater medium, containing two different PGRs, that is benzylaminopurin and naphthalene acetic acid. The medium was a solid medium containing 0.8% agar. After the inoculation of explants on the plates were kept at 24 degrees Celsius in the culture room, and the photo period of 12 hours was continuously maintained. To maintain the explants, subculturing was done on every 20 days, and the explants were transferred on fresh medium, which contained the PGRs as well as the agar concentration in a decreasing order. So coming to the results, in the first picture here, you can see that after inoculation of the explant on the medium, after the first subculture, we could see clumps of cytoplasm forming, which indicated that the callus induction had already begun. And further, you can see in the second picture, the callus has started forming. After the second subculture, the medium was altered, the agar percentage was continuously decreased, and here you can see that the callus growth is increasing on every subculture. In the fifth picture, you can see that the callus filaments have started elongating, which tells us that
You are not audible, Darshita. Darshita? Hello, Darshita? I think we lost connection. Darshita,
Hello?
are you there?
Hello?
Yes, Darshita, you can continue.
Yes, after the elongation of callus was properly observed, the explants were brought on a plate with the same medium constituents, but 0.3% agar, and slowly clumps of calluses were found on the explants. Here in the second picture, you can see that the elongated fibers have started maturation, which indicates that the possibility of microglobule formation from the callus is 100%. Further, after the maturation of callus, small clumps of callus were formed, and from that you can see in the last picture here that small micropropagules have been generated. Now this work is already under progress, and further these explants were transformed into liquid culture for growth of tissue culture regenerates. There were certain problems that we faced in the process. The first problem was tissue necrosis and bleaching of explant. Here you can see in the picture that some of the explants are completely bleached and have completely lost its activity. And the other problem that we have faced is direct regeneration. So what happens is the buds have started forming without the induction of callus. So these are some of the problems that we have faced in the work, and here I would like to conclude the presentation. So thank you.
Thank you so much, Darshita, for this presentation. Now we'll have the question answer.
Darshita, can you hear me?
Yes, ma'am.
One basic question: where did you get the explant?
Ma'am, the explants were brought from a commercial cultivation site near Okha.
Okay, so it is confirmed the explant you are using is the species you said.
Yes, ma'am.
So there's the second is you mentioned some difficulties during your work.
Yes, ma'am.
Right. Okay. So what are the methods you can use to overcome this the particular problem during your work? Have you any plan?
Ma'am, the problem of bleaching cannot be overcome completely, because there are possibilities that even from the same explant, two pieces are cut, one might get bleached and the other might survive. So that is completely selective. But after second subculture, the explants that reach the second subculture, there is 80% probability that it will at least reach on the stage of callus proliferation.
Okay.
Thank you so much, Darshita.
Thank you, ma'am. I'm sorry for the inconvenience, ma'am.
It's okay. Our next presenter is Dinesh Kumar. Dinesh, are you there? Okay. Moving on to next presenter, Poonam Patel. Poonam, are you there?
Hello.
Hello. Yes.
Yes, ma'am.
Okay. So our next presenter is Poonam Patel, and she is presenting under the theme of plant biotechnology. She is from Gujarat Biotechnology Research Center, and the title of her presentation is Genetic Variability, Physiological Characteristics, Pathogenicity, and Molecular Diversity of Fusarium oxysporum F species, Cumini infecting Cumin cyminum L in India. Can you make presentation more?
Ma'am, actually, I cannot download it, right? Just a second. Ma'am, is it visible?
But it is not in presentation mode.
Okay.
Poonam, if it will take more time, then we can move on to next presentation.
Yeah, please, can you move it?
Okay,
I'm
we'll
trying,
come back to you
okay?
later.
Yes,
Okay,
ma'am,
we'll
sure.
come back to you later.
Okay.
Okay.
Yeah, thank you, ma'am.
Okay. Akshat Kumar Joshi. Akshat, are you there?
Yes, ma'am. Am I audible?
Yes, you are audible. So moving to our next presenter, we have Akshat Kumar Joshi from Department of Genetics and Plant Breeding, Sardar SD Agriculture University. And the title of his presentation is Genetic Variability Analysis in Sugarcane through In Vitro Chemical Mutagenesis on Callus Culture. Your presentation is visible and you are also audible. You can go ahead, Akshat.
Good afternoon to respected sir and all dear spectators. Today's my presentation topic is genetic variability analysis in sugarcane through in vitro mutagenesis on callus culture. So, this is my master research work, and I had that time one promising sugarcane genotype, that is CON 13073, which is subjected for new protocol of callus and regeneration development standardization, and its callus were subjected for induction of mutagenesis through different treatment of mutagenic agents. So objective of my study is to create somaclonal variation through mutation and selection of that variant with physiological parameters of 25 days of plantlets. And these all the treatments I gave during the callus culture in factorial CRD design. So there are 6 factors of different mutagenic agents and 2 factors of different treatments for submerging the callus into the mutagen. So I recorded these all observations. So survival percent, regeneration percent, number of days for shoot formation, multiple shoots, root shoot lengths, biomass, leaf area, chlorophyll, and multiplication rate of the callus. So I found the results in which the survival percent found highest in 81.7% in the 2% ethyl methyl sulfonate with 60% of submergence. Here we can see that survival percentage of the callus with 2% of EMS and 60 minutes submergence in that mutagenic agent. Now the regeneration percent of the callus. So highest regeneration percent found in treatment of 30 minute submergence in the sodium methyl methane sulfonate 0.3%. So here 0.3% methyl methyl sulfonate giving the highest regeneration percentage of the callus. Regeneration percent, here we can see a highest shoot formation found in this treatment. Now here
We cannot see your screen.
We can see here multiple shoots, highest number of multiple shoots 15.2 per callus found in the treatment of ethyl methyl sulfonate 2% with 60% expression of 60 minute for submerged condition in this mutagen treatment. So here we can see the multiple shoots from regenerated callus. With ethyl methane sulfonate having 30 minutes of treatment. Now we have the shoot length. Highest shoot length found in same treatment, ethyl methane sulfonate 2%, 30 minutes of the submerged condition, and here we can see the shoot length after 25 days of hardening. This shoot length be found of 26.6 centimeters. Now this is the root length, which is 12.8 centimeter, found in the treatment of methyl methane sulfonate 3.3% with 30 minutes of submergence. Here we can see that root length 0.2 ethyl methane
Akshat, please
sulfonate 30
conclude
minutes.
your presentation in 30 seconds.
Yes, sir. Now this is the multiplication rate. So this is the multiplication rate of 44.7% in methyl methane sulfonate with 30 minutes of submergence. And now this is the chlorophyll content. So this fraction of chlorophyll content, this is the chlorophyll content not in index, found in EMS 2% with 30 minutes of submergence. This multiplication rate 12.45 means 12.45 multiple shoots we generated from the callus with this treatment, same treatment. So we can conclude that the effective mutagenic agent was found to be sufficient to create variability at lower concentration for the lower time interval for most of the character under studies. In present study, ethyl methane sulfonate 2% and 0.3% found to be more effective, and effectiveness decreased with the increase in time interval. Thank you.
Thank you so much, Akshat, for this presentation. Now I request our panelists to ask the question.
So your major objective was to produce the callus culture.
Major objective was first of all, I was working on new promising genotype, which is sugarcane 13073. So callus regeneration and callus culture and callus regeneration protocol was not standardized at that time. So I standardized first of all that protocols, and then I gave mutagenic treatment on callus condition. These mutagenic treatments.
What you get from mutagenic condition? What you want to do to use mutagen? What is your should be
first
your
of all, my main objective was sugarcane. Basically, it is clonally propagated crop, and natural reproduction system in sugarcane is too much difficult to create a new variability in sugarcane because it's having a clonally propagation technique. So fixation of heterosis is not question in sugarcane. So any new variability create that is already fixed one due to mitosis. Meiosis is not going to happen in sugarcane during the sugarcane cultivation. So my objective was to create a new variability through mutagenesis.
In variability in terms of what? At genetic level or protein content?
At genetic level variability. Mutagenic mutation, we can say it is sudden inheritable change in genotype of individual.
Okay, thank you.
Thank you so much, Akshat. Moving to our next presenter, Dinesh, are you there? Dinesh? Poonam, are you ready?
Hello, yes, ma'am. I'm there.
Okay, Dinesh. So our next presenter is Dinesh Kumar from Gujarat Biotechnology Research Center, and the title of his presentation is Understanding Rhizomicrobiome Interactions, Microbial Community Structure Analysis Associated with Barleria prionitis. Over to you, Dinesh. Please share your screen.
Yes, can you?
Yes, your screen is visible.
So can you hear, ma'am?
Yes, we can see your screen and we can, you are audible. You can go ahead.
And my screen is visible?
Yes, yes, you can go
Okay.
ahead.
So for today's presentation, my talk is on the understanding rhizomicrobiome interaction, microbial community structure analysis associated with the Barlenia praetinis. Barlenia is known as the Vajradanti in Hindi. Most commonly, it is a medicinal plant utilized for the toothache. That is the most common use of this herbal medicinal plant. Apart from that, it is also a widely known as weed kind of plant, which can be seen growing everywhere in and around our neighborhood, especially in you can see in the dry region also. It can also be seen in the so to understand the microorganisms in the rhizosphere of this particular plant, I have tried to analyze the data related to this particular plant. So as I said, that soil is a very rich source of microorganisms, microbial population, and some basic definitions. What is rhizoplane? What is and endophytes are there? Some rhizosphere that is below the ground, phyllosphere that is above the ground. So this exploration studies below the ground, that is in the soil, associated with the roots. So that makes it a really interesting topic, because the microorganisms associated in the soil and the root associated microorganism, that is rhizomicrobiome, that deliberately and it influence the plant growth and productivity. It also influence, there are papers that can be literature can be reviewed on this, but apart from that, it also affect the plant disease resistance capacity. But not all kind of microorganisms can be seen in this particular zone, and this zone is very critical for the plant growth, development, disease resistance, and there are other several biotic abiotic factors. So further to move on further, the rhizomicrobiome that deliberately and intentionally, it is a mutual relationship where we can see the symbiosis is present in this zone, and both plant and microorganisms benefit from for each of their activity in terms of nutrient absorption. And so that is, one is healthy, that is good one, other one is a bad microbiome that cause the disease. So what makes these interactions fruitful, and how we can improvise these interactions to benefit in agricultural practices, where agriculture can be improved or sustainable agricultural practices can be improved. So that kind of exploration has been carried out in this study. It is preliminary work. So and this graphical abstract indicates that this plant's soil from this root part has been taken, and based on the 16S sequencing, this characterization of the diversity has been done in this paper. So what were the findings? So the findings indicate that the dominant microbial community at the phylum level were represented by Proteobacteria, Actinobacteria, Firmicutes, and Actinobacteria, and similarly at the family level, the Bacillus, Pseudomonas, and Streptospongiosis, Pseudomonaceae. So these were the families that were dominant and that were found to be dominant in the soil rhizosphere associated with this particular plant. And this study is of natural environment. From the natural environment, these samples were sequenced. So the dominant, and these are the some photographs highlighting the features of this particular plant. It is spiny spines are there on this plant, you can see. So these are kind of the adaptive features, desert adaptive features or semi-arid zone. This kind of plants can be seen growing in and around. So as far as the method and methodology is concerned, the DNA was isolated from the soil using the Kaizen kit, and it was quality checked, visualized on agarose electrophoresis, and further the amplification was carried out using the primer sets. Now the PCR conditions are there. So these are the methods that has been carried out to perform to this in this experiment. And after that, sequencing was carried out on end-to-end sequencing platform. So summary of the results, we can see some of the data that has been obtained from this work. So the first 6 samples are that they are representing this particular data associated with this particular plant. So where we can see the good amount of reads that were obtained in this study with the mean read length of around 350 base pair. Now, the bioinformatics analysis that was carried out to understand the community structure analysis, the data was filtered and that it was processed in the MGRAST online server with the best fit criteria with the taxonomical analysis with the Silva SSU database. And core microbiome and co-occurrence network analysis were performed using the microbiome analyst, the phylum and species level of taxonomic abundance in the pre and post seasonal soil samples was characterized. Now the statistical analysis was also performed with to identify the diversity indices, species richness, and estimation of the parametric, non-parametric indices were carried out using past PCA and other stamp was used to perform the NMDS analysis. So these are the 2 graphs where we can see in that one phylum level and other one is at the class level. So where in the phylum level, diversity proteobacteria, as I said that in the some of
Please
these
conclude your
samples,
session in 30 seconds, Dinesh.
okay, ma'am. So in planktomycetes, bacteroids, and some of these dominant phyla are represented in this particular slide. So these are the diversity indices where alpha diversity, beta diversity, and the dendrogram clustering was performed. Heat tree analysis indicated that the closest statistically significant closest dominant phyla that it is at the class level. So class dominant class were represented in this particular heat map where we can see the dominant in the yellow color. And in this core microbiome co-occurrence network analysis was also performed with the help of microbiome analyst, and where we can see Burkhalderio, Pseudomonaldi at the order level, and the Bacilli were dominant in particular rhizobials. So these are 6 different orders that are represented here in this on the right side panel. So this data is now publicly available. It can be accessed. And this is so to summarize in a graphical summary, I can say in this particular slide is summarizing these findings. So where I have sampled and then performed the DNA isolation, then performed the sequencing, analyzed the data, and some of these findings should help in identifying the better plant growth promoting rhizobia, PGPR microorganisms can be identified, but it requires further studies to characterize those microorganisms. So this is my conclusive slides. Thanks a lot. Thank you so much.
Thank you so much, Dinesh, for this wonderful presentation. Now we'll have questions from our panelists.
Okay.
Dinesh, which type of further studies are required for characterization? Can you please?
Other type of study means I mean to say that where we can individual bacterial species can be isolated, and then those as an inoculum, those bacterial isolates can be tried on the growth promoting properties and their biochemical characterization of those species that need to be done, because it is a sequencing, so that it is metagenome sequencing. It was performed in this study. So in metagenome. Metagenome will give the entire pool of genomic content from this particular soil samples or the particular environmental site.
So you have collected samples from different places or a single place?
From 3 different places. These were from Gandhinagar sites only.
Okay. So did you find any difference between the microorganism on these places?
There was difference, but that differences were not statistically significant to compare between these sites.
Okay. Okay. Thank you.
Okay.
Thank you so much, Dinesh. Poonam, are you there? Poonam?
Yes, ma'am. Hello.
Yes, yes, you are
Ma'am, the problem is that my phone is not having the PowerPoint mode presentation. So I have to present in that form only, or I'm not able to present then. Hello.
Can you present someone else?
Sorry. Sorry.
From someone else sometime. Poonam?
Yes, ma'am.
Can you present after some time from someone
Yes, ma'am.
else's laptop?
Yeah, no problem, ma'am. But the problem is that I have to present in that form only because my mobile is not allowing in presentation mode. I have tried, but my
That's
iOS
what
was...
I'm saying. You don't present from your mobile, you present from someone else's laptop. Take your time.
Okay. Yeah.
Call you after some time. Okay.
Yeah, sure. Okay. Thank you.
So our next presenter is Maitri Trivedi. Maitri, are you there?
Yes, ma'am.
Yes, we can
Am
see
I
you.
audible?
Yes, you are audible. You can share your screen. So
Okay.
our next presenter is Maitri Trivedi from Gujarat Biotechnology Research Center. And the title of our presentation is Role of Plant Proteases in Plant Development.
Yes, good afternoon, everyone. I am Maitri. Right now, I am working.
Your presentation is visible. You can.
Am I audible, ma'am?
Yes, yes, you are audible and your presentation is
screen is visible?
Yes, yes, you can go ahead.
Okay. So I am Maitri Trivedi. Currently, I am working at JBRC. Previously, I was working in MS University, Baroda. Today, I am presenting on role of plant proteases in plant development. So we all know that this program cell death occurs throughout the life of plant, as this picture depicts clearly that throughout the life cycle of plant, whether it is senescence, or it is tertiary element formation, or parenchyma formation, or root cells, everywhere this PCD plays important role. When we compare the PCD of this animal cell shown on the C, in picture C, we can see that first apoptotic body formation takes place, and the macrophages engulf that cells, and cell death occurs in case of animals. But when in case of plants, it has rigid cell walls, and this apoptotic body formation, and this cannot happen. So there are two types of cell death occurs in plant cell. The first is figure A, this is necrosis, and third is vacuolar cell. Second is figure B, that is vacuolar cell death. So the key enzyme which are important in this animal cellular death is caspases, but plants surprisingly do not have caspases, and we need to
simple simple nothing.
We need to check the candidates which are present in this plant cell in case of plant cell, and thus the already research articles showed that there are few candidates called as metacaspases which are there in plants, which can play similar role to the caspases which are there in animals. So based on this, we have identified metacaspases in fungi, protozoa, and plant, which has similar structural properties just like caspases, and they are cysteine-dependent arginine lysine specific proteases. Plants have both type of metacaspases, whereas fungi and protozoa has only one type of metacaspases. So we selected few economically important plants like potato, tomato, rice, tobacco, grape, capsicum, brinjal, and Arabidopsis, obviously, to check the presence of metacaspase gene family. And the number depicting under the image shows the number of metacaspases present in that plant. So we have took this metacaspases and went for phylogenetic analysis and identified total 75 orthologs, in which 52 orthologs are from Solanaceae family, that is from potato, tomato, eggplant, capsicum, and tobacco. Tomato and tobacco also have their biotype alternatives. That's why there are total 7 Solanaceae family members. And other than the Solanaceae members, there are 3 other members: Arabidopsis, grape, and rice. This all 75 members are distributed in 2 groups, type 1 and type 2, which we can clearly see from this phylogeny. The right one group is the type 1 group, and left one group is type 2 group. Then we went for ortholog identification and evolutionary divergence of analysis of metacaspases, for which we have used these synonymous and non-synonymous substitution of this metacaspases. What is this? This is how the during the evolution, how these metacaspases are evolved with the plant evolution, and we found that the DNDS ratio was less than 1. When DNDS ratio was less than 1, it suggests that these gene pairs are under the purifying selection or positive selection or natural selection, we can say. And these are the depiction of the molecular speciation time of the gene family and as well as plants. These are the Sarko's analysis to show the orthologous relationship. Then we also checked expression analysis of these metacaspases in different these tissues, developing tissues of potato plant. This is tuber, sprout, teeth. This is cortex, whole tuber, and its mesocarp and endocarp layer, and check the expression of that 8 metacaspase gene present in the this, and we found that there is a differential expression. So what we can get with this result, we got to know that suppose this one is getting very high expression, we can select this gene, this candidate gene. We can either overexpress or this silence or knock out this gene, and we can have better kind of plant or product. This work was already been published back in 2019. And I would like to thank for the DSP sir for the financial support, and I would like to have questions.
Thank you so much, Maitri, for such a wonderful presentation. Now we'll have questions from our panelists.
What is the approach you have used to identify the proteins? You have just downloaded from database?
No, ma'am. First, as I have showed here, this Arabidopsis, this metacaspase data was there, but other data was not there. So I procured the data from Arabidopsis, and using BLAST search, I have identified other metacaspases in all other plant family.
Okay, so just that means you have first do the in silico work, then with the QRT-PCR you have just checked the expression of metacaspase.
Yes. That is how we got this candidate gene, and then the future work which has already been done, but I am not presenting due to time limit. That we have selected this candidate gene and went for overexpression as well as silencing as well as knockout in the plant.
Okay, so what you do with silencing and like genetic engineering?
As I have shown that these metacaspases are really important for development, and if we silence this gene, it will surely have some impact for the development, right? So for example, in one of the result what I am getting is that senescence is getting delayed in the transgenic plant. So we will have longer life of the plant. So we will have more number of potatoes from the plant. That is just an example.
Thank you.
Thank you so much, Maitri.
Okay.
So our next presenter is Dr. Haider Abbas Masih. Dr. Haider, are you there?
Hello. Yes.
Yes, you are audible. So our next presenter is Dr. Haider Abbas Masih from Gujarat Biotechnology Research Center. And the title of his presentation is Bacillus mujavensis, an endophytic bacteria combating salt stress in rice.
Okay. Very good afternoon. So am I audible?
Yes, you're audible and your screen is also visible. You can go ahead.
Okay. So my topic is about Bacillus mujavensis, which is an endophytic bacteria, which is actually used in the combating salt stress in the rice. So initially, to understand what is the endophytes are a basic microbes. This can be a bacterial or a fungus. So they associated with these plants, particularly root systems, and then they are providing the plant growth promoting activities. Okay, and for that, actually, they have to enter inside the plants. So in entering entrations in this plant is facilitated by the several enzymes such as pectinases, chitinases, and cellulosis, which helps in the degradations of plant cell walls. And after entering inside this plant, they are asymptomatic actually, and they are facilitating with these several criteria. For example, that is plant growth promoting traits such as phosphate solubilizations, nitrogen fixations, then plant hormone productions. Okay, and then others, some of those bacteria also promotes salt stress mitigations. That could be by abiotic stress management, like prolines and sugar synthesis, exopolysaccharide secretions, and volatiles productions, and glycine butane contents, and dehydrins, and also. So these are all these criteria in which they are providing such chemical constituents to these root systems. Particularly, I'm talking about the root systems, because my bacteria was isolated from root, and I have given the treatment in the root also. But that doesn't mean that bacteria doesn't present in the aerial parts. They are also present in the aerial parts, and they can be also cultivated from that. And these bacteria, I am talking about this bacteria only, but there are also fungus. For example, SWAM fungus and all. They are very classified funguses are there. So along with that, they are providing the stress management. This could be in terms of biotic or abiotic both. So if we consider rot, salts, or heavy metal stress, in that conditions, bacteria is in association with these plants' root systems in rhizospheric conditions. They are actually making some of the chemical constituents. For example, some of them, this could be hormones also. Okay, and that helps the plants to survive under adverse conditions. So these are the ways they are actually working. So I have just given the overview in this figure. There are multiple other pathways through which they are working actually, and this crosstalk is very much important. That sometimes it is believed that the bacterial consortia also, or a single bacteria also, they are modulating the gene expressions of this plant system also at certain extent. And so in that cases, they are believed to provide certain secondary metabolites is also, okay, which are the secondary metabolites for these bacterial conditions. But this can be used by plant systems to provide this specific stress, provide specific stress mitigations. So this could be achieved by a crosstalk between these plants and bacteria. Okay, so there are the several other mechanisms. It is just listed over here. So basically, we have to identify these plant growth promoting activities. This could be done by the several biochemical analysis. For example, phosphate solubilization is done by this Picoskaya agar. Then siderophore production is done by the CAS media. So there are several and nitrogen fixation, for example, Jensen's media and all. So there are several media. I am not discussing all this media and all, but this is the basic microbiological aspects we can isolate from these sterile conditions. So after that, we can inoculate that particular bacteria. After identifying, after screening all these parameters, we can identify several bacteria, and that bacteria could be multiplied inside in vitro conditions, and that can be used as a biofertilizers. Okay, so this is the whole idea behind endophytes and plant associative studies. And there are several ways of application. For example, this can be applied by the sprays. This could be done by the seed treatments. Okay, and this could be also done by this sowing of granules or beads, which can be given to the rhizospheric soils during this cultivation practice in this agriculture. So there are several ways by which these endophytes could be given to these plants artificially also. Naturally, no plant is actually sterile in the nature conditions. So that is the one point to note it out, that all plants in natural conditions are having one or other types of this bacteria, and they are actually getting benefited from that associations, just like it was started with this rhizobia. Yeah, and we know very well examples of other bacteria also, like Pseudomonas and others, Azotobacters and all. So there are several other examples also there. And so this is the whole workflow by which first we have to identify the plants, which plants you want to study. For example, in my studies, I have isolated the bacteria or endophytes that is from salt tolerant species, some of the Salvadora persica and Salicornia brachiata and all. So in that conditions, so that plant is already having certain extent of salt tolerances, and also it is inhabited with these bacteria. So that bacteria could be isolated in laboratory conditions, and we have to follow the stringent or specific surface sterilization techniques from which we have to isolate these bacteria. And these endophytes could be cultivated inside laboratories, and these could be.
Conclude your presentation in 30 seconds.
1 minute. Okay. So I'm just moving towards directly to the results. Okay, so I'm just giving the introduction regarding endophytes. So I have isolated this Bacillus mojavensis, and this Bacillus mojavensis is having the plant growth promoting activities. You can see that this is the treatment in which the only endophytes treated plants, and over here endophytes with this salt stress. We can differentiate that there is the good result of endophyte treatment. And there are others, analysis was done that was with these gene expression studies, in which the genes related to these plant growth promotions along with these salts mitigations were upregulated. Mostly these genes are in the upregulation forms, but that is not the whole case. Sometimes you might get this in the downregulated genes also, particularly talking about this EIL and all. So they are reported in several papers that these genes are having the stress tolerance mechanisms, and that's why it is getting upregulated. So overall, in this paper, I want to conclude that there are many ways to isolate these endophytes, and we can actually study between this crosstalk between plants and microbes, that is endophytes, and we can further use this association to understand the beneficial genes which are present that could be upregulated or downregulated, and these endophytes could be used for making these farm-based formulations. Thank you.
Thank you so much, Dr. Haider, for this presentation. Now we'll have questions from our panelists.
Dr. Haider,
yes,
there are different, all our other organisms are also there which can be used or identified for the salt stress in rice. Okay.
Yes.
So
yes,
what is the yes, ji boliye.
Yes, there are many organisms, because nothing, no plant part is actually sterile. Actually, in if we see in this, if we analyze.
Okay, so the presence of this bacteria also depends upon the geographical location.
Yes.
Different rice plant in different places get different organisms?
Yes, and that's what the productivity is actually getting increases also. Some of them, they are cultivating several other or several bacteria or even algae also is there. So there are, that depends upon the geographical area and then the treatments and types of the soils also.
So how this bacteria can be used for further in cultivation?
So what we can do that is we can identify such bacteria, and that bacteria could be characterized, and after that we can provide these formulations to these farmers, so that could be used for salt stress mitigations actually, so that plants can survive in adverse conditions also. That is the first part, and we can easily understand between these genes, and there are several results also is there. They have isolated these genes, particular genes which from this bacteria, and incorporated into these plants, and they made this transgenic, and they got these successful results for salt stress mitigations also.
Okay, thank you.
Okay, thank you very much, ma'am.
Dr. Heather, what is the role of lysine betaine in the maintenance of osmotic potential?
Actually, glycine betaine is the actually osmolytic compounds which maintains the osmosis of cells, inside which there is all biochemicals or enzymatic activations are doing. So what is happening during the salt stress? These secondary metabolites and reactive oxygen species, particularly reactive oxygen species, it's a major role. They are creating the chaos inside the cells, and to protect these enzymes, they are making these hydrations around these enzymes, and that's why these osmolytes are very much important.
Thank you so much, Dr. Heather.
Yes, thank you, ma'am.
Moving to our next theme, that is environmental biotechnology. Our first presenter is Nirali Mehta. Nirali, are you there?
Yes, ma'am, I am here.
Okay, so you can share your screen. So we have Nirali Mehta. She is from Botany Department, Sri PP Institute of Science, Maharaja Krishna Kumar Singh Ji, Bhavnagar University, Bhavnagar. And the title of her presentation is Quality Analysis of Coastal Water at Habitat Coast, Hathab Coast, Gujarat, India.
Okay, ma'am. My slide is visible?
Yes, your slide is visible. And
Okay.
you're audible too. You can go ahead.
Okay, ma'am. Thank you. Okay, so because of the time limit, I directly forward on my introduction slide. But before that, my topic is the quality analysis of coastal water at Hathab Coast, Gujarat, India. So under this title, first of all, we all know coastal area is a very complex and dynamic in nature, as it contains the different kind of the marine flora, fauna, and other biotic and abiotic component, right? Which is directly or directly affect the marine environment. But if we want to understand the marine environment, then first we have to understand the nature of the hydrological component of seawater. And that's why seawater quality monitoring is very necessary. Right? And Gujarat coast consists of 1600 kilometer long coastline with 14 coastal districts. And from that, the Hathab coast is located in Bhavnagar district, which is 28 kilometers far from the Bhavnagar city. And Hathab coast is muddy, as it contained majority of the portion muddy sediment, because if we talk about those sediments, then all sediments is like a clay particles, like a clay sediments. That's why this coast is we can consider as a muddy coast. And for the monitoring or analysis purpose, seawater chemistry measured by the multi-parameter kit for 3 seasons for 1 year study. And different parameter like temperature, pH, electrical conductivity, TDS, dissolved oxygen, biological oxygen demand, turbidity were measured. So as a result, in summer season, we can see here in summer season, every parameter or you can say that the water chemistry is increases with increase in a temperature, because red because mostly chemical reaction is generally increase at the high temperature. That's why we can see here the mostly parameter, mostly value is increases with the increase in temperature. But in case of the turbidity was increased in monsoon season due to the rainfall, which leads input of the more organic, inorganic particles, some large size of the debris, large size of the sediments in the seawater through the water runoff from the land or river to oceans. There are lots of farms is near to coastline, so that is also reason for the high turbidity. So basically, this investigation conclude that the data which observe and record will be important when anyone want to do further study at this site, because it shows the condition of the marine environment. So in that case, if we want to study on climate change effect, diversity of the marine organism, that then at that time this investigated data we can use as a base data, and as well as we can aware other peoples to conserve our coast by the making some planning and strategy. And that's why this study will be carried out. So this is my reference. And I would like to thank my guide, Dr. Shailesh Mehta sir, for their guidance, Education Department, Government of Gujarat for providing short scholarship, and Dr. Bharat Singh Gohil, Head of the Life Science Department, for providing necessary facility and his supports. So thank you everyone for listening me. If there are any question, please may you ask. You can ask.
Thank you so much, Nirali. We have a question from panelists.
What is the permissible limit for potable water? For
Hello, sir.
What is the permissible limit for the potable water for BOD?
Sir, actually, normally it's in a range, but at cost of the at Hatha coast, it's vary from season to season.
Okay. What is the reason of increased chemical reaction during the summertime?
Sorry, hello, sir. What is?
There is increase in the chemical reactions during the summer. You
Yes,
showed
sir.
one graph. So what is the reason for increased reactivity during the summer?
Which activity, sir? Sorry, pardon.
So you have shown one graph in which there were several parameters like pH and everything. So what is
Yes.
the reason? Yeah, this one. So what is the reason of, and while presenting you said that during the summer there is increase in chemical reaction, rate of chemical reaction, right?
Chemical value, sir. Yes.
So what is the reason for that?
Sir,
Can you
because
explain it? It is related to some thermodynamics. Can you explain what is the, why
sir,
the
actually
rate of
what
reaction
happened? Yes,
increasing
yes.
during the summer?
Yes, sir. Sir, what happened actually, when temperature is goes up, then what happened? The temperature will change the chemical mechanism. If we talk about the electrical conductivity or total dissolved solids, in that case, was happened, the minerals particles or molecules or whatever present in seawater, the transformation or movement of ions and all molecules will goes high, right? That's why TDS value is goes high, is increases. EC, electrical conductivity, also increases. In case of the turbidity, then in the summer it is a low. pH will be also increased, because at a high temperature the concentration of CO2 particular in a seawater will decreases. That why the buffering capacity is goes down, and that lead to the high pH concentration, high pH.
Thank you so much, Nirali.
Okay, ma'am. Thank
So
you.
our next presenter is Alka Kumari. Alka, are you there? Alka?
Hello.
Yes, you are audible, Alka. So our next presenter is Alka Kumari from Plant Omics Division, CSIR CSM CRI, Bhavnagar. And the title of our presentation is Isolation and Cloning of Plastic Degrading Gene from Marine Bacterium. The screen is visible, Alka. You can make it PowerPoint.
Is it in the slideshow mode? Okay.
No. Is it? No. Yes. Now it is. You can go ahead.
My presentation topic is isolation and cloning of plastic degrading gene from marine bacteria. As the topic suggests, I'm going to talk on the plastic pollution, as the plastics. The problem we focused on is the plastic, the complex and stable structure of plastic provides it a high hydrophobicity and makes it non-degradability. The plastic waste has been increased from 2 million to 300 million tons since the 1915 to 2015 worldwide. And according to the Pollution Control Board, India has suggested up to 6 million tons of plastic generation every year, in which the only PET contributes to the 20% of the world plastic waste. Incineration, landfilling, and recycling are the approaches which are practiced from the very earlier time, which releases toxic chemicals like carbon monoxide, CO2, dioxin, furan, which causes land fertility and high health risk. Bioremediation is an eco-friendly solution which has been studied from very earlier time till now, but has due to low efficiency and inadequacy in the approaches according as per international standard regulations. We have now the modern biotechnological approaches such as high throughput sequencing and molecular cloning present today. We can target the gene responsible for the particular activity like plastic degradation. Here we studied the marine bacteria as it has a pioneering surface colonizing, and they have reported to have role in biogeochemical cycling. So this is the pictorial representation of our overall work. So we have screened the bacterial isolates, where some bacterial isolates are able to grow in presence of plastic, and others are not growing, starving, and it's like dead. And for knowing the degradation, detail of the degradation, we can study the expression profile. We can study the expression profile and trace the gene through genome-wide comparative transcriptome involved during the PET utilization. Then the isolation and cloning of the involved gene through molecular cloning technique, and expressing the required gene in the bacterial expression system, and for targeted and improved degradations. First, we screen the bacterial isolate for the presence of bacterial strain in presence of plastic and without plastic. So we observed that in the tetrazolium reduction assay, which is a colorless compound turned into the red color, shows the growth of the viable bacteria in presence of PET plastic. And the hydrophobicity of the strain was also checked by during the log phase. Which shows the bacteria has affinity towards the hydrophobic hexadecane at up to the 200 microliter concentration has shown 55% of turbidity, and in through microscopic observation also the bacterial adherence was observed over the PET film, and this through the fluorescent probe the viral bacteria can be seen on the surface of the PET film, and through the same micrograph the bacterial adherence was also observed, and the surface deterioration was observed after washing the after washing the PET film after incubation in comparison to the control film. This is the structural group study where we found that the PET film growing with bacteria has increased the carbonyl and ester bond indexes and reducing vinyl and internal bond indexes, which shows that bacteria has PET film has got some functional group, and reduce the reduction in the internal bonds, and through the degradation was confirmed through weight loss determination and cumulative CO2 evolution resulting from the basal respiration of the bacterial strain. So it was observed that up to the 2% of the weight reduction has been observed after 90 days of incubation, and the cumulative evolution of CO2 was up to the 120 mg per gram carbon. The genome-wide comparative transcriptome expression profiling suggested that in the treated condition, that is in presence of the PET, bacteria has mainly the hydrolytic gene and transporters and carbon metabolizing genes are upregulated, while in the control conditions the spore forming genes are upregulated. The transcriptome profiling was validated through QRT-PCR, and the correlation plot was significant. This is the proposed PET metabolism, which is proposed according to the pathway prediction system by the University of Minnesota, where we found that the PET based and also
conclude your presentation in 30 seconds.
Yes, ma'am, only 2 slides there. And the PET metabolisms, we found that the PET is hydrolyzed, the carboxyl esterase is the candidate enzyme, we choose as a candidate enzyme as it is hydrolyzing through the two product, that is bis hydroxyethyl and terephthalate, and we have other side it is going with the aldehyde dehydrogenase, and we choose the carboxyl esterase for cloning study, and we clone the carboxyl esterase of which was a 606 base pair gene in PGMT cloning vector. Then we digested the PGMT PGMT construct, and we cloned the digested product in gene product into the PET expression vector, and we confirmed that after restriction digestion and PCR amplification, then we transform it into the E. coli. After transforming, we confirm the expression of the protein by checking it on the page SDS, and the activity of the protein was checked from the purified fraction, and the activity was in presence of by providing the PET as a substrate, and we got up to the 30 unit per microliter of activity after 36 hours, and the weight loss of up to 1.2% of the weight loss after 36 hours. And the phylogenetic characterization has been done through nearest neighbor algorithm of carboxyl esterase with other esterases involved during the polyester degradation. and it has forming separate clad with 67% of novelty. So
Please conclude.
this work is concluded as this marine strain has the potential to adhere and actively grow when PET provided as the carbon source. The PET degradation was confirmed by altered surface topography, functional group, weight loss, and CO2 mineralization. The highly expressed gene annotated in a high throughput RNA sequence QRT-PCR and selected for cloning in bacterial expression system. The degradation pathway had been elucidated based on the changes in functional group, RNA sequencing, and qPCR observation. The carboxyl esterase is candidate enzyme for hydrolyzing PET into BET and TPA, and to met by aldehyde dehydrogenase activity. CXC gene isolated from marine strain was cloned in a bacterial expression system. The expressed protein was characterized and confirmed through PET degrading activity. Thank you so much.
Thank you so much, Alka, for such a nice presentation. Now I request the panelists to ask the questions.
So which
Yes, sir.
expression vector did you use for the transformation of CXC?
We used PET 28A plus.
Okay. And what was the IPTG concentration for the induction?
160 millimolar.
160 millimolar?
Yeah.
For at what temperature?
30. We optimize the temperature. Actually, there is more data for optimization of the step, but for this data, we use 30 degrees Celsius. We choose it was optimum at 30 degrees.
Okay. And did the CXC gene was upregulated in your transcriptome as well?
Yes, sir. I can show you.
Thank you so much, Alka.
Thank you.
Our next presenter is Dr. Paritosh Parmar from Gujarat Biotechnology Research Center. And the title of his topic is Depicting the Nexus of Heavy Metal Bioremediation Using Bioluminescent Bacteria.
Shall I start? Hello, shall I start?
Yes. Screen is visible
So
and you're audible.
I'm going to present my PhD work entitled under Depicting the Nexus of Heavy Metal Bioremediation Using Bioluminescent Bacteria. So these are the contents. Introduction summarizes this graphical abstract summarizes the research problem as well as the proposed solution. As we all know that heavy metal gets water system and land natural environmental system get contaminated by heavy metals by using through various natural as well as anthropogenic sources. This contamination has to be detected, monitored, and remediated continuously in order to control or to maintain the environmental stability. There are certain physical and chemical analysis method available, but due to their major advantages, the researchers have opted for biological approach. In biological approach, major diverse microbes have been used to remediate this kind of heavy metal. But we believe that bioluminescent bacteria may have upper hand among those microbes because it can detect the heavy metal presence. It can also monitor the heavy metal presence, and it can also remediate the heavy metals by one way or the other way. So this is how the heavy metal gets entered into the food chain by biomagnification process. This figure shows the biochemical and molecular aspects of luminescence in the Vibrio fishery. A global scenario, as well as we look into global and national scenario, many work has been carried out by using bioluminescent bacteria to detect and remediate the heavy metals. So these are the objective: isolation, biochemical characterization, and molecular identification of bioluminescent bacteria, assessment of heavy metal tolerance and absorption by means of exopolysaccharides produced by this bioluminescent organism, application of bioluminescent bacterium in heavy metal detection by means of bioluminescence inhibition assay, and application of dead biomass to remediate the heavy metal. I'm going to skip the material and method part due to time constraint. So however, this graphical abstract summarizes the objective 1 and 2. This graphical abstract summarizes the objective 3, that is the bioluminescence inhibition assay, the detection of heavy metal, and the simultaneous removal of lead and cadmium using this dead biomass of the bioluminescent organism. So in the resultant discussion, we found two isolate from two different organisms. One was screened from Indian mickerel and labeled as PBR1. Second was screened from Indian squid and labeled as PBL. Both of them were gram-negative organism. This table represents the biochemical characterization compared with the other closely related species of both isolates, PBR1 and PBL1. The PBR1 was identified as Vibrio alginolaticus and PBR1 was identified as Vibrio rotiferinus through 16S sequencing approach. Harvick clade of Vibrio genus include pathogenic and as well as non-pathogenic bacteria with the inclusion of Vibrio alginolaticus into pathogenic group. That's why we had to go for the hemolysin test, and we found that both the isolates were tested negative for hemolysin test, so that we can use them further for the study. Both of them are able to produce APS. PBR1 was able to produce 1.3 gram per percent of APS, and PBL1 was able to produce 2.45 gram percent of APS from 5% sucrose. This graph shows the heavy metal tolerance assessment of both the isolate in terms of zone of growth inhibitions, where we found that cadmium was found to be most toxic and copper was found to be least toxic. However, the both organism PBR1 and PBL1 was found to be resistant against lead and strontium. This photograph shows the effect of heavy metal and their various concentration on biomass growth as well as luminescence. As you can see, this indicates that the low concentration of cerium, mercury, and arsenic enhances the luminescence, where the higher concentration have a detrimental effect on the growth and luminescence directly. Here you can see the effect of heavy metal cadmium on growth as well as on luminescence also. And in cerium, you can see the particular concentration, the bioluminescence has observed to be enhanced. The further FTR analysis showed that the APS produced by both the organisms are able to bind with the metal tested under the study, that is PP, lead and strontium. The FTR spectra revealed that hydroxyl, carboxyl, and phosphate groups were majorly responsible for the binding of these metals onto APS. The shifting of peaks and change in the intensity of peaks area suggest the increase or decrease in the functional group content directly engaging with the metal. Similar results were also obtained on the second isolate. The assessment of heavy metal toxicity, you can see the green, the value you can see over here in green font are the EC50 value, that is the concentration at which the luminescence get reduced by 50%. So we have tested 6 heavy metals: arsenic, cadmium, chromium, and copper, then lead and mercury. And we observed that the cadmium, in case of cadmium and chromium, our isolate was found to be better detector when compared to the other commercially available kits. This table shows the media optimization by box Bentham design. We took 3 factors, that is pH, bioabsorbent dosage, and metal concentration into the study. Here you can see the actual values that were obtained during the study and the predicted values. The model and the value suggests the model, both the model for the both cadmium removal as well as lead removal was non-significant, whereas lack of it was found to be non-significant. Yes, ma'am. This shows the normal distribution of data. These are the surface plots of showing the interactive effect of the factors. This is for lead. And further, research showed that bioabsorption efficiency of Vibrio alginolaticus was found to be 58.24% cadmium, and for cadmium, and 82.92% for leadmium. Mind well, this study was conducted with the simultaneous removal for the cadmium and leadmium. So the system contains both cadmium and lead as well. And further we confirm through the FTR analysis that the biomass of PBR1 is able to absorb these metals present in the system. So we isolated two organisms from the two different marine organisms. That was the first report of violation Vibrio alginolyticus and rotiferins to be polysaccharide producers. The FTR analysis also revealed that PPS can bind with the lead and strontium, and they both the organism can grow in the presence of these two metals. Sensitivity against Vibrio alginolyticus, the PBR1 showed the better sensitivity against arsenic, cadmium, chromium, and lead. And the dead biomass of Vibrio alginolyticus was employed for the simultaneous removal of lead and cadmium. And this was the first report depicting the simultaneous removal of the metals by the Vibrio species. These are the references. 3 research publications were made out of this study. And thank you.
Thank you so much, Paritosh, for such a wonderful presentation. I request the panelists to ask the question.
Paritosh, just a
Yes,
basic question. Is there any relationship between the heavy metals and bioluminescent bacteria?
Yes, ma'am.
No?
Actually, it is directly related. Heavy metal acts as a pollutant for the organism, though it has a detrimental effect on organism. If organism's growth is affected, their luminescence is also tends to get reduced. That is why they are commercially used for the detection of this kind of pollutants.
Okay, no, there is any physiological or mechanistic relationship? Means like.
They get inside and they can bind to the DNA and other organelles, so that the organism failed to grow further, and that is how the cytotoxic effect works, and the luminescence get reduced, or they are not able to produce HSSL, which is the key inducer for the luminescence.
And what is the rationale to be a bioluminescent? Because you can, if you are doing just growth study, you can do with normal bacteria also.
Yes, so we have tested the bioluminescence sensor that 6 metal I have shown in the second objective for sensor. The PBR1 was found to be better sensor in terms of cadmium and chromium detection when compared to the other commercially available kits.
Okay.
So this is the single organism through which we can detect, monitor, and remediate the heavy metal.
Work as a biosensor.
Sorry.
Can we
yes.
use
If the organisms are resistant towards the heavy metal, so they can be employed as a bioremediating agent. And if they are sensitive towards the heavy metal, so they can be employed as a biosensor. That was the idea behind the choosing for this particular bacteria.
So Paritosh, mostly this FTIR is used for the characterization of organic molecules, as
Yes,
most
sir.
of the metal ions are not in active region of IR. So how
Yes.
did you correlate the spectra that you have shown with respect to the metal ions? And did you use some ICP, OES, MS, or AES techniques for?
Sir, ICP I have used for bioabsorption study, and as far as the FTR spectra is concerned, I have gone through the previous report published by the reputed journals, and they have stated that the change in the peaks and shifting of the peaks can be correlated with the bioabsorption mechanism.
And did you check the ICP as well?
Yes, sir. In absorption study, the values that we have obtained, that is through the ICP. This.
Okay, and did you also calculate the hazard quotient for this cadmium and lead?
Sorry, sir.
Did you calculate the hazard quotient for cadmium and lead?
No, sir.
Okay.
I have just checked the viability. I have just checked if the metals are getting detected by the organism, or they can be remediated by the organism by means of biomeans or biomass or EPS only.
Actually, hazard quotient you should have calculated, because that is the indicator certified by CDC. If
Okay.
the concentration of cadmium and lead is above that hazard portion, then it is only concerned. Otherwise,
Sir, that is actually the main concern for choosing the cadmium and lead. As you can see over here, the concentration that was found in Gulf and Khambat and the South Gujarat region in mangrove sediments were higher. That is why we selected this two metal for the study. That is was based on the literature survey.
Thank you so much, Paritosh. And next presenter is Shrikant Khande. Shrikant, are you there?
Yes, ma'am, I'm here.
Yes. Okay. So our next presenter is Shrikant Khande from CSIR CSM CRI, and the title of his presentation is Enzyme-Based Biodegradation of Low-Density Polyethylene Plastic Film by Marine Bacteria. Over to you, Shrikant. Yes, your screen is visible. You can make it presentation mode.
Ma'am.
Yes.
Good afternoon, everyone. My topic for today presentation is enzyme-based biodegradation of low-density polyethylene plastic by marine bacteria. This work was carried under guidance of Dr. D.R. Chaudhary and Professor Bhavna Jha. We talk about overview of my biodegradation study. I started with the isolation of marine bacteria. This marine bacteria was isolated from seawater and plastic debris. This marine totally around 200 marine bacteria I have isolated and gone for screening for LDP degradation. Total 3 marine bacteria I have got shown a potential candidate for degradation of LDP film in screening method. Further, I've gone through a biodegradation assay. After completion of my biodegradation assay, I've gone for the LDP characterization, the film which is treated by the bacteria and the control film. Both are tested and characterized to check whether what are the changes made in this film to the bacteria. Similarly, I carried out some enzymes assay and enzyme isolation and check to check which are the enzymes responsible for the degradation process. Starting with identification, with marine bacteria, in which two are Marino species bacteria, 46 and 244, and one is Bacillus subtilis. Total 3 marine bacteria. This is Interferons ZMB. You can clearly visible over here. We talk about the characterization. I've gone for FESM and FM analysis. FESM, this control film, very clear. It's not shown any erosion or crack and all. But after the bacterial treatment is shown a high surface erosion and high roughness compared to the control film. Similarly, here we can see in AFM analysis, this control film are smooth and they have shown a major cavities and pits over the bacterial treated film. I gone to check the bacterial degradation by weight loss assay, in which weight loss by bacteria H246 has shown a maximum weight loss. It is around 1.68% after 90 days of bacterial treatment. Similarly, I have checked the thermal stability of this film after 90 days to check whether what is the strength of it as compared to, you can see here clearly the control film is showing a high thermal stability as compared to the bacterial treated film has decreased gradually, and the complete degradation was up to 99.68% by bacterial isolate H246. Similarly, we have done the carbon content in the degradation area, where in control film we can see 86% percentage of degradation the content of carbon, but in bacterial treatment it has reduced gradually as compared to the control film. Similarly, we gone through the tensile strength also, and it's shown a maximum decrease in the tensile strength. Similarly, we gone through the surface analysis by AFM method, and it's shown a very high roughness on the PVC surface, on the LDP surface. Similarly, we check on the FTR analysis, in which here this peak at 2200 to 2400 is shown an increase in its intensity because of more stretching of carbon-carbon bond and interstretching of oxygen-carbon bond, which is mostly possible due to the bacterial action on that film. And further, there was one more addition of peak here as compared to the control, which is not observed in control, which clearly indicate the CO stretching and the alcohol group stretching. Further, we confirmed this data with the GCMS, and we found that there was a formation of carboxylic acids in the media after the during the bacterial treatment. And we found that there was one more compound, which is this phosphate, which is used as a which is used as a plastic stabilizer by the companies to make the plastic. And in control, it was not shown. So what make the plastic degrade? So we gone through the enzyme isolation and purification, in which we have seen we have gone for multiple numbers of enzyme screening, in which esterase has shown a good candidate of degradation in my process. In enzyme assay, we have seen there was an increase in the enzyme quantity till the 20th date of my incubation, and further it's shown a steady pattern in all three bacteria. This enzyme was isolated further by using ammonium sulfate method and sulfatic G50 G50 chromatography column, and we found that there was a 32 kd of band in Maridobacter species and 42 kd in Bacillus species. And further this was carried confirmed by native gel analysis, and it's shown a clear blue color halos over here. The highest activity during my experiment was 0.60 unit per ml by S244 and
You can get 30 seconds to conclude your talk.
And 0.50 unit per ml by both bacteria. And this purified enzyme shown 53 to 60% of recovery after the chromatography column of Cephalix G50. When I conclude, biodegradation is an eco-friendly process. Similarly, my three bacteria have shown a very good rate of degradation, and the esterase enzyme are responsible, which are clearly isolated by Cephalix G50 column, and this can be used in waste management also. My future prospect is to characterize this enzyme on the basis of pH, temperature, organic solvent, etc. Similarly, I want to carry out the toxicity analysis of the degraded byproduct which are released in the media to check whether it is harmful or not. Similarly, I want to do a transcriptome analysis for the enzyme, at what time these enzymes are responsible is regulating for the degradation process. I greatly acknowledge CSIR New Delhi for providing fund for this work. Similarly, I acknowledge funding agency Bharti Pune and UGC New Delhi for providing me fellowship. Thanks to my colleagues during this work. Thank you.
Thank you so much, Shrikant, for this presentation. I request the panelists to ask the question.
So, Shrikant, how did you confirm the degradation of LDP? Like, what are the changes?
Sorry, sir, I can't hear. Hello.
Okay. So, can you go to the FTIS spectra slide?
Yes, sir.
So, can you explain like how the bacteria affects the degradation of? Is it just the intensity of the peak, or you are getting some novel peak? Because looking at this graph, all of them look.
Yes, sir. Yes, sir. I will explain. So, here you can see in control film at this region, below 1500 region, it's very hard to have a peak without any treatment. So after bacterial treatment, it was a broad, small peak in all three bacterial treated film. But at the region of 2000 to 2400, the intensity was very low. So we tried for multiple time and we found that this intensity was higher in the bacterial treated field. And this intensity was mainly because of the stretching of carbon-oxygen bond or alcohol group addition, means addition of extra oxygen over the polythene surface, means it's decreasing its hydrophobic nature. That's it.
Do you also have the only bacterial control FTS factor?
Yes, yes.
So
Yes,
that doesn't have these kinds of peaks.
No, bacterial means I have did for the control this without any bacterial treatment, means it's a negative control.
Without any bacterial treatment, it is only LDP.
Yes.
But you should also check the dry powder of your bacterium and kind of have seen if those peaks are overlapping your degradation ones. Then they might also arise from bacterial metabolite. Those are also organic compounds.
No, we before processing this to FTR, we wash it. We are having a particular protocol with which we wash that film, and after that we will process for FTR. Because of that, there will be no any contamination during this process.
Okay, what was the magnification you use for ACM imaging?
Which one, sir? Hello, sir.
For ACM, what was the magnification?
Magnification. Hello?
Magnification of your ACM images, like how 1000x.
Same. Okay, sir. Same. I have gone for 5000x.
Okay.
Because the image for 5000x, even I have taken for other 10,000, 2500, even lower magnification, but it very good images which were shown in 5000x. So which I have put in over here.
What was the acceleration voltage?
Sorry, sir. I don't know about that. Sorry, sir.
Why did you go for marine isolates only?
As we know, sir, ma'am, ultimately this pollution means all the plastic which are going are dumping and getting to the marine environment. And there are many reports which are showing a good degradation rate as compared to the land terrestrial microorganism. So I targeted with this approach to isolate a marine bacteria for the plastic degradation. And we chosen the plastic debris and all near the means 1 to 30 centimeter of seawater column.
And have you checked using the consortia of your isolates?
No, ma'am, no, I have not dealt with consortia.
Okay. Thank you so much, Shrikant.
Thank you.
Our next presenter is Surbhi Agrawal. Surbhi, you can share your screen. Surbhi Agrawal is from Applied Phycology and Biotechnology Division, CSIM and PRI, and she is presenting on potential seaweed feedstocks for the production of bio-based chemicals. Over to you.
Am I audible?
Yes, you are audible.
Okay, ma'am. Is my screen in full screen mode?
No, make it presentation mode.
Okay, ma'am. Just a second. Is it in presentable form?
Yes, you can go ahead.
Okay. Very good afternoon to all. Today I will be presenting on my topic, potential seaweed feedstocks for the production of bio-based chemicals. This I will give a brief introduction about the seaweed as the time does not permit in going further. So I will just give a brief introduction. Seaweeds are multicellular organisms that are also referred as macroscopic algae, live attached to rock or hard substrata in coastal area. And on the basis of pigmentation, they are classified into three types, that is red, green, and brown algae. And since we are residing near Gujarat, so Gujarat provides an advantage for the availability of biomass. That is because Gujarat has the longest coastline in India, that is about 1600 kilometers, which is surrounding by the Arabian Sea. Hence, Gujarat provides a suitable environment for the seaweeds. So we can use seaweeds for various purposes, as we know that seaweeds have very good biochemical constituents and very novel also. So that can be used as chemicals for biofuels and for food and feed, fertilizers, as well as nutraceuticals. So in this presentation, I will be giving more emphasis how seaweeds can be used in biofuels. So there are various advantages of using seaweeds for the biofuel production. Firstly, it serves as a renewable source of energy and which is less explored, and it has fast growth as compared to the terrestrial plants. So unlike terrestrial plants, it does not require any extra nutrients or fertilizers for the growth. And there is also no need of using pesticides, so it does not create any environmental pollution using pesticides or washing of the fertilizer or nutrients in the ocean, in the river bodies. And as seaweed grow in the marine environment, so it does not compete for the land and food like food crops, which are conventionally used for the production of biofuel. It has a very good advantage that it does not require fresh water and directly can be cultivated in the seawater. So there is no need of the fresh water for its cultivation. Next, if we go to the biochemical constituent, it does not require any complex pre-treatment because it does not have lignin content, and very few seaweeds are reported to have very low amount of lignin content, so that is negligible. In methodology, I will discuss from the collected seaweeds, which I have collected from the Gujarat region. I have chosen 3 seaweeds of 3 domains, that is from brown, red, and green seaweeds. Then this have been grounded into the fine powder. Further, for the hydrolysis, that is for saccharification, since seaweed has a major constituent as carbohydrate, so saccharification has to be done for conversion of polysaccharides into monosaccharides. For that, I have chosen sulfuric acid method for the saccharification. This is a 2-step method in which acid at higher concentration was used at lower temperature, and in next step, acid at lower concentration was used at higher temperature. So firstly, the seaweed powder was hydrolyzed with the help of sulfuric acid. That is about 72% concentration sulfuric acid was used, and the temperature was given at the room temperature. Then further, for the second step, it was bring to the 4% concentration, that is sulfuric acid got down to the 4%, and temperature was given was around 121 degrees Celsius. So this is the seaweed hydrolysate, which was neutralized, and that was further used for the fermentation medium. In this, I have used the yeast as Saccharomyces cerevisiae, that is conventional ethanolic yeast, which is used for the fermentation. And after the 5 fermentation, the hydrolysate was centrifuged and analyzed for GCMS, analyzed with the help of GCMS. In this, I found that GCMS peak depicted the ethanol and acetone peak, which I have cut with using with the standard. These are my results. When the seaweeds was hydrolyzed, these are the monomeric sugar which was found in all these 3 domains of the seaweed, that is red, brown, and the monomers which were found are glucuronic, galacturonic acid, glucose, galactose, and fucose. In red and green seaweed, the galactose content was found to be higher, and the glucose content was found to be higher in green and brown seed. So this shows that the seaweed polymer get hydrolyzed, then they have been produced into the monomeric form, which is easily fermentable and can be very easily utilized by the microorganism. So these are the GCMS chromatogram of all the 3 seaweeds which were used for the fermentation. And in as we can see in the graph, in all the 3 chromatograms, the peak of ethanol and acetone was clearly visible. This shows that seaweed feedstock is ideally used for the production of bioethanol. And since we are residing near the Gujarat coast, there are many seaweeds which are yet to be explored. And as I have already discussed earlier, the advantages and benefits of using the seaweeds. So my study strongly promotes that seaweed can be used for the production of bioethanol.
Could we please conclude in
as
30
well as
seconds?
other chemicals. Thank you, ma'am. I have concluded.
Thank you so much, Survi, for this wonderful presentation. I request the
Thank you,
panelists to
ma'am.
ask the question.
Yes.
Maybe I just want to ask that
Yes,
you
ma'am.
used acid hydrolysis and other fermentation for the production of this ethanol. So
Yes,
it will be extra cost for this ethanol production. Don't you think so? Acid hydrolysis and all these things. Because if
yeah,
we
but
from the sea water, we cannot get that much biomass of that particular seaweed. So that considering that also biomass as well and this acid hydrolysis, both things can increase the cost of this bioethanol. So what are your comments regarding this?
actually, ma'am, the acid hydrolysis was used, and I found that it is better than using the other type of method of hydrolysis, like using the enzyme and all. So I think that could be used for the cost reduction. And if you go to the terrestrial one, so their structure already contains many lignocellulosic structure which uses the complex procedure. So that is minimized in case of seaweed. And as far as if we consider the biomass, then if we go to the coast, then we can use the seaweed as biomass, because there are immense amount of the seaweed which is used to be laid on the sea coast, and that is of no use. So we can use that waste, which we consider it to be waste, into the fruitful one.
But the biomass collection and the transportation and all these parameters will include the cost.
Actually, ma'am, if we considering the transportation matters, so our we are already residing in the Gujarat. So I think this could be the advantage. If we set up some refinery or some plant near coast, then that can be beneficial, and it will also use less cost.
Thank you so much, Surbi.
Thank you, ma'am.
Our next presenter is Jagubai M. Makwana. Jagubai, you can share your screen. Jagubai is a Senior Research Fellow at Department of Renewable Energy Engineering, College of Agricultural Engineering and Technology, Junagadh Agriculture University. And he is presenting on development of biomass pyrolyzer for biochar production.
I am audible, madam?
Yes, you are audible.
Okay. Next. My topic is development of biomass pyrolyzer for the biochar production. So the biomass is a renewable energy sources. So some agriculture waste, likewise the cotton stalk and castor stalk and other agriculture residue. So the farmer is directly burned in the field. So this process is the introduced in farmer, then the extra income to generate in directly for farmer. So first of all, to collect the biomass in the on farmer field, and then the shredding operation, and then pyrolyzer, so some thermal degradation of the biomass, and then produce the biochar. The biochar is normally used in a soil amendment. Then they increase the water holding capacity and listing nutrient and other. The biochar is a charcoal made and for pyrolysis from biochar is a one of the value added of the product. So the higher energy content as compared to higher rank coals. The biochar is as a fuel, and the other use of the biochar is the filtration and absorption of the pollutant as well as the soil amendment. So the by addition of the biochar influence of the physical property of the soil by altering the structure, porosity, pore size, distribution, and density, and potential to improve the plant growth. And then the biochar can reduce the acidity, improve the soil, CEC, water holding capacity, and environment for beneficial microbes. So the objectives to develop the soil capacity 5 kg per base fixed bed pyrolyzers. So the shredding operation of the biomass. Some parameter of the pyrolysis setup, the reactor, manual operator, stirrer, electric heater, and insulator, temperature control and measuring device, feeding hopper, condenser, electric motor, pumps, and stand. So the reactor, first of all, to made the 65 centimeter long and 56 centimeter width of the reactor, and then they for insulated in the insulating material. So the reactor is the insulating and then the covered by 2.5 mm of the GI sheet. Then the manually operated stirrer. The stirrer is in for inner side of the reactor. So the 2.5 centimeter dia of the stirrer and number of the spoke is attachment. Then the electrical heater. So the 90 kilowatt of capacity in heater is installed. The per heater capacity is 600, and with the help of Sira wool is covered. So the Sira wool is to control the heat loss and minimize the heat loss. So some temperature control and measuring devices. So this unit is to set the temperature and time for the pyrolysis operation, and the K-type thermocouple is used for the temperature, and K-type thermocouple sensor holds the range for 0 to 112 degrees centigrade. So the feeding hopper is 45-degree angle of the feeding hopper, and condenser. The condenser is the perforated. This condenser used for to condense the gas and is collect as for the bio oil form. Electric heater, GI stand. So biomass setup is all component are assembled, and that's why to look the pyrolyzers design. So the biochar making setup is developed by pyrolyzers. So the condensers, hopper, and main reactor temperature control, and the cooling system is used for as a potable water to circulate the as a condensers. Then all the parameter of the pyrolyzers like the volume of the reactor, diameter of reactor, height of reactor, capacity of reactor, electric setter. Then the production of the biochar at the various temperature and time. So the 100, 200, 300, and 500 and temperature variation as compared to retention time. So the some elemental analysis of biochar and shredded cotton stock. So the nitrogen content is also increases with increases in temperature. Carbon content is increases with increases in temperature, and hydrogen is also decreases with increase in temperature. And CN ratio is also increases with increase in temperatures. So the carbon content is also beneficial for the soil element, carbon content and nitrogen. So the carbon content is increases with increases the plant growth and nutrient capacity. So the biochar is adding for the soil is to improve the soil quality, water holding capacity, as well as to root media. So the biochar is to reduce the leaching capacity of the fertilizer in the soil. So thank you. Any question?
Thank you so much, Raghubai, for this presentation. Now I request the panelists to ask the question.
Hello. When we add this biomass, this biochar to the soil, then what does it affect the pH of the soil?
The pH is soil is 6 to 8.52, but biochar increases in the soil, so the pH of the soil is maintained, and the saline soil is also improved with adding of the biochar.
How saline soil gets improved?
So more than 8 or more pH. I am also working in this soil application of the biochar.
Does
Oh,
this
with
biochar make the saline
trial.
soil fertile?
Hello.
Hello.
हेलो
Does
आवाज
this
नहीं आ रही
biochar? Hello.
हेलो
Does this biochar make the soil fertile? Saline soil fertile?
हेलो हेलो
Hello. Does this biochar makes the saline soil fertile?
हेलो हेलो मैडम हेलो
Hello, Jagu bhai, can you hear me?
हेलो आवाज मैडम कट हो रही है हेलो
Hello,
नेटवर्क आ रही है हेलो
can you hear me?
यस मैडम बोलिए
एक minute. Does the application of this biochar makes the saline soil fertile, or just
यस
it
मैडम
improves the soil?
yes मैडम biochar is to improve the soil quality.
And is there any parameter per this much acre we have to add this much biochar or anything?
Biochar improves the production yield of the plant growth and root media, and our true experiment was done the wheat or groundnut. The wheat is increased the best result for 20% addition of the biochar, and the groundnut is increased the yield by 15% of the biochar.
Okay. Thank you so much, Jagu bhai.
Okay, madam. Thank you.
Roshni K. Mishra. Roshni, you can share your screen. Roshni Mishra is from Gujarat Biotechnology Research Center, and she is presenting on determination of bioremediation potential of developed consortium in MFC.
Should I start? Hello.
Yes, you are audible and your presentation is also visible. You can go ahead. Over to you.
Okay. Good evening. My name is Roshni Mishra. And today I'll be presenting my dissertation research work that was on the topic determination of bioremediation potential of developed consortium in microbial fuel cell. Okay, so the study was mainly designed around two major environmental problems. That was, the first one was overexploitation of non-renewable resources due to the increase in population, which leads to an increase in demand of energy. By overexploitation, the combustion of fossil fuels, it leads to production of greenhouse gases, which leads to air pollution. And the second problem was of wastewater disposal by large-scale industries such as textile industries and tanneries. They dispose their wastewater, which is above the level permitted by GPCB, that is the BOD and COD level, that leads to the water being polluted, and the water is then non-potable. So the main alternative green sustainable solution was of MFC, and this is the working principle of MFC, and this is the schematic representation of two-chamber MFC system. In MFC system, there are two chambers. The first one is anodic chamber and the second one is cathodic chamber. In the anodic chamber, the wastewater is present and the bacteria or the consortium that we're going to add is going to be present. The anodic chamber is anaerobic in nature, and while the cathodic chamber is has oxygen in it, that is aerobic in nature. The bacteria in the anodic chamber, they utilize the substrate present in the wastewater. They then form biofilm around the electrode. In this case, we have used graphite as the electrode. Then they release electrons, protons, and CO2. The electrons are then they migrate to the external wires, and then they reach the cathodic chamber, and the protons, they are migrated towards the cathodic chamber through a salt bridge that is which contains a PM, that is proton exchange membrane. The electricity that is produced is then measured through a voltmeter through the external wires. So in the study, there were three main objectives. The first one was the characterization of the consortium that was developed. The second one was the electron transfer monitoring of strains, which was done using SEM, and the viability was checked using CFU per ml. And the third one was the determination of bioremediation potential to check the bioelectricity that is being produced after a degradation of aromatic hydrocarbon. These are the materials and methods. The first one was the characterization, in which the nitrogen source was checked, in which the optimal growth of the organisms were checked in static as well as shaking condition. These are the organisms that were used. They were isolated and screened earlier in the lab from actual wastewater collected from a different source of isolations. The second objective was to monitor the electron transfer monitoring. In this, we needed to check the biofilm formation capability of the consortia and the cell viability through 30 days. And in this setup, we have used two sets. The first set contained electrodes that were coated with biofilm, and the second set contained electrodes that weren't coated with biofilm. They were free cell suspended in the media. The third objective was to monitor the efficiency of consortium. Here we optimized the salt bridge length and correlated it with the production of bioelectricity. The second one was to monitor the degradation of naphthalene, which was spiked in the synthetic wastewater. And the third one was to monitor the voltage output using our developed consortia with actual wastewater that was collected from different sites of Vatva. This is the actual setup of our MFC. Here we have used PBS as the catalyte, and we have used synthetic wastewater as well as actual wastewater in the analyte. The volt was measured using a data logger. These setups were arranged in a series. This is the methodology of the optimization of salt bridge and the naphthalene degradation. Both of the procedure are the same. In the salt bridge method, the substrate that was used was sodium benzoate, and in the second practical, the substrate that was used was naphthalene. They were kept in the system for 30 days and was checked for bioelectricity, and the volt was measured after 30 days, as well as the BOD was measured for the third objective was to collect actual wastewater from Vatva. These are the sites. There were 4 sites that were collected. And then these actual wastewater were used along with our consortium for 30 days, and the volt was measured. These are the results and discussions. This is the first objective, in which we measured the optimal growth in different nitrogen sources. Here we can see that after 15 days, our organisms were able to grow optimally when urea was added in the synthetic wastewater. Now, urea is organic in nature, and thus, as our organisms are isolated from actual wastewater, we can say that it is able to utilize organic nitrogen source due to that. This is the second part of the first objective, in which we measured the growth of the organisms in static as well as shaking condition. Here we can see that over time, the bacteria, the consortium grew optimally when it was in static condition, and so it facilitated our experiment, which was also done in static conditions. This is the result of second objective, which was scanning electron micrograph. This is a scanning microelectrograph of activated graphite electrode. We can see the ridges here, which facilitates the latching of the cells and the formation of biofilm. These are the images of set A. Now, set A was the electrode were already coated with biofilm. This is the image of 0 day, and we can see bacillus and cocci and EPS production already on the electrode. These are the images of set A on 15th day. Here we can see that not much, we can't differentiate individual cells, and there is a densely populated EPS. That just means that over time, the organisms were able to form biofilm on the electrode. This is the image of same image of set B. In set B, the electrode weren't coated with biofilm; they were activated, and then free cells were inoculated in the anodic chamber. So we can see on 0 day that the cells are starting to latch onto the ridges of the graphite electrode. These are the images on 15th day in which we can see that there is an EPS production and the cells have clumped together on the electrode surface, and we can differentiate that there are the cocci and the bacillus species are present, which is characteristic of our consortia.
Please conclude, Roshni.
Sorry?
Please conclude.
Oh, okay. This is the cell viability. We checked the plankton cells were less while on the 0th day, and on 15th day, the plankton cells decreased while the cells in the biofilm increased. The third objective was to check the salt bridge optimization for bioelectricity. Here we can see that the salt bridge, which had 3 centimeter in size, produced more electricity. And the BOD reduction was also maximum in the 3 centimeter size separator. This concludes that the decrease in salt bridge size increased the bioelectricity. This is the naphthalene system in which naphthalene was used as substrate, and here we can see that in the 20 ppm naphthalene, the voltage production was more, followed by 40 ppm concentration. The BOD reduction was also seen maximally in the 40 ppm naphthalene construction and then 20 ppm naphthalene concentration. That signified that when present in lower amount, our consortium was able to degrade naphthalene. So for further degradation studies, GCFID was performed, and with different concentration. This is the chromatograph of 20 ppm naphthalene. Here we can see that the above chromatogram is of 0 day and the second one is of 30th day. So we can see that a degradation percent was 93.35%. Here, these are the chromatographs of 40 ppm. The degradation is 66.32%. This is of 60 ppm, that is 64%. 80 ppm, we saw 91.59% degradation. So when present in lower amount, the degradation was more. This is the characteristics of the actual wastewater that was collected. And this is the voltage estimation of monitoring of 30 days, in which we can see that the site to wastewater produced more electricity after 30 days. These are the conclusions and acknowledgements, references. Thank you.
Thank you so much, Roshni, for this lovely presentation. We'll have a question from the panelists.
So Roshni, what did you use for the sample preparation for scanning electron microscopy?
Sorry, sir. Will you repeat the question?
What was the method for the sample preparation of SCM?
We took out we scrapped the cells from electrode. And
did you use any specific agent to maintain the structure or?
No, sir. Actually, I'm not really sure about it. So.
Okay, and so once at any one slide you said that you have collected those cells in series, right?
Yes, sir.
So what happens when you connect them in parallel?
So the internal resistance changes when the combinations of the setup is changed. So like the study was not concluded, and that's why I've not included in it. But when it is connected in CTs, the internal resistance is more. But when you connect it in parallel or in combination, that is in CTs and parallel, it affects the internal resistance. So when the internal resistance decreases, the bioelectricity that is produced over time increases as well.
Thank you so much, Roshni. Our next presenter is Poonam Patel, and she is presenting under the theme of plant biotechnology. Poonam, you can share your screen.
Yes.
Poonam is from Gujarat Biotechnology Research Center. and she is presenting on genetic variability, physiological characteristics, pathogenicity, and molecular diversity of Fusarium oxysporum F species cumini infecting cumin cyminum L in India.
Hello, good evening, everyone. Myself,
Your,
Poonam.
your
Hello?
screen is
Not visible?
No.
Okay. Is it visible now?
No. Yes, now it is visible.
Okay. Okay. Good evening, everyone. Myself, Poonam Patel, and I'm presenting my partial PhD work on genetic variability testing of Fusarium oxysporum cumini in cumin. So as for introduction, cumin is well known as zira, and it is worldwide used as a spice crop. And it is most popular second spice crop after black pepper. And the main production and consumer as well is India. And it is widely cultivated in Gujarat, Rajasthan, and some parts of Madhya Pradesh and Uttar Pradesh. So as I told, it is widely grown in Gujarat and Rajasthan regions. And in cumin production in Gujarat is mainly in Banaskantha, Jamnagar, Junagadh, Porbandar, Surendranagar. But the biggest problem which cumin faces is problem of fungus which causes Fusarium wilt disease, and which is widely destructive disease, and it causes approx 40% yield loss, and it also can increase up to 80%. And it is about 5 to 25% loss is accounted in Gujarat, and 5 to 60% is in Rajasthan. So it overall affects the growth of cumin, and it can affect on at any stage. So the severity of wilt increases with the plant age, and when wilt comes, the plant are dying. So in case of wilt, it is a soil-borne disease, and it infects the cumin plants from roots, and it blocks the vascular region of the of cumin plants, and so the plant movement stops, and thus the plant dies because of loss of water and wilting. So the objective was to identify different isolates of Fusarium oxysporum cumini and its collection, identification, and characterization, and then assessment of genetic variability through RAPD markers and how much virulent it is. So for materials and methods, the cumin variety GC4 was taken from SDAU, that is Sardar Kushinagar University, and the place was Jagudan, and the plants were raised in test tubes, and 2 weeks old plants were infected with the Fusarium spores, and then the samples were collected after 1 week. Treating seedlings were analyzed for the molecular work. So the collection and identification of samples samples of wheat infected cumin were collected from different areas of Gujarat and Rajasthan. And from Rajasthan, the direct isolates were collected, and the identification was done through microscopic observation, and its genetic variability was assessed. For species specificity, it was ITS primers was used, and for variability, the RAPD primers were used. So the collection and identification of samples shows we have collected various isolates, that is from 1 to 13. So total 13 isolates were collected. And from I1 to I8 was from different areas of Gujarat, that is Rajkot, Anand, Jamnagar, Junagadh, Jagudan, SK Nagar, while R1 from I5 was from Rajasthan directly. The isolates were collected from Ajmer University. So the morphological variation among isolates were checked, and there was a wide variation in growth and their pattern of growth, from circular to irregular, like fluffy growth, raised culture, and moderate to sparse, like luxuriant growth. So the pathogenic variation among isolates was denoted to physiological attributes, and we infected the isolates to plants, and we checked the wilting of plants in terms of chlorosis, wilting of leaves, and disease percentage. So I1 and I4, I8 and R1 were 100% disease incidence was observed, while others were like from not infecting, or the symptoms were not seen, or to high wilting. So this is the transverse root sorry.
The screen is also not visible, Poonam.
Oh, sorry, ma'am. Just a second. Okay.
Yes.
Yeah, sorry. Okay. So transverse root section of seedlings infected with different isolates of Vigyan is present in this figure from control, and different isolates which were blocking the vascular region of the roots. And in biochemical assay, we did the different protein, total protein, soluble protein assay, peroxidase activity test, like enzyme activity test, polyphenol oxidase activity test, and catalase activity test. And the isolate I4 and R4 was showing a higher enzyme activity in these isolates. The species specificity study was done using ITS 1 and 4 primer to check that all the Fusarium isolates are belonging to the same family. Then we did the RAPD marker analysis, and in that we found around 43 polymorphic RAPD primers screening different isolates into various categories, and in these we obtained around 14 polymorphic bands in each isolate, and the highest polymorphic index was in 0.95.
No, you can conclude in 30 seconds.
Okay, ma'am. So the final conclusion is that the isolates which were collected from Gujarat and Rajasthan bifurcates themselves in different clusters, and this study is helpful for screening and validating further use in different transcriptome study or different plant breeding experiments also. So this is the conclusion. Thank you.
Thank you so much for this presentation, Poonam. We'll
Yes,
have
ma'am.
questions from panel.
Okay.
You have used RAPD, right?
Yes, sir.
So RAPD is what kind of marker? Is it dominant marker? Or?
Yes, sir. It's dominant marker.
So would it be able to select heterozygote?
Yes, sir.
Thank you so much, Poonam.
Thank you, ma'am.
So there are 3 participants who have not presented yet: Babita Chaudhary, Amita Mishra, and Savan Donga. Is there anyone present here? Okay, I think no one is present from them. So we have completed all the presentations. And now moving towards the concluding session of this event, the team Gujarat Vigyan Sammelan would like to express their gratitude towards all the dignitaries, including Shri Bhupendrabhai Patel, Honorable Chief Minister, Government of Gujarat, Shri Mansukh Mandavia, Honorable Health Minister, Government of India, Shri Jitubhai Waghani, Honorable Education Minister, Government of Gujarat, Dr. Kuberbhai Dindor, Honorable State Level Minister, Government of Gujarat, Shri Jayantji Sahasrabuddhe, National Organizing Secretary, VIVA, as well as Honorable Vice Chancellors from various universities for the support they have provided in organizing GVS 2022. Coming to today's event on biological sciences and technology, we would like to thank our panel discussion session experts, Dr. Arun K. Rawat, Dr. Sunil Raghav, Dr. Snehal Baktharia, and Mr. Akash Bhavsar for sparing their valuable time and knowledge for this event. We would also like to thank the expert speakers of different subject areas, Dr. Alok Dhawan, Dr. Bakulesh Khamar, Dr. Subir Majumdar, Dr. Subhas Jagsera, Dr. Mangal Rathod, Dr. Rukam Singh Tomar, Dr. Pratap Mukhopadhyay, Dr. Snehal Bakhtaria, Dr. Manish Rach, Dr. Manish Kumar, Professor Datta Madamwar, and Dr. Shailesh Dave for sparing time for delivering the talks and enlightening the attendees with their knowledge. This event was hosted by Gujarat Biotechnology Research Center, Gandhinagar. This would not have been possible without the support and guidance of Professor Chaitanya Joshi, Director GBRC, and Dr. Madhvi Joshi and Dr. Amrutlal Patel, Joint Directors of GBRC. We are also thankful to all the team members who were directly or indirectly involved in organization of this event. At last, we are also thankful to the participants of this event for making this event successful. The results of oral presentations will be declared on 5th February. Thank you so much everyone for your time and patience. With this, I conclude today's event. Jai Jai Garvi Gujarat, Jai Hind. Thank you.